Morning everyone and welcome to Extramarks live question paper discussion for NEET 2021 conducted yesterday from 2 to 5 o'clock. In this session, we are going to not just discuss the question paper, we will be giving the complete analysis of the question paper on different patterns so that you can have a better understanding about the paper and the pattern and what kind of questions they ask in the exam and how to approach the questions. We'll be giving you different types of approach, out of the box thinking, how you can smartly answer the questions. And in this analysis session, we will be giving you analysis on difficulty level of the question, how many questions were easy, how many questions were moderate, how many questions were difficult, overall what was the difficulty level of the question paper. We will also inform you about whether the question paper was NCERT based or questions in the paper were out of the NCERT, which questions were out of the NCERT, what was the cognitive skills required to solve the questions, whether the questions were just fact based questions wherein you have to remember, or it required some conceptual understanding to solve the questions, or there were some analytical skills also utilized or involved in solving the questions. So we'll be giving you complete detailed analysis of the examination paper that has been conducted yesterday. And myself, Lokendra, I'll be taking the zoology part, analyzing the zoology section of the question paper. And let me introduce our faculties here with us who will be discussing their respective segments of the question. So I'll start with calling up Mr. Kishore, who's our chemistry faculty, and he will be discussing the chemistry question paper, giving you the proper analysis. Kishore sir, please. Welcome sir.
Hello sir. Hello everyone. And if you talk about the chemistry part, as usual, as per the standards of parts that is taken care that the important chapters like chapters like aldehydes, and there were topics which were repeated, chapters like whole concept which were repeated. Chapter we have two from colligative properties as well as from Rolle's law. So they have taken care that they have touched each and every topic of all the chemistry altogether, and there were questions which were directly from NCERT. That is straight away from the lines of NCERT, which we always recommend our students to always follow the NCERT book from where they get questions always. So there are questions from NCERT, there are questions from applications over the NCERT lines, and there were a few questions which were out of the NCERT. I would not say out of the NCERT, from the basics of NCERT. So there were questions which were covered up all together.
Nice.
In chemistry, yeah.
So we can say, sir, that overall the question paper in chemistry was quite predictable along with the syllabus. What we advise the students about the important topics and important chapters, and
Yes.
the paper was mostly based on those parts. So it was a predictable paper.
Yes, it was completely predictable, and I'm very happy, and I think almost all the teachers who are the NEET teachers were very happier, because all the questions which I have seen in the exam were the questions which we usually give in our class, as sometimes like illustration, like in our live classes we usually give a lot of interactive classes, and we give a lot of illustrations in our class. So most of my questions were from illustrations, few of my questions were from homeworks which I give them, and few of the questions were from worksheets.
Oh, that's
So,
nice. So
yeah.
the students, your students must have been happy looking at the question
Yeah.
paper, and they would have jumped out of their seats.
Yeah, because
looking
all
at
the questions, like all the 50 questions which were there, were all from either from homework or from illustrations or from our question bank. So all the questions were covered up. Just the numericals were different. So the models were all same, but the numericals were different, and it's completely easier to moderate level for a student who was sincerely prepared for the NEET exam. He can score full.
Good to know, sir. Thank you very much for the insights. And
Okay.
now I would like to call upon. Now I would like to invite Mr. Goutham, who is an expert in physics over here at Extramarks. And Goutham sir, welcome
Hello,
here.
sir. Thank you,
it's
sir.
nice
Thanks
to
a
have
lot.
you here. And I would really want you to share with the students about the physics question paper that appeared in the NEET yesterday, and give them useful insights. What kind of difficulty level was the question, and how it would have, what would have been the experience of the students? What could it have mean?
Yes, sir. Coming to yesterday's paper, we found that the questions, the physics questions, it was lengthy actually. That is what many people said. And it was not like the last year paper. Here you had to solve to get the answers. So that was the case. And all the 50 questions that if you take from section A and section B, right? So most of the questions were asked from more than 50%, you can say. Definitely more than 60% or 60 to 65%. It was asked from 12th standard. So the paper has been designed keeping 12th standard students in mind, so that I can say. Now, like we used to see in the last year, right? So, they said that chemistry was easy, right? And most of the questions were from NCERT. That was not the case in physics. So you had to solve to get the answer. But I don't say that it was completely lengthy or something like that. So there were questions which were straight out of NCERT book, not many of them, but it was tricky. I can say the question paper was tricky. You had to solve it to get the answer. That is, you have to put your pen on the paper to solve and get the answer. In that case, it would have been easy. And yes, sir.
Also, we can say that physics was overall the tricky part.
Yes.
Compared to, I have seen the zoology part. It was easy and NCERT based. So physics was the, you can say, Achilles' heel, wherein the student must have faced some difficulties.
Definitely, so, sir.
Okay, so definitely when you will be discussing the physics section, we would just hope that you will be giving the students some tricks and tips to solve the question faster.
Definitely,
and
sir.
As you said, it was lengthy.
Yes.
And we have time restriction.
They felt it lengthy. The students felt it lengthy.
So, and we have a time limit on solving the question.
Yes.
So, I hope that the students will be benefited with your session, especially since physics was on the trickier part. And thank you so much for providing the insights.
Thank you, sir.
So now I'd like to introduce our botany faculty, Dr. Niranjan here. He's an expert in botany at Extramarks, and will get some inputs about the botany section of the question paper as well. Welcome, sir.
Thank you, sir.
And please enlighten the students.
Like Lokendra sir saying, this year the 90% of the entire NEET paper directly came from NCERT. We were all nervous due to Corona and the new pattern of exam, how the question paper is going to turn out to be. And especially the biology part, both botany and zoology, it seemed like everything is out of the NCERT. We keep on saying in the classes that think your NCERT book as a Bible. Read in between the lines, read all the lines, never forget. But if you see the paper, there is nothing called as read in between the lines.
Yes.
Everything was directly from those paragraphs, if you understand. And even those 10% where we talk about moderate and art, those questions were directly understanding, not even what we call as a applications level were there. And if you had prepared well from your NCERT paper, NCERT book, the entire paper could be solved easily in the time they had given this year. And even for zoology, I think Lokendra sir have already spoken. Except physics, the entire paper this year was somewhat 90% to 95% easiest compared to all our previous year papers.
Great. So we can safely conclude that biology especially was the most scoring section, the easiest section, and since it forms 50% of the question paper, the entire question paper, so students, you must have felt it very easy and scored maximum out of it. Then second was chemistry, and the most trickier part, as the students have felt and as teachers have analyzed, was the physics. It was not trickier but a little bit lengthy, wherein the questions needed to be solved and you had to work hard, students had to work hard to get to the answers. So now I would like Niranjan sir to continue with the discussion of botany section and enlighten the students. So sir, over to you.
Thank you,
I'll
sir.
take your leave. Thank you so much for being here.
Thank you, sir. Now we will have a detailed analysis of the paper and we will be discussing one part at a time. We will be starting with the botany, followed with zoology, chemistry as well as the physics. Now before we go for a discussion, let's now directly jump for all the questions analysis. First let us see how the pattern was and how the questions has been formed and how can we segregate them based on the easy to hardest paper. If you take the entire pattern this year, the questions from easy and moderate took close to 90%. That means there was hardly 10% of the difficulty questions this year especially. Like I already told you, we were all nervous thinking how the question pattern is going to be this year, especially how the question pattern is going to be this year. And this new pattern where the questions are 50 nowadays, and we have something called as a part A and a part B, where there was an option for us to choose between the 15 questions and we can write a 10 questions. Majority of you students were nervous before taking the answers. But luckily this year, except physics, even though physics we say that it was a lengthy, but if you solve the entire problems, you could achieve to your answer. But when it comes to chemistry as well as the entire biology, it was directly NCERT based, 90%. In that, we can easily say that 50% was easy. Majority of the students would have scored all the questions in this 50%, and moderate, close to 80% students would have attended. Difficulty was somewhere between to 8 to 10% in the difficulty analysis. And if we talk about the overall paper based on how many questions which had came from directly NCERT or which was completely understanding to NCERT or out of NCERT, or we can simply say that application level, we can say that the entire 90% of the questions which had arisen this year are directly from NCERT based. In every class, we keep on saying to students, always read your NCERT first, then go for other textbooks. NCERT is going to lay the foundations, right? We have to understand each concepts in a sequential manner, then only we can go for other understanding the concepts. So the 90% of these questions came from NCERT. Those who were thorough with NCERT book this year, they would have scored maximum amount of marks. Now let us see in which particular area do we have different kinds of questions which we have arisen. The three questions can be divided into three taxonomical hierarchy based on their level. If you directly see a question and if you already know the answer, nothing called as application understanding. There will be a question, and if you know the answer, we call this as remembering. In yesterday's NEET paper, the majority of the questions are what we call as a remembering type. Then we have a understanding. We have to use little applications for the understanding type. And finally, the 10%, the difficulty level, were directly by applying. Now, we would like to start the botany paper analysis. How the botany paper? If you ask me, the entire 50 questions which had arrived in botany, 44 to 46 questions are directly from your NCERT, and majority of them are remembering or either understanding. There were close to 3 to 4 questions which were difficulty, and you have to just use your head for a little bit to apply them and to come with an answer. Now let's directly jump to the questions, and we will discuss one question at a time, and we will understand how the answer is formed. So when we talk about the first question, a typical angiosperm embryo sac at maturity is. They are talking about a female gamete. They are talking about a female gamete in a flowering plants. They are talking about the structure of female gamete. Now the structure of female gamete comes from what we call it as a megasporogenesis. In a typical angiosperm, the megasporogenesis structure varies from one organism to another organism, and in this question they are asking about a typical. So in which majority organisms do we see majority of the embryo sac structure? So the question has 4 options. One says 7 nucleate and 7 celled, then we have 8 nucleate 8 celled, then we have 8 nucleate 7 celled, then we have 7 nucleate 8 celled. The typical embryo sac. This question has been directly taken from sexual reproduction in flowering plants from your 12th standard. Now, if you see and if you remember in your NCERT textbook, there is an entire explanation how the female gamete is developed, and we call it as a megasporogenesis. And if you see, there is a complete structure of this picture, and the picture of this megasporogenesis, if you take, there are 3 cells in the above structure, there are 3 cells on the below structure. We call them as a egg apparatus, where 2 synergids as well as the 1 egg cells will be there. On the other end, during a chalazal end, we have 3 antipodal cells, and in the center we have 2 polar nucleus. So typically we have 8 nucleus, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus, but if you count the cell, we have 1 cell, 2 cell, 3 cell, 4 cell, 5 cell, 6 and 7. So in a typical embryo sac, we have 8 nucleated 7 cell stage. This question is directly based from our NCERT. So this question is directly based from our NCERT, and when we think about what kind of understanding or we think about a remembering, it's all about remembering, and this question would have been attempted by more than 90% of the students. Now let us go for the next question. This question is match the list 1 with the list 2. So if you know answer for one option, you can easily answer for the remaining 3 options. So what we are going to do here is we will see, and all these in the list 2 are simple definitions of the list 1. This has been directly taken from the unit Plant Physiology and a chapter called Transports in Plant. If you can see in a list A, they have given some of the options like cohesions, adhesion, surface tension, guttation. Everything has to do with the transporting of mineral nutrients as well as water. So this comes in a transport in plants. Now if you see, there are list 2. So in this question, which is directly from our NCERT textbook, it is more about options, where exactly the questions is about the definition. If you see in the list A, we have what we call to say cohesion, adhesion, surface tension as well as a guttation. And if you see on the list 2, you can see the what are the definition for this particular option. Now let us see the options. The option A, option B, option C, option D. Now even if you know the answer for one of this particular concept, you can easily understand. So if you all know about a guttation is what we call it's a early morning. If you see a leaves, there is a tiny droplet of water which is coming from end of the leaves, and we call those structures as a hydrothodes. And this is a way how leaves will take out the water from the plant body, because during morning there is a photosynthesis, but during night there won't be a photosynthesis because light will be absent. But root keeps on pumping the blood. So roots keeps on pumping the water, and where exactly that the roots, when it keeps on pumping the water, it will go if there is no photosynthesis at all. That means this water has to be utilized or it has to be thrown out. Otherwise there will kind of pressure will be created, right? So guttation is also one of the method. And if you see your option D as a guttation and water loss in liquid phase, you can easily see water loss in liquid phase. And if you see the D as 3 options, so you have directly came for B and C answers. Now let us go for cohesion, adhesion and surface tension. When we talk about the cohesion, we are going to talk about attraction between the water molecule. When 2 water molecules are there, there is a bond created between, there is an attraction for the movement, and the cohesion directly go for the option B. So you can see in this again we have B and C both have the option 2 as well as the option 3. Then we go for next, adhesion. When you think about adhesion, what is adhesion? Adhesion is something what we call it as a attraction towards the polar surfaces. They have continuous attraction among themselves. So we can see adhesion comes to the attraction toward the polar surface, and our option will go to the option C is the right answer. Now let us go for next question. The next question is the plant hormone used to destroy weeds in a field is. These are the 4 different kinds of hormones which are formed not only in a plants as a natural, which can also be artificially synthesized. They are asking which of this hormone helps in removing the weeds. Weeds are what? What are weeds? Weeds are nothing but unwanted plants. What are weeds? Weeds are nothing but unwanted plants. In majority of this hormone, IBA, IAA, NAA, all these hormones are what we call the help in the development of the plant, both in shoots as well as the root. But there is one hormone called as a 2,4-D which acts as a weedicide. This question is also directly from NCERT. This question has been taken from Plant Physiology unit as well as a chapter called as a Plant Growth Hormone and its Development chapter. And this question, since it is directly from your NCERT, this is also directly from NCERT, and it is about understanding, because we have to understand each hormone, what their function, and close to 70% of the students would have attempted this answer as a right. Now let us go for the next question. Gemme are present in. Now what are gemme? Gemme are what we call it as a asexual reproductive organs. Where exactly these gemmes are found? These gemmes are found in a plant group called as amphibians, which we already know. Amphibians are the bryophytes. Amphibians of the plant groups are nothing but the bryophytes. Now let us see the option. They are found in some gymnosperms, some liverworts, mosses and pteridophytes. Since I have already told you, gemme are the asexual organs. Gemme are the asexual organs which are found in amphibians of the plant kingdom, that is bryophytes. That is bryophytes. So we are concluded that it comes in bryophyte. In our options we have gymnosperm, so this has to be wrong. Then we have pteridophytes, this has to be wrong. But some liverworts and mosses, both of them are the two groups of bryophytes. But we see Jamaicaps only in some groups of liverworts. It is completely absent in the group of mosses. It is completely absent in the group of mosses. It is seen in some liverworts. Again, this is also NCERT based question. And this is all about remembering. If the question was regarding the Jammeh, then it was about an understanding. Here we just have to remember where this particular organ is found. And this is also what we call as a, it will be attended by majority of them. So remembering, and 90% of them will got a answer right. Now let us see the next question. The next question is the amount of carbon such as carbon, nitrogen, phosphorus, calcium present in the soil at any given time referred to. Now, this question can be attributed to two chapters. This question can be attributed to two chapters. One, we can call it as a mineral nutrition, which comes under plant physiology. One chapter we can also attribute this into ecology unit. So, at any given time, a amount of nutrition found, amounts of nutrition like these nutrients found in a soil, any given time, what is it referred as? This is a definition. And the options are standing state, standing crop, climax, climax community. So at any given state, a amount of nutrition means it will be ready, means they will be ready to do. In Indian army, we call it as a standing army, which will be ready to fight for any kind of if enemy attacks, right? We call them as a standing army. So here, if we say they are ready means we come to two options. But here they are talking about a crop, we are going to talking about a state. Since we are not talking about a crop, directly you can remove all the answers and you can go for standing state. Climax and climax community are the terminologies we use in a ecology and in a particular concept called as ecological succession. That is the end of the plant forest which will be grown in both hydrarch as well as a xerarch community. So our answer is standing state. Since this is also direct definition and it is from NCERT and it is about understanding the difference between standing state and crop. So this is from NCERT, it is about understanding. Close to 70% would have written is right. There is a small confusion between you students where you would have gone wrong between standing crop and standing state. Now let us go for next question. The next question is, which of the following stages of meiosis involves division of centromere? Which of the following stages involves meiosis involves division of a centromere? There are different divisions will be happening in the throughout the phases where the chromosomes will be coming in the center. Sometimes they will be moving in a plane. Sometimes they will be separated in all the other ends. But in a one phase where the centromere will be broken down and these chromosomes will move apart to each poles, and that is what we call it as a anaphase. In each of these phase, the arrangement based of the arrangement of the chromosome movement of the chromosome, we have divided them into different phases for our better understanding. So in anaphase, this centromere, they will be involved in the division of the centromere. So this chromosome, one can move this side, one can move this side. That is where we can observe. So this is about understanding, and this entire system, how the meiosis works, is in our NCERT in complete 2 pages. All we have to do is understand. So again, it is from our NCERT, it is understanding. Close to 70% students would have written right. Why I am saying 70% it? When we talk about a centromere, certain times students think that centromere division will also take in what we call as a late metaphase. They think that. But it's always anaphase 2. That is where the chromosomal, the centromere, the division is going to be taking place. Next question. Next question. In the equation GPP minus R is equal to NPP, what does this R represent? Now it looks like a physics or mathematics formula. What this has to do with the ecology? Now, even though this question is in our NCERT, majority of the time students keep on ignoring this chapter. Majority of the students, they keep on ignoring the unit ecology. But in this year, there were close to 8 to 9 questions from ecology alone. That means you could count, it is easy 36 to 40 marks, easy 40 marks. Now, what is this formula? If you all remember a chapter called as a ecosystem in a unit ecology in your 12th standard, there is a concept called as productivity. How an organism produce, how can we calculate that? In productivity, we study two types. One we call it as a gross producers productivity and one was called as net productivity. Now gross means overall. If I am going to give you 10 rupees and if you are going to invest that 10 rupees and later on if you invest 10 rupees and you get 12 rupees in the outcome, overall is 10 plus 2. That will be our overall gross, right? But what will be the net? So that means when your plants are producing food, is the plant producing food without using anything? No, plant is also spending energy, right? How exactly they are spending energy? So that will be represented by R. So what is this R referred to? Environmental factor, respiration losses, radiation energy, retardation factor. R is the one which we use for a respiration. During respiration, what is the amount of ATP utilized? What are the ATP synthesized for plants to increase its biomass? So here we are talking about increase the producer's productivity is being increased in the biomass. To increase this biomass, what exactly these plants have taken in the surrounding environment? That is nothing but their respiration losses. R represents the respiration losses. This is also directly from NCERT, but we can consider it as a little advanced because majority of the time students will keep on ignoring this equation and only 50% students would have taken this as a right. Now let us go for the next question. Match the list 1 with the list 2. This is simply a structure of structures of reproductive or some structure of our photosynthesis organ. So they have given a parts of our organelles. We have to see where exactly these parts are found. If you identify one or two, it will be easy for you. They have given a parts of mitochondria, they have given a parts of chloroplast. So we have to check where exactly these organs are found. Now, you all know the structure. Of cytoplasm. When we write a cytoplasm structure, in cytoplasm, when we write a chloroplast structure, if you write a chloroplast structure, in this chloroplast you can see the arrangement of granas one after the another in a flat like structure, and they will be connected with one another grana connecting what we call it as a stromal lamellae. These connections and this grana is what we call it as a arrangement, a disc-like arrangement in one after the another. And you see, we call them as a thylakoids. And see where exactly thylakoids is and where exactly disc-shaped sacs. Disc-shaped sacs will be found here. So we can see like that. And you can see they are also flattened membranes because I told you they are found in chloroplast. But here you will get a confusion. There is a disc-shaped sacs in a Golgi apparatus and there is a flattened membranous sacs in the stroma of the plastids. At the same time, we have infoldings in mitochondria, primary constrictions in chromosomes. Let us see the option. Option A, option B, option C, option D. Cristae and cisternae. These are the other two where you will be getting confused with it. Now, when these discs are found in the chloroplast, that has to be closely related to the stroma of the flacids. So, our thylakoid B can be directly, the B answers can be directly involved with the 4. We will be getting A and D. But what about the remaining answers? There is a cristae, centromere as well as a cisternae. Now, they are asking primary constrictions in chromosome. Now, what are the primary constriction in chromosome? If you see, centromere will be in the middle as well as a chromosome in the, and so the primary constriction in the middle will become what we call as a centromere. So, C, we have to talk about 1, and if you take C as a 1, our option is going to become A. So, this is how you have to divide the right answers with a wrong answers, and you can elude whatever which is a wrong and you can come for. This is little bit of thinking question. You have to remember all the concepts. So this is what we call as a moderate question where a difficulty level is little more. Now, next question. The site of perception of light in a plants during photoperiodism. That means they are asking if there is a sun, where exactly the sunlight going to come and hit the plant. Photoperiodism is a technique where plant utilizes certain amount of light to produce the flower. So, where exactly light will be taken if flower has to be produced in a plant, which we already know, it's a direct answer. Options are axillary bud, leaf, shoot, apex, stem. Majority of them, they will go for axillary buds or shoot apex, thinking that flower is a modified shoot. A flower is nothing but a modified shoot, right? We call it the reproductive part. But where exactly the light will be taken for photoperiodism? It is always in the leaves. So this is also direct question from our NCERT based from a direct question from our NCERT based, all about remembering. Close to 90% of the students would have answered this question right. Next we have what we call which of the following statement is not correct. You have to remember this not word. Which of the following statement is not correct. Then we have what we call pyramid of energy is always upright. Pyramid of number in the grassland ecosystem is upright. Pyramid of biomass in a sea is generally inverted. Pyramid of biomass in sea is generally upright. This question comes from 12th standard from a chapter called as a ecosystem. In ecosystem, there is a scientist called as a Elton. He has given us something pyramids called as a Eltonian pyramids. Now what are these Eltonian pyramids? He has taken a 3 criteria to give us a Eltonian pyramid. These 3 criteria which he has taken to give us Eltonian pyramids are biomass, number and energy. So these 3 criteria, by taking this, when we take a different kinds of ecosystem, we keep on getting 3 kinds of pyramid. One is upright pyramid, second is inverted pyramid, and finally we have spindle pyramid. So in our ecosystem, these are the 3 pyramids which we can observe according to Elton. Hence we call it as a Eltonian pyramid. We call them as a Eltonian pyramids. Now let us take the options and we will see which is not correct. Now in these 3 pyramid, one of the criteria, if we take, the pyramids will be always upright, that is energy. If you remember, energy always goes from producer to the consumer. So it will be always upright pyramid. So wherever there is energy, if it is not upright, that should be wrong. But if you see here, they have written that pyramid of energy is always upright. That means this is a right statement. Next we go for pyramid of numbers in a grassland ecosystem is upright. So they are talking based on the numbers. In a trophic level, when we are talking about a trophic level, the lower will be taken by the producers. Then we have a primary consumers, then we have a secondary consumers, then we have a tertiary consumers. So when we take a grassland as an ecosystem, the first producers will be grasses, and they are talking about numbers. Of course, there will be millions of grasses. Are there going to be millions of lions? Are there going to be millions of elephants? No. But grasses, there will be millions. Since they are talking, there are millions of grasslands, so they will occupy the largest position. And all the other organisms which will come above them will be smaller. So again, they are talking, it will say upright. So this is also right statement. Then comes pyramid of biomass in sea is generally inverted. They are talking about the size. Pyramid of biomass in sea is generally inverted. Let's go for the next option also. Next option says pyramid of biomass in sea is generally upright. So we will take 2 options at a time. They are simply talking about the weight. Now think like this. There are small microorganisms like phytoplanktons and zooplanktons. We call them as a floating plants as well as a floating microscopic zoo animals. These plants will be eaten by these zooplanktons. It will be eaten by smaller fishes. Smaller fishes will be eaten by bigger fishes. Bigger fishes will be eaten by sharks. Sharks will be eaten by bigger another fishes. So if you keep on seeing that, the weight of the organism is increasing. So when the weight of the organism is increasing, that means our pyramid, when we take a biomass, it should be inverted. Because we have taken weight as a consideration in a sea. We are not talking about food. When we talk about the energy, it will be upright. When we are talking about numbers also, it will be kind of upright. But here we have taken what we call it as a weight. So our answer should be the pyramid of biomass in sea is generally inverted. Because we have taken biomass. Shark weighs more than the algae. As simple as that. Because sharks will come here. Algae will come here. Even though algae is a producer of food, but when we take a weight of a shark and as well as algae, who will weigh more? Fishes are going to weigh more. So pyramid will become inverted when we take a biomass. So this is the answer. So this is a little bit of what we call it as a application. You have to apply. This is also directly from NCERT and you have to apply, and close to 50% students would have got this answer right. Now let us go for next question. Amensalism can be represented as. See, again one more question from ecology. This is why I keep on saying to students, never ignore your last unit, which all the students will be doing this. We should never ignore the last unit. If you take the last unit, again this questions comes from the ecology unit from a organisms and population chapters. Now if you see in this concept, the amensalism can be represented. Now what is amensalism? It is a kind of interaction between two different species or two different organisms. What kind of? Let us see the option. Option A says that species A negative and B negative. Now what is this negative and what is this positive and what is this symbols I am going to use? I am going to say. See, if I use a negative, that means those organisms are getting affected. If I use a negative, that means those organisms are getting affected. If I use a positive, that means those organisms are getting benefited. And if I use O, that means neither they are getting affected, neither they are getting benefited, nor harmed. Neither harmed or benefited. So when there is an interaction between two organisms, if one of them is getting benefited or if they are getting harmed, based on this, the interaction has been divided into so many types, like a symbiosis, predation, parasitism, amensalism, commensalism, all this type. Question is, amensalism can be represented as. Here what happens in amensalism is, one neither get benefited nor get harmed, while other one will get benefited. I have already told you the symbols. Now let us see where one neither get benefited, harmed, while other is getting harmed. If you see option C, species B is getting neither benefited, neither harmed, but a species A getting harmed. This is a right answer. Shall we take one example? One example is, you can take an example of an elephant. Elephant keeps on walking, right? We will be elephant checking whether some ant is going there. I have to put my leg or not? No, it won't do. Will the elephant thinking there is a grass, if I press on that grass is going to die? No. That doesn't mean by elephant putting his foot in the front, it will go sad thinking I just killed an ant. It is a way of nature. At the same time, when elephants are walking, if ant dies, that has no advantage to the elephant, right? So we call this as a amensalism, where one will neither get benefited nor harmed, while another organisms will be harmed. So this is one of the type, and this is directly from your NCERT as well, but this is somewhat understanding question, so 70% of you would have got this question right. Now let us go for the next question. Next question. Which of the following algae produces carrageen? This is straight out of the NCERT. There is a big tabular column in a plant kingdom chapter. There they would have mentioned what kind of chemicals each plant groups has been given. Now we all know algae is the lowest plant groups in the plant kingdom, where we see all the primitive characters. Algae has been divided into three groups based on the pigments, which we already know. They are divided into three groups based on the pigments. We call it as a green algae, we call it as a brown algae, we call it as a red algae, or we have a names called as a chlorophyceae, phaeophyceae, rhodophyceae. Now let us see where this carrageen is found. Either it is a red algae, blue algae, green algae, or a brown algae. They are asking where exactly this carrageen will be found. Now green algae and blue-green algae, in our 12th we will be studying blue-green algae as a cyanobacteria, which we will be studying in kingdom Monera. Green algae is a chlorophyceae which doesn't produce carrageen. Then we have a red algae and brown algae. So our answer should be between the red algae as well as a brown algae. If you remember, red algae produces more of these chemicals which has a more economical importance, and as well as the brown algae. If you want to guess whether it is between a red algae and if you are a brown algae, the answer itself in the carrageen. It has to do with something a brownish color. It has to do with something brownish. Some of the red algae also produces carrageen, but brown algae produces majority of more carrageen compared to the red algae. And the questions belonging here is directly from our NCERT, and the answer is what we call say understanding level, and 70% of them would have written it right. Now let us go for the next question. When the centromere is situated in the middle of the two equal arms of the chromosome is referred. Now when we write this structure, so this is the centromere. This is our centromere. They are asking based on this position of centromere, where it is found, how chromosome can be divided. Based on the position of this centromere, they are asking what are the names given for the chromosomes. See, they are given submetacentric, acrocentric, C-metacentric, telocentric. All these are submetacentric, acrocentric, C-metacentric are where when the centromere keeps on moving this side or this side when we have unequal arms. We call them as arms, right? We call them as arms. When these arms are unequal and centromere is not exactly in the center, we can call them acrocentric, C-metacentric, submetacentric. If it is completely situated in the middle, that's like what we call a metaphase. During metaphase, we say, right, they form metaplate in the center, the same word meta, so it will be completely situated in the metacentric. So there is submetacentric, so our answer should be metacentric, see metacentric. And this is also directly from our NCERT, and it is a easy, it is a what we call it a remembering question, easiest question, 90% would have written it right. Next we have what we call a list from a and list from a 2. We have to see for a options between A and a B. We have to read here and we have to see. This can be again attributed to 2 chapters. We can study this in a reproduction in organism chapter as well as a strategies in your enhancement of the food, where we are going to study in a plant breeding chapter. Now let us see what are the options. Now options in the list A says that protoplasm fusion, plant tissue culture, meristem culture, micropropagation. So totally everything is talking about artificial vegetation. It is just talking about artificial vegetations where we are using tissue culture as a medium to produce new different kinds of plants. In the option 2 it says totipotency, pomato, somoclones, virus-free plants. Totipotency is a ability of each cell to produce a new plant. It is seen in all the plants. That is why vegetative propagation in plants is higher. Pomato is a cross between a potato as well as a tomato. Then we have somoclones, then we have virus-free plants. Even though if you don't know about pomato and somoclonals, just remember about totipotency and virus-free plant. Now if you see the options, option A, option B, option D, the first option you have to always jump is virus-free plant. Virus can be found only in plants when they have a vascular system. Virus can survive only in a vascular system. But if you want a virus-free plant, we can cultivate that in a laboratory condition without using this vascular system. We call those cells as a meristem culture. Meristems are a young cells. Meristems are a young cells which has no vasculature, vasculature at all. They are young stems which are actively dividing. That means meristem culture is a virus-free plant. So C for option C, where is the fourth? Our answer is going to be meristem culture, virus-free plant, and our option B will be like this. And why protoplasm fusion is used for somoclones? Then we have plant tissue culture, which we can go for totipotency. I just told you that ability of each cell is capable of dividing and producing a new organism at all is called as a totipotency. You take a small amount of leaf, you can grow entire plant. Take a small stick from a plant, pot it, you can get an entire new plant. How this plant is doing? This is what we call as a totipotency. Our option is D. This is also directly from your NCERT, but you have to understand. So close to 70% of the students would have got right. Now let us see the next question. During the purification process of recombinant DNA technology, addition of chilled ethanol precipitate out. Which of these will get precipitated out during the process of recombinant DNA? As the name states itself, there is a small crew. We are during a purification process of recombinant DNA technology. We want what to be chilled out. We want what to be precipitated out because we are doing recombinant DNA, right? Answer itself in the question itself, direct questions. Histones, polysaccharides, RNA and DNA. When we add a chilled ethanol, you can extract DNA. When you add a chilled ethanol for this experiment, you can extract a DNA. This is kind of remembering plus understanding. So close to 70% of students would have done this right. Now, next question. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. See, if the plant is found in desert condition or a xerophytic condition, you know about all this cactus, opuntia, right? Leaf has been modified into spines. Stem has been modified into such a way it produces photosynthetic activity. In the same way, plants have this ability to avoid these environmental external factors which are unfavorable to them and make them useful for them. What is this ability called as? This is directly taken from plant physiology chapter, and there is only one word, we call it as a plasticity. We call it as a plasticity, which you can observe in majority of the plants, where when they are young they produce different kinds of leaves. When they are adult they change those leaves into different kinds of things. So this is also directly a definition based question which is taken from NCERT and remembering, and close to 90% students would have written right. Maturity is about the cells maturing. Elasticity as well as flexibility, we will be talking about some cells in both animals as well as the plants. But plasticity is a condition where they look like different in the young and suddenly there will be a change and new kinds of leaves will be produced. Next, genera like Selaginella and Salvinia. Genera like Selaginella and Salvinia produces two kinds of spores. Such plants are known as. Now when we say two kinds of spores, it means both the male and female spores has to be different. When both the male and female spores has to be differentiated, we use a terminology called as heterospory. We use a terminology called as heterospory. If both the spores are same, we use a terminology called as homospory. Now, Selaginella and Salvinia are a pteridophytes. Selaginella and Salvinia are a pteridophytes. And for the very first time, homospory to heterospory change has started in pteridophytes itself. They are talking genera like Selaginella salvinia produces two kinds of spores. They have already given two kinds of spore. If we already know if there are two kinds, that is male spore differ from the female spore, it is a heterospory. So let us go for the option. They are saying homosporous. No, in a homosporous the condition is same. Then comes heterosporous, then comes homosporous, then comes heterosporous. So our answer is what we call it a direct answer, heterosporous. There has been small mistake in these options. I'm very sorry, but option answer is heterosporous conditions, and question belonging directly from our NCERT, and the tag will be for remembering. 90% of the students would have attended this. Next we have what we call it a which of the following plant is a monoecious. Now what is the difference between monoecious? What is the difference between dioecious? When we talk about a dioecious and monoecious, it has to do with reproductive part. It has to do with reproductive part. If both the reproductive part are found, if both the reproductive part are found in the same organisms, there is a certain words. We call them as a bisexual flowers. If only one kind of reproductive part are found in different kinds of organisms, we call them as a unisexuals. Here they have used three different kinds of plants and they are asking which of these following shows a monoecious characters. When we are talking about monoecious, dioecious means we are not just talking about a reproductive organs which are found. In the flower, sometimes what happens in some plants there is a male flower, in some plants there is a female flower. This also has to be taken into account. So Markantia comes in a bryophytes, Cycas comes in a gymnosperms, Carica papaya is our papaya which comes in angiosperms, Cara is a algae. They are asking which of the following plants is a monoecious kind of things. This question is directly taken from all the plant kingdom chapters and they are talking about monoecious. Now when we talk about Markantia polymorpha, in Markantia we have a male plant, we have a female plant. In the Cycas also we have a male and a female plant. In a Carica papaya also it has a male as well as female. In Cara we can see both the male and female reproductive in the same plant. We call it as a monoecious type of kind. The answer is Cara. This is again directly from NCERT and it is remembering. 90% of your students would have written it right. Now next question is mutations in plants cells can be induced by. How can the mutation be induced? Mutation can be induced by various methods. It may be chemical, it may be radiation, it may be prolonged evolution, etc. Let us see the options. Gamma rays, kinetin, infrared rays, zeatin. Kinetin and zeatins are the plant hormones which helps in the development of the plants. Infrared rays, even though it has a slight amount of what we call the affection on the mutation, but it has to be exposed longer for the infrared rays. But gamma rays is completely toxic. If any living cell which is affected to the gamma rays, it is going to change into mutation. I'm pretty sure you would have all heard about Hulk. When we see a movie called Avengers, you would have all about Hulk. A scientist will do experiment on gamma rays, you will get mutated to something else. But in a real life, it won't work. But when you see in a living organism, the mutation can lead a positive or a negative, but gamma rays can completely lead into what we call a mutation. This is also directly from the NCERT book, but you just have to understand to answer this. Close to 70% of the students would have answered this right. Next question is, which of the following are not secondary metabolites in plants? Plants produce various chemicals. We keep on extracting all these chemicals from plants each day, right? Where exactly they produce these various chemicals? Inside them. There is something called as a procedure, this physiological activity inside the plants. They produce chemicals to defend. Means they are producing this chemical to defend from other herbivores, predators who are attacking them. But we are extracting these chemicals for our own use. Now in plants, chemicals can be divided into primary metabolites, secondary metabolites, tertiary metabolites based on the chemical nature. Now they are asking in following which are not, you have to read, which are not the secondary metabolites. So the answer is vinblastine and curcumin. Vinblastine and vincristine are anti-cancer drug which are extracted from Catharanthus roseus, which is given during the chemotherapy. Curcumin is extracted from curcuma, which has various anti-cancer, antiseptic agent region. They are secondary metabolites. They are asking not secondary metabolites. Rubber and gums. Rubber and gums also comes from our secondary metabolite. Later on they get secreted and released outside when we damage the plant. So next we have what we call morphine and codeine. They are also secondary metabolites. Then finally we have amino acid and glucose. They are not secondary metabolite. One of them comes in a primary metabolite, one of them comes in a tertiary metabolite. So they are asking not. So this is our right answer. This is something what we call say understanding question. Even though it is from a NCERT, it is understanding, and close to 70% of the students would have written this right. Next question is, in spite of interspecies specific competition in nature, which mechanisms the competing species might have evolved for their survival? There are certain organisms, let us say we humans, we will be fighting, right? This land is mine, that land is mine. In organisms also in a nature, they will be fighting for a food as well as a land. You would have all heard about a territorial conflict of tigers. When two tigers come into contact in a close proximity, they will fight for their territory, to defend the territory. There are interspecific, means if they belong to this interspecific means that species, even though it changes, if they belong to same genus, there will be always this territorial fight for the land as well as a food. Now if they all came into conclusion, they have all evolved in such a way thinking that you take this place, I will take this place. Let's not fight. Both of us will be happy. We call this as a evolutionary mechanism, how they have evolved. And what we call this is a definition for what we call it as a resource partitioning. This is a direct definition. Resource means we have a land, this is a resource, you take this part, I will take this part, someone will take this part, so we will all be happy together, we will not fight. This is what we call a resource partitioning. Mutualism is where one organism lives with the other organisms for a benefit. Both of them are getting benefited. Predation is something like a fight between a tiger and a deer. And competitive release, in the competition, one of them will be always getting affected, one will be always lost. But in resource partitioning, all sat together, they spoke, and we have. So in animals also we can see that they have developed and evolved in such a way they don't have a conflict. We call it as a resource partitioning, which we can see in a nature. And this is also directly even though not directly from our NCERT, it can be taken from our NCERT from a chapter, ecology unit. And even though we call it as a NCERT out of the NCERT, it is little bit hard because of the concept, but it is understanding. Next question. Next question is, which of the following algae contains mannitol as reserve food material? They are asking about which of the following has mannitol as a reserve food material. Now, the mannitol is one of the chemicals. If you remember, in a previous year, they had asked about Floridian starch. In a previous year, the same algae they used, they asked about Floridian starch. This year, they are talking about mannitol. If we take all the options, we have green algae, we have a brown algae, and we have a red algae. And if you remember, mannitol is the something which is extracted from brown algae. Woolwox and woolothrix are green algae, which doesn't give mannitol. Then we have ectocarpus, which is a brown algae, which gives a mannitol. Then we have Gracilia, which is a red algae from which agar-agar is extracted. Agar-agar is extracted. So option is ectocarpus, which is a brown algae from where we extract mannitol chemicals as a reserve food. Now this is directly from our NCERT, which is just about remembering. Close to 90% of you would have answered this answer right. Next we have what we call the term used for transfer of pollen grains from anthers of one plant. So we have a one plant here, we have a flower which has a stamen, and we have one plant here. We have a stamen. So they are saying the process of transferring pollen grains, from one stamen to another stamen, the process of transferring pollens from one stamen to another stamen, another stigma from plant, but they are different plants. That is what they are saying. They are different plants. Pollen grain will be transferred from one anther to the stigmatic surface of the other plant. That means they become genetically different. So they are asking one plant to stigma of a different plant which during pollination brings genetically different. That means they are completely different. Types of pollen grain to stigma is chasmogamy, cleistogamy, xenogamy, gaetnogamy. Plasmogamy and cleistogamy, we call them as a, based on if the flower is closed or if flower is open, so they are talking about flower, whether it is a open or a closed. Xenogamy and gaetnogamy is about a fertilization process. In a gaetnogamy, it is between the same plant, same flower, it may be different flowers where it will be coming, but they are genetically identical. Even if it is the same plant, the pollen grains come from here and it may go for this flower, but still it is from the same plant, so genetically, so this is wrong, and this both are about a flower opening and close. Xenogamy is where pollen grains from two differently genetically identical, genetically different, where the pollen grains will be transferred for the fusion, and we call it as a xenogamy. Directly from our NCERT, it is about understanding. Close to 70% would have right. Some of you would have got confused with gaetnogamy. Next question. Didelphous stamens are found in. What are didelphous stamens? When we take a plant as an example, if this is our receptacle, this is our female reproductive organ, stamens will come here, right? When stamens are free like this, these are two, they are separated, we call it as a didelphous conditions. Certain times what happens is, bunch of stamens will be together, where their filament will be fused. Anther will be free while one stamen will be upright. They are asking didelphous stamens are found in which of the groups? Either P, China rose, citrus, China rose as well as a citrus. Now, where exactly do we see where one bunch of stamens will be on the other end and one or two will be separated? This is a specialized character of a family Fabaceae or Leguminosae. And you can see in the entire option there is only one plant which belongs to Fabaceae, that is a pea plant, same as a groundnut family, where we can see didelphous condition like 9+1 or sometimes 9+2 or sometimes 10+1 or sometimes 13+1 stamen conditions. And we call this directly from your NCERT, where the differentiation between didelphous, monodelphous, NCERT, it is about understanding. Close to 70% of you would have gotten this right. Then we have what we call say DNA strands on a gel stained with ethidium bromide when viewed under UV radiation. Normally, ethidium bromide is used in the process called gel electrophoresis. We will use in a process called as a gel electrophoresis as well as a DNA fingerprinting studies to determine where these DNA strands are formed. So we will use under a UV radiation, when those gel is treated with ethidium bromide, what color it is going to appear. If you have all seen the books, if you have seen the colorful pictures, you can see the entire gel will look somewhat bluish color, but these structures which are formed in the middle, these strands which are formed in the middle, they look bright orange bands. So it is bright orange band. When you use a ethidium bromide, we get a bright orange band on this gel. This is directly taken from our NCERT, but it is about understanding. It is about understanding what kind of and where we have to see all those questions. Close to 70% students would have answered this question. Next, we have what we call to say DNA fingerprinting involves identifying differences in the some species specific region in DNA sequence. Now, DNA fingerprinting works on only one concept. That is what we call as a repetitive sequence, or we call them as a VNTR. Variable number tandem repeats, which are varies from one person to another. Even if you see the DNA with your parents will have different VNTRs, you will have different VNTRs. So the variable number tandem repeats varies completely from one person to another. Using this, we have what we call as a DNA sequences is called. So they are asking what exactly. So these are repeated sequence. Let us see in option. We have single nucleotides, polymorphic DNA, satellite DNA, repetitive DNA. Satellite DNA is also what we call for a large amount of bases when their repetitive sequence are found together, but the entire DNA fingerprinting techniques works on VNTR or repetitive DNA, again directly from NCERT, and all you have to remember is understand, and 70% of the students would have done this answer right. Next, we have match list 1 with the list 2. They are asking about the bonds. What kind of bonds can be seen with the list A with the list 2? If you see here, we have a proteins and we have unsaturated fatty acid, nucleic acid, polysaccharides, etc. Let us see what kind of bonds will be found. Now, if you see all this protein, unsaturated nucleic acid and polysaccharide, we will have different kinds of bond. But nucleic acid, when we are talking about nucleic acid, there is something called as a glycosidic bonds. There is something called as a glycosidic bonds. There is something called as a phosphodiester bonds. There is also something called as a hydrogen bonds. All these three bonds are seen. But when it comes to nucleic acid, we will be seeing what we call as a glycosid. What about proteins? When proteins are formed, the peptide bonds will be formed. So A is what we call it as a 4, and we got B as well as a C. When nucleic acid becomes 3, so our answer will be directly B. Then comes what we call as a C. Then comes what we call it as a, one second students. So protein comes for our peptide bonds, where we will be having a bonds between two amino acids. Polysaccharides will come for what we call as a phosphodiester bonds. Polysaccharides will come for what we call as a phosphodiester bonds. D comes to, so our option will go for what we call it as a, one second, nucleic acids will go for phosphodiester bonds. B1. So our option will be C, because the unsaturated fatty acids will always give us carbon-carbon double kinds of bonds. In the exponential growth equation, Nt is equal to N naught e to the power of Rt. E represents the base of natural logarithms, the base of geometrical logarithms, the base of number logarithms, the base of exponential logarithms. So they are asking what exactly E gives for. The E gives for the base of natural logarithms, which we are using in ecology system to calculate the exponential growth as well as the logarithms. Next questions. Match list 1 with list 2. Here they have given certain names, we have to match with list 2. We have a S phase, we have a G2 phase, we have a quiescent stage, we have a G1 phase. In a S phase, what exactly happens? That is DNA replication happens. In G2 phase, what it happens? In quiescent stage, what is happens? In G1 phase, what? In G1 phase, we already know proteins are would be synthesized. Let us see the options. So D means it has to be 1. So there is two options with a D, 1 and 1, B and D. Let us see S phase. Interval between mitosis and initiation of DNA replication is there. That is interval means we are taking a break, that makes quiescent stage. So C has to be 3. See, if C has to be 3, you can see both of them again have the C has to be 3. And let us see for the next. S phase is DNA replication. A goes for 4, and you can see the option is D, where in S phase, a synthesis phase, DNA replication is going to be taking place. Then we have what we call which of the following statement is correct. ATP is synthesized through complex V. They are asking correct. Oxidation, reduction reactions produces proton gradient in respiration. Then we have during aerobic respiration, role of oxygen is limited to the terminal stage. In ETC, electron transport chain, one molecule of NADH2 and gives rise to 2 ATP molecules and 1 FADH2 give rise to 3 molecules. This, what they are saying about, this is wrong because NADH2 gives us 3 ATP, FADH2 gives us 2 ATP. This is wrong, but they have asked for correct sequence. Then they are asking during aerobic respiration, role of oxygen is limited to the terminal stage, and the oxidation reduction reactions produces gradient in respiration. ATP is synthesized through the complex V. Yes, all these are of these options are wrong. ATP will be synthesized through the complex V, then it will go to oxidative phosphorylation. This is again from plant physiology, which is comes under understanding. 70% would have gotten it right. Then we have what we call what is the role of RNA polymerase 3 in the process of transcription in eukaryotes. What is role of RNA 3? The role of RNA 3 in the transcription on eukaryotes is it produces tRNA, 5S sRNA as well as a snRNA in the process of for splicing as well as a capping technique where snRNA is required. This is about understanding a direct answer, and 70% of you would have gotten this right. Then we have the next. I would like to call zoologist Professor Lokendra sir. I hope you would have all done the entire botany concept in the easiest way, because majority of them has came from NCERT. Majority of those questions has come from NCERT. Now I would like to call Lokendra sir to further discuss the zoology pattern, where you will be talking about the entire zoology where all the questions, how they have came from NCERT, whether they are related to our NCERT or applications, all those concepts will be spoken by Lokendra sir in a few minutes. When you are writing any paper, when you are writing whether it is a NEET or CET, we all have to remember one important concept, that is the major concept what you have to remember is about, it comes from our NCERT first, then you have to go for other textbooks. Without going for NCERT, we cannot jump directly into the other textbooks. Read NCERT completely, then go for other textbooks where you can identify what is the difference between here. First, you have to get your concepts ready. Once you get your concepts stronger, answering all these questions would be a fine answers for you. And I would like to welcome Lokendra sir to take further about zoology concepts. Thank you. Is going to continue with a zoology concepts. Thank you, sir.
Thank you so much, sir. So, welcome everyone to the NEET discussion session. And in this session, I will be discussing the part of zoology and talking about the overall analysis of the zoology section. Most of the questions were NCERT based and direct questions from NCERT. Most of the questions, if we talk about the cognitive skills required to attempt the question to get to the answer, it was memory based paper mostly. A few questions required a little bit of understanding of the concept, and just a couple of questions had the application part in this section. So zoology section overall was easier compared to previous years as well as compared to the other sections, and students would have been able to score most of the questions in zoology. So without wasting further time, let us start discussing the questions which appeared in zoology. Now the first question here, it says that the chronic autoimmune disorder which affects the neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscles, right, is called as, the first option they has given is myasthenia gravis, the second option gout, third one is arthritis, and the fourth one is muscular dystrophy. Now the question says that it is an autoimmune disorder. In autoimmune disorders, the antibodies which are formed in the body, they start attacking our own body cells. Is not an autoimmune disorder, so this option is wrong, right? It has arthritis like symptoms, but it is due to deposition of particles in the junctions, at bones. Muscular dystrophy, this is a genetic disorder which causes degeneration of muscles. This is also not an autoimmune disorder. So this option is also wrong. Now we are left with two autoimmune disorders, myasthenia gravis as well and arthritis. Now rheumatoid arthritis, this is an autoimmune disorder because of formation of rheumatoid bodies which starts attacking the major joints, especially the knees, and the synovial fluid which is present in these joints becomes hardened and mineralized, causing pain. So arthritis also does not lead to fatigue, weakening and paralysis of skeletal muscles. It is due to thickening of the synovial membrane and the synovial fluid. So this option is also wrong. Myasthenia gravis. Myasthenia gravis is an autoimmune disorder wherein the antibodies, they start attacking the neuromuscular junction, decreasing the nerve transmission at the neuromuscular junction. So the mobility of the muscles, the activity of the muscles decreases and ultimately leads to degeneration of the muscles, resulting in paralysis. So the question was simple and memory based. In the autoimmune disorders, we have myasthenia gravis, arthritis, two important examples with the symptoms. So this question, the first question that we have discussed, mostly memory based, that we have to remember and directly from NCERT. Now, so if we can say the question belonging is NCERT, right? And if we use the cognitive skills, then the question is mostly remembering or memory based questions, right? And most of the students who had been familiar with autoimmune disorders and remembered the symptoms correctly, right? So according to me, almost 70% of the students must have answered this question easily. Now the next question, right? Which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? Now in this, the question is asked from meiosis 1, the prophase 1, right? Because it talks about terminalization of chiasmata. Chiasmata is formed when the non-sister chromatids of homologous chromosome exchange their parts, right? That junction is called as chiasmata. So the question is directly from NCERT, from cell division meiosis prophase 1. And if we see the stages of prophase 1, right, the 4 stages which are given here as the options are diakinesis, pachytene, lactotine and zygotine. In prophase 1, when the recombination and crossing over takes place, right, prophase 1 is further divided into sub-stages. The first stage is lactotine, which is also called as the bouquet stage, where the chromosomes appear just like a bunch of flowers. The second phase is zygotine, and zygotine is characterized by pairing and synapsis. So the homologous chromosomes, they form pairs in zygotine stage. Pachytene stage is characterized by crossing over. The non-sister chromatids of homologous chromosomes, they form the chiasmata. So chiasmata formation starts in pachytene stage. But the question is asking which phase shows terminalization as a distinctive feature. After pachytene, the next stage is diplotene, where the chiasmata starts sliding down and starts unzipping. While the last stage, diakinesis, clearly marks the end of chiasmata and the 2 homologous chromosomes, and the non-sister chromatids of homologous chromosome, they again separate from each other. So the correct answer here for this question will be diakinesis. Again, the question is NCERT based and based on memory. Most of the students must have answered this question correctly. Now coming on to the next question, venereal diseases can spread through, and they have given us some options. What are the mode of spreading of venereal disease, whether it is using sterile needles, transfusion of blood from infected persons, infected mother to the fetus, kissing and inheritance. And the options, the question was to choose how many of these methods, modes of spreading of venereal disease are correct. Now venereal disease, they are sexually transmitted diseases. So sexually transmitted disease are also known as venereal disease. Now if we are using sterile needles, if the using of sterile needles will not spread the disease, only contaminated needles, needles which have already been contaminated with an infected person's blood, can spread the disease. So the first method is wrong. Transfusion of blood from an infected person. Of course, if the person is already infected with a venereal disease, and from the infected person, if a healthy person is given blood, right, or the blood of the infected person is transfused to a healthy person, the disease also is transferred and the healthy person is infected by the same disease. Yes, this is a mode of transmission. Infected mother to fetus, right? The next option they have given is infected mother to fetus, kissing and inheritance, right? Again, let us come kissing, right? Venereal disease, if we just have to answer, let's take the example of AIDS, which is the most common sexually transmitted disease, right? And we say that kissing, touching the patient does not spread the disease. This is also not the right answer. Inheritance, are the venereal diseases inherited? No, they are pathogen related diseases. They are not genetic disorders. Only genetic disorders can be inherited from parents to the offspring. So this is also wrong. Infected mother to fetus, right? Infected mother to fetus, yes. The disease like HIV, it can be, it has very low chances, but the disease can be transmitted from infected mother to its fetus via placenta. So there are only two modes which are correct for spreading of sexually transmitted disease, and according to these, the answer is the option A, B and C only. Now, if we look at this, even if some students could not have guessed all the eliminated all the methods of transmission of sexually transmitted disease, we know that transfusion of blood from infected person definitely will spread the disease. So B is definitely the correct answer. So if B is the right method, that means option 2, which says that only A and C are correct, this is wrong, right? And we have already eliminated that using sterile needles. If we are already sterilizing the needles, right, making them pathogen free, that means the pathogen cannot be transmitted. So A is wrong and B is correct. These two facts we have established, right? So now this option is also wrong because it says A is a method through which the disease can spread, right? So wherever option A is present, these two are wrong. Now we are left with only two options, right? B and C only, B, C and D only, right? Now if we see D, kissing. Does kissing can transmit a venereal disease, sexually transmitted disease like HIV or gonorrhea? No, it comes only when the bodily fluids from the genital organs, especially the blood and the plasma, are exchanged between the two infected persons. So that means D is also not a correct option. So this option is also wrong and we are left with the one option. So if you could have smartly guessed, the students could have smartly worked around the options. We already eliminated two options because they say that A, option A, use of sterile needles is a mode of transmission. This is obviously wrong. Now we had to just find out about option D. We have to check because B and C are common in both the options. That means logically B and C must be correct. We have to check only about the fourth mode, that is kissing, which is not a mode of transmission. So the solving this question was easy, right? And the question was also directly on memory based, right, and NCERT based, right? Almost 50 to 60% students must have answered these questions, and those who would have worked smartly around the options, right, they could have easily guessed the answers and got the answers, saving time. Now, moving on to the next question. The question says, which is the only enzyme, right, highlighting the part only enzyme that has the capacity to catalyze initiation, elongation and termination, all the three processes in the process of transcription in prokaryotes. The enzyme, the question is asking, the enzyme which catalyzes all the three processes of transcription, initiation, elongation as well as terminalization or termination in case of prokaryotes. Now the four options given, we have DNA ligase, the second option DNase, the third option DNA dependent DNA polymerase, and the fourth option is DNA dependent RNA polymerase. Now, again, working with the options to find out the answer. The process is called as transcription. The process is transcription, and the process of transcription means formation of mRNA from DNA. Using DNA as a template, mRNA is being synthesized. DNA ligase, the word ligase, these are joining enzymes, right? Ligase means joining, ligation. The process of ligation means joining, and the ligase enzymes, they catalyze joining of two molecules. If we say DNA ligase, this means this enzyme will help in joining of two molecules of DNA. DNase, these are digesting enzymes, the enzymes which digest DNA or break DNA. What we are talking about is synthesis. We are not talking about breakdown or digestion. We are talking about synthesis of RNA. Now, synthesis will be catalyzed by polymerase enzyme, because both DNA and RNA, they are polymers of nucleotides, and to synthesize a polymer we require an enzyme polymerase, right? So a polymerase enzyme is required. So that means option 1 and 2, A and B options, they are obviously wrong. The third and the fourth options, they both have polymerase enzymes. Now what we have to synthesize, it clearly says that we are talking about transcription, that means synthesis of mRNA using DNA as a template. That means we require the enzyme RNA polymerase, because we have to synthesize RNA, right? And there is only one option which says RNA polymerase enzyme. Now, what does this mean by DNA dependent RNA polymerase? It means, now the answer is correct. The fourth option is obviously the correct answer, because since we are synthesizing RNA, we require a polymerase enzyme, and since it is RNA synthesis, we require RNA polymerase. If it is DNA synthesis, then we will require DNA polymerase enzymes. It's very obvious. But what is the meaning of these two terms, DNA dependent DNA polymerase and DNA dependent RNA polymerase? DNA dependent DNA polymerase. Now let us understand the meaning. DNA polymerase, that means there is synthesis of DNA. DNA dependent, that means this enzyme uses DNA as a template. Using DNA as a template, the enzyme synthesizes another molecule of DNA. Similarly, in the fourth option, using DNA as a template, the enzyme synthesizes RNA. If we say RNA dependent DNA polymerase, there is one more enzyme which is called as RNA dependent DNA polymerase. So what we are synthesizing here is DNA using RNA as a template. So from RNA, if we are synthesizing DNA, this process is reverse transcription, and this enzyme must be the reverse transcriptase enzyme. So the naming of the enzyme is based on what is the template it is using for the synthesis and which molecule is being synthesized. So since the process was transcription, and we know transcription means synthesis of mRNA using DNA, so the enzyme should be DNA dependent RNA polymerase. Or even if we don't know what is DNA dependent RNA polymerase, we don't know about the two terms, the first two terms. We simply know that the synthesis of RNA requires RNA polymerase enzyme, and there is only one option which contains the RNA polymerase enzyme. So eliminating three obvious answers and getting the only option with RNA polymerase, that is the correct answer. So again, the question is memory based and directly from NCERT, because the main transcription enzyme is RNA polymerase enzyme, right? So memory based question directly from NCERT and easy. So almost all of the students must have answered this question correctly. Moving on to the next question. The question says sphincter of Oddi is present at. This question is from digestion and absorption, human physiology, right? The four options: gastroesophageal junction, junction of jejunum and duodenum, ileocecal junction, and junction of hepatopancreatic duct and duodenum. Sphincter of Oddi, it is present at the junction of or at the opening of hepatopancreatic duct into the duodenum. The part of the duodenum where the hepatopancreatic duct combines with the, what happens is the hepatic duct from the liver and the pancreatic duct, just before opening into duodenum, they combine together and form a common hepatopancreatic duct. This opening of common hepatopancreatic duct into the duodenum is guarded by sphincter of Oddi. So the question was memory based question, again directly from NCERT, wherein in the pancreas section it is clearly written in the structure of digestive system that the hepatopancreatic duct, the opening is guarded by sphincter of Oddi. NCERT based, simple, easy question, memory based questions, but not all the students must have been able to attempt this, because remembering the names, right, there are two sphincters here, sphincter of Boyden as well as sphincter of Oddi. Sphincter of Boyden is present only in the hepatic duct and sphincter of Oddi is present on the common hepatopancreatic duct. Moving on to the next question. Erythropoietin hormone, which stimulates formation of RBCs, is produced by. The options given are the cells of bone marrow, juxtaglomerular cells of the kidney, alpha cells of pancreas, and the fourth option, the cells of rostral adenohypophysis. Now, erythropoietin, this hormone initiates or stimulates the formation of RBCs. Production of RBCs or formation of blood, erythropoiesis, the formation of RBCs, formation of blood, hematopoiesis. This process occurs in the bone marrow. So the cells of bone marrow, they complete the process of erythropoiesis or hematopoiesis. They do not secrete the hormone which stimulates. This hormone, erythropoietin hormone, it stimulates the cells of bone marrow to start producing RBCs. So the cells of bone marrow, they do not release this hormone. So the first option is wrong. This hormone is released by the kidneys. The juxtaglomerular cells present in the kidneys, in response to hypoxia or decrease in level of blood in the body, they secrete the hormone erythropoietin, which stimulates the bone marrow to start the process of hematopoiesis, to start synthesis of more RBCs. Wherein the alpha cells of pancreas, they secrete the hormone glucagon. It is related with digestion. The cells of rostral adenohypophysis, this is pars distalis, the anterior pituitary gland. And we know that anterior pituitary gland, the hormones released by anterior pituitary gland are FSH, LH, right? Gonadotropin stimulating hormones, thyroid stimulating hormone, growth hormones, right? But it does not release erythropoietin. So the question was moderate level I cannot say that question was difficult, it was moderate level, but yes, students must have been confused because erythropoiesis generally is studied in circulation, body fluids in circulation, but the hormone, erythropoietin hormone, this is secreted by kidneys. So here the erythropoietin hormone secreted by kidneys in the excretory system. So the question was slightly moderate level, right? But again, based on NCERT itself, it is not from outside the NCERT, right? Again, the cognitive skills required, it is purely memory based questions, but most of the students must not have been, would have faced difficulty in answering correctly. So the correct answer here is the JG cells, the juxtaglomerular cells in the kidney. Now, going to the next question, we have a list given and we have to find out the correct match in this list, right? The 4 options have been given here. Physalia, right? Physalia, a coelenterate, commonly known as the Portuguese man of war, is the most common example, most commonly asked in the classes also. We discuss, when we discuss the examples of coelentrates, class Hydrozoa, Physalia, the common name is very popular and has been, it's directly given in NCERT. Physalia is known as the Portuguese man of war. So for A, the correct match is option, is the second option from list 2. Now if we see the options, right, let us try to solve this question smartly. If we see the options, A is matched with 2 in only 2 options. The fourth and the second options are incorrect because we have already established Physalia is living fossil. Now we have to again match one more to get the correct answer. Now Limulus, the horseshoe crab, Limulus, the horseshoe crab in arthropods, it is a living fossil. It is a living fossil, again very famous example in the phylum arthropod. Limulus is a living fossil, so for B it is third, right? In both the options, B is matched with third, so still we have not got to the answer, right? Better if we try to match C and D, because either one of these, if we match, definitely we will get the correct answer. Encyclostoma and Pinctada. Pinctada is the pearl oyster, right, from phylum Mollusca. Again, a very famous example. The oyster industry is dependent, the most cultured mollusk for formation of pearls is the Pinctada, right? So if we match option D with first one, right, and D out of the first and third option, D is matched with first one in only one of the options. So in this, if we want to attempt the question smartly, we look at the matchings, right? We matched first one because Physalia, commonly known as Portuguese man of war, is a very easy and most common example given. And out of, once we have matched the first one, Physalia, we have eliminated 2 options. Now in the remaining 2 options, rather than going for matching the Limulus, B, because it is common, so it is given in both, so we can directly match it. That for B, correct matching is third. Even if you don't remember whether the Limulus is a living fossil or not, it is given in both of them. In both the correct options, it is matched with the third one, so it is a living fossil. Now Encyclostoma is hookworm in the nematodes, while Pinctada is the pearl oyster. So whichever you find easy. If you match anyone out of C and D, you will get the correct answer, right? So here the correct option is option A. Question is easy, memory based, directly from NCERT examples. However, since they have mixed different phylums, so from invertebrates, from all the phylums, the important examples the student must have been able to remember. but the question was not on the difficult part of it. We can say for some students who must have not memorized all the phylums and the important examples, they would have find it difficult, but I can say that the question was between easy and moderate difficulty level, but again based on NCERT only. The next question here is an interesting one. If adenine makes 30% of the DNA, what will be the percentage of thymine, guanine and cytosine in it? Now this question requires a little bit of understanding as well as application both, right? The question has said that adenine makes 30% of the DNA molecule, and it has asked what is the percentage of the remaining 3 nitrogenous bases, nucleotides, thymine, guanine and cytosine. Now in DNA, we know that there are complementary bases. Adenine always forms hydrogen bonds with thymine. So adenine always forms 2 hydrogen bonds with thymine, and guanine, it always forms 3 hydrogen bonds with cytosine. So guanine and cytosine, they are complementary bases, and adenine and thymine are complementary bases. Both adenine and guanine, these are the purines, and thymine and cytosine are the pyrimidines. So we can say that the ratio of purines and pyrimidines is also equal in DNA. That is the Chargaff's base equivalency rule. But here the question has asked the different percentage. Now, if 30% of the DNA contains adenine, and adenine always forms bond or always pairs with thymine, that means if there is 30% adenine, thymine should be equal to adenine. So in the remaining 70%, we have 30% thymine. Now amount of guanine and cytosine should also be equal, because whatever is the number of guanines, same will be the number of cytosine, because guanine always pairs with cytosine. So if 30% is adenine, that means thymine also must be 30%. So 60% of the DNA is made up of adenine and thymine. In the remaining 40%, guanine and cytosine are present, and we know that amount of guanine will be equal to amount of cytosine because they are complementary base pairs. So that means both guanine and cytosine should be 20% each. Right? So this question, this required a little bit of understanding the structure of DNA and also the application part of the concept. So here the correct option will be thymine 30%, and even if the student does not remember the application part or was not able to go for the application part of it, right? Still you could have solved this question. How? Because when you know that adenine always pairs with thymine, so amount of adenine and thymine must be equal, right? So if adenine is given as 30%, thymine also should be 30%, and as we can see in the options, there is only one option which says that thymine is 30%. All the remaining 3 options, they are obviously incorrect. So there was no requirement, there was no need to solve this question in the exam, because the answer was obvious, as any student who knew that adenine and thymine are complementary bases and guanine and cytosine are complementary bases, he would have solved this question within fraction of seconds, because if adenine is 30%, thymine also must be 30%. The remaining 3 options says thymine is 20%. Obviously these are wrong, right? Let's move on to the next question. Question says that the fruit fly, the Drosophila, it has 8 chromosomes in diploid state, right? The number of chromosomes to end in the diploid state is 8 in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, right? What would be the number of chromosomes in the S phase? 4 options are given here: 4, 32, 8 and 16. Now here a little bit of understanding was required in this particular question, right? If we see the cell cycle is divided in 2 parts: interphase and a division phase. The interphase part or the M phase, it can be mitosis, it can be meiosis. The remaining part is interphase. Division phase, either by mitosis, the cell can divide mitotically, or the cell can divide meiotically. The remaining part, interphase, the longest phase of cell cycle, this is further divided into 3 stages: G1, S and G2. S phase, this is the synthesis phase, and hence the name is S phase. In the synthesis phase, the name itself says that there is something being synthesized. In the synthesis phase, DNA replication, the DNA is replicated. So each chromosome at the end of the S phase, once DNA replication is complete, each chromosome, it contains 2 chromatids. Each chromosome will contain 2 chromatids, but the number of chromosomes will not change. The number of chromosomes will not change. The chromosome has just duplicated. The amount of the genetic material, the overall amount of the DNA has doubled, but number of chromosomes is still the same. So even after S phase, number of chromosomes will increase, right? Both the chromatids, they will separate only during the division phase. Before that, even after duplication of the chromosome, both the chromatids are attached together with the centromere. So the number of chromosomes will not change. That means at the end of S phase, after the S phase also, the number of chromosomes will remain same, right? So this particular question required a little bit of understanding. Again, the question is from NCERT, but a little bit of extrapolation or using the concept from NCERT to get the answer was required. So some level of thought process, right, was expected from the student. But again, the question is in the moderate category. It is not difficult question. Now, coming on to the next question, we have person with AB blood group. The question says that persons who have AB blood group, they are called as universal recipients. And what is the reason for calling them universal recipients? When we say universal recipients, it means that they can receive blood from any blood group. Now the 4 options that were given: presence of antibodies, anti-A and anti-B, absence of antibodies, absence of antigens A and B on the surface of RBC, and absence of antigens A and B in the plasma. Now, here again the student required a little bit of understanding of the concept. So we can say moderate to difficult, slightly shifting towards the difficult side. The basis of blood transfusion is based on the presence or absence of antibodies in the plasma. If the blood from the donor, which is coming in the body has the same antigen, has the same antigen, then it is recognized as self by the body, by the recipient, and the recipient does not synthesize any antibodies against it. Now we say the blood groups, the 4 different blood groups that we have, A, B, AB and O. They are defined on the basis of presence and absence of antigens. So in blood group A, antigen A is present. In blood group B, antigen B is present on the surface of RBC. In AB, both antigens A and B are present on the surface of RBC, and O does not have any antigen. So for blood group A, A antigen is the self antigen and B becomes the foreign. So if we say the antibodies, here anti-B antibodies will be present. For blood group B, B is the self antigen and A becomes foreign. So this person will have anti-A antibodies. For blood group AB, both A and B antigens are self antigens. So that means there is no foreign antigen for this. So here antibodies are absent. And for blood group O, since there is no antigen present, that means both A and B antigens are foreign antigens. So both anti-A and anti-B antibodies are present. So now AB blood group is universal recipient. Why? Because there is no antibodies present. If there is no antibodies present, that means there will be no antigen antibody reaction between the donor blood and the recipient's blood. Because and for antigen antibody reaction, we require both antigen as well as antibody are required. Isn't it obvious? So if both the antibodies are absent, so whatever blood group, blood with whatever group is transfused into the person, there are no antibodies to react with the antigens. So that means it can receive blood from any blood group. So AB is universal donor based on the fact that there is no antibodies present. So absence of anti-A and anti-B antibodies in the plasma are the reason for blood group AB being universal recipient. Similarly, blood group O becomes the universal donor. It becomes universal donor on the fact that it has no antigens. So if blood group O is transfused to any donor, there is no antigen, foreign antigen entering inside. And if there is no foreign antigen entering inside, even if the antibodies are present in the recipient, it would not react because there is no antigen to react with. So O becomes universal donor. Reason for O being universal donor is absence of both the antigens. So the answer is option B. Now here again, if we look at the options closely, we know that antigens are present on the surface of the cells and antibodies are present in the plasma. Antibodies, they are present in the plasma as they are released by B lymphocytes, right? Antigens are cell surface proteins present on the plasma membrane of the cells. So first option, if we see presence of antibodies, anti-A and anti-B on RBCs. Antibodies cannot be present on RBCs because they are present in the plasma. So of course, first option is wrong. It is conceptually wrong, right? If we say fourth option, absence of antigens A and B in the plasma. Now antigens are not present in the plasma. Antibodies are present in the plasma. That means fourth option is also wrong. So even if we were not sure about the answer, we could have easily eliminated two options, and those students who wanted to guess, now your chances of getting the right answer increases to 50%. Now you have two options, and even if you make a smart guess here, right, absence of antigens A and B on the surface of RBC, right? If there are no antigens A and B, how can we call it the blood group AB? Because the basis of discrimination of blood group is antigens. So if we are calling it AB blood group, that means both the antigens must be present. So that means the third option is obviously wrong. And we are left with only one option which must be the correct answer. Right? So question was NCERT based but required understanding of the concept. Now moving on to the next question. The partial pressures of oxygen and carbon dioxide at the alveoli, the site of diffusion, the alveoli where oxygen and carbon dioxide are exchanged between blood and the alveoli. They have given us different values of oxygen and carbon dioxide. Now, in the alveoli, when there is a exchange of gases between blood and alveoli, right? We know that oxygen from the alveoli enters into the blood, and from blood, carbon dioxide is diffused into the alveoli. And the mechanism of exchange of gases is completely based on difference in the partial pressures. Difference in the partial pressures of the gases. So gases tend to move from higher partial pressure to lower partial pressure. Now we already know that at the alveoli, when the exchange takes place between blood and alveoli, oxygen will diffuse into the blood and CO2 will diffuse out, right? Since it is asking what is the partial pressure in the alveoli, right? So if oxygen is diffusing into the blood, that means oxygen partial pressure should be higher side, and CO2 is diffusing from blood to alveoli. That means in alveoli, pressure of oxygen should be higher and pressure of CO2 should be lower. Actually, this is a memory based question directly from NCERT. Now, if we see here, in all of these, the fourth option is wrong because the partial pressure of oxygen is extremely less. Partial pressure of oxygen in alveolar air is 104 mm of Hg. Right? Option B is also wrong. Option B is also wrong because PCO2 0.3. Right? It is not that much less. Right? So option C is the correct answer here, and this is a completely memory based questions on the from respiratory system, right? Breathing and exchange of gases, right? NCERT based easy question, memory based question. Now let's take a look at the next question here. Receptors for sperm binding in mammals are present on. Yes, here the question is again in connection with NCERT but not directly, indirectly from NCERT. We have the options given as perivitelline space, zona pellucida, corona radiata and vitelline membrane. Right? This is again a remembering question. The students who would have studied thoroughly must have remembered it, right? There is no application or understanding required here. But still, perivitelline space, this is the space present between the vitelline membrane and the primary egg membrane, which is filled with perivitelline fluid, right? Now, so this option A is of course not the answer, right? The receptors cannot be present in the space. They must be present on some membrane, right? So first option A is not the answer. In case of humans, zona pellucida is the primary egg membrane which is gelatinous, right, and secreted by the egg itself. And since it is made up of glycoproteins which acts as receptors for binding of sperm. Corona radiata, it is made up of granulosa cells secreted by the ovary and is the secondary cell membrane. So receptors for binding, and we already know that corona radiata being secondary membrane is dissolved by the sperm. Ovum, it is surrounded by zona pellucida, which is the primary egg membrane, and then it is surrounded by corona radiata cells, right? So when sperm has to reach and bind with the ovum, that means corona radiata cells or this layer must be dissolved, otherwise sperm cannot reach up to the ovum. So corona radiata cells, they do not contain the receptors for sperm binding, right? They are dissolved, and the zona pellucida, the primary egg membrane, contains receptors for binding of sperm with the ovum, right? So this question was memory based but slightly on a moderate difficulty level, and not many of the students must have been able to answer this because it is an extrapolation of NCERT. Right, moving on to the next question. The organelles which are included in the endomembrane system are, right, 4 options: Golgi complex, mitochondria, ribosomes and lysosomes. Next option, Golgi complex, endoplasmic reticulum, mitochondria and lysosomes. The third one is endoplasmic reticulum, mitochondria, ribosomes and lysosomes. And the fourth option, endoplasmic reticulum, Golgi, lysosomes and vacuoles, right? This is again NCERT based question only, but we have to understand what is endomembrane system, right? So slightly on the difficult side, on the moderate side, but and using understanding levels of the knowledge of the concept. Endomembrane system is a system of cell organelles whose functions are related with each other. Those cell organelles which have related functions with each other, they constitute the endomembrane system. Now Golgi and endoplasmic reticulum, of course these two are interconnected, they are related with each other because endoplasmic reticulum, right, serves as the site of protein synthesis, the rough endoplasmic reticulum, and as a cytoskeleton, and after folding of the protein, once a new protein is formed, after folding of the protein, it is transferred to Golgi complex for further modifications, right? So Golgi and endoplasmic reticulum, they are interconnected, and this means first option is wrong, right? Only Golgi is present. Mitochondria and ribosomes, they have completely different function. Mitochondria for the synthesis of energy, always involved in respiration. Ribosomes, they do not have membranes and they are the primary site of translation. Translation takes place in ribosomes. Ribosomes are attached on endoplasmic reticulum. So wherever mitochondria and ribosomes are present, since mitochondria and ribosomes, they do not have related functions with endoplasmic reticulum and Golgi, that means the option 2 must also be wrong, option 3 also must be wrong, and option 1 also is wrong. So we are left with only one option which is correct: endoplasmic reticulum, Golgi, lysosome and vacuoles. All the 4 cell organelles have interrelated functions. Lysosomes, they contain the hydrolytic enzymes. They contain the hydrolytic enzymes. These enzymes are synthesized on the surface of rough endoplasmic reticulum, modified in the Golgi complex and then transferred to lysosomes. Vacuoles are connected with lysosomes because these vacuoles, food vacuoles, which are formed during phagocytosis, they merge with the lysosome, forming a phagosome, wherein lysosomal hydrolytic enzymes digest the food particles. So these 4 cell organelles, they have interconnected function and they form the endomembrane system. So again, it is an NCERT based question, but slight understanding of the topic is required. Now, let's take a look at the next question. Question looks big. It says, for effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Yes, it's true, right? If we want effective treatment of the disease, we must be able to diagnose the disease very early, as soon as possible, and we should be able to understand the pathophysiology, right? Pathophysiology means how the pathogen is affecting the system, right? Which of these molecular diagnostic techniques is very useful for early detection? Right? Which molecular diagnostic technique is useful for early detection? The question is again out of NCERT, but we have 4 options: ELISA, hybridization, Western blotting and Southern blotting technique. Now, if we are talking about early detection, ELISA, enzyme linked immunosorbent assay, is the most convenient and the fastest technique used to diagnose a particular disease. There are different variations of ELISA, but in the most common principle, ELISA detects the presence of antigens or the presence of antibodies, the presence of antigen or the presence of antibodies in the patient's blood. So ELISA is the obvious answer here, as it is the most rapid molecular diagnostic technique, as it diagnoses or detects the presence of antigens or antibodies present in the patient's blood. Hybridization technique. Hybridization technique is also a molecular technique, but is used mostly in DNA fingerprinting. It is not used for diagnosis of diagnostic as a diagnostic technique for disease. Western blotting and Southern blotting. Western blotting is for detection of proteins, while Southern blotting is for detection of DNA, right? So these are molecular techniques used in molecular genetics for finding out the DNA match between 2 persons. So most of hybridization, Western blotting and Southern blotting, these are mostly used in DNA fingerprinting and solving paternity cases or any criminal cases, identifying the person. For diagnostic techniques, ELISA is the only diagnostic technique here. So the obvious answer is first. But since the question is out of NCERT, we can call it a little bit towards the difficult side. But again, ELISA, we study this in human health and disease as a diagnostic technique, and out of the 4 options given, this is the only diagnostic technique given here. So this must have been the obvious answer also. Now let's move on to the next question after this. Again, the question says we have to match the 2 lists given, right? We have been given 4 options. In this kind of case, again, let us first try with the conventional approach. Aspergillus niger, Acetobacter aceti, Clostridium botulinum and Lactobacillus. We have been given 4 microorganisms, and in the list 2, we have acetic acid, lactic acid, citric acid and butyric acid, the products obtained from these microorganisms, right? Aspergillus niger, Acetobacter aceti. Now here again, if we start matching the list, right? Aceti means acetic acid, right? The word aceti resembles with acetic acid. So Acetobacter aceti, from this bacteria we obtain acetic acid. So option B is matched with first option in the list 2. And if we see the options clearly, there is only one option where Acetobacter aceti is matched with acetic acid. That makes it the obvious answer. Again, if we have not remembered, remembered about Acetobacter aceti, Clostridium butylicum, butyric acid, right? Clostridium butylicum, in the name itself, from butylicum we know that it is used for obtaining butyric acid, right? And if we see matching of C with fourth, this is again only in one of the options, right? Same way, lactobacillus, lacto for lactic acid, right? So the question was from use of microbes for human welfare chapter and was NCERT based question. But again, it was a very easy question since the match options were so easy and they were differentiated that if you have matched only one of these correct, you will get the rest all correct answers, making it easiest question so far, right? Let's have a look at next question, which is our way. Which enzyme is responsible for the conversion of inactive fibrinogens to fibrin? The question, this belongs to circulatory system, right? We have been given 4 options: epinephrine, thrombokinase, thrombin and renin. Out of these 4 enzymes, which enzyme is responsible for the conversion of inactive fibrinogens? Now, it says which enzyme, right? In the 4 options, renin and epinephrine, these are not enzymes, they are hormones. So we are left with only 2 enzymes, thrombokinase and thrombin. Thrombokinase enzyme, it catalyzes the conversion of inactive prothrombin into thrombin. Thrombin converts inactive fibrinogen to fibrins. So the correct answer here is thrombin protein, which acts as an enzyme, converts fibrinogen, soluble fibrinogen into insoluble fibrins, which form a network at the site of wound, and the blood corpuscles get trapped in this mesh-like network and blood clots. So the question is again NCERT based, from the mechanism of blood clotting, the 3 steps of blood clotting, and was easy question, memory based question. Right? Moving on, a specific recognition sequence identified by endonucleases to make cuts at specific positions within the DNA is. Now, in simpler language, the question is asking about what is the recognition site of restriction endonuclease enzymes. right? But the same thing is written in a different way. So the terminology of the question, the way the sentence has been formulated, right, makes it difficult to interpret for students that a specific recognition sequence identified by endonucleases to make cut at specific position, right? Endonucleases which identify a specific sequence and cuts at a specific part, they are called as restriction endonuclease. So indirectly the question is simply asking us what is the recognition site of restriction endonuclease enzymes. We have been given as 4 options: palindromic nucleotide sequences, poly A tail sequences, degenerate primer sequences, and Okazaki sequences. Now restriction endonucleases, their property is that they always recognize a very specific 4 to 8 base pair sequence in the DNA. And this 4 to 8 base pair sequence, which is recognized by restriction endonuclease enzymes, is a palindrome. What do we mean by palindromic sequence? Palindromic sequence is such a sequence which, when read from both the sides, reads the same. For example, if we take the restriction site of the restriction endonuclease ECOR1, the restriction site recognized is a 6 base pair sequence GAATTC, wherein on the complementary end, on the complementary strand, the site is read same from both the ends, GAATTC from one end and from the other end again GAATTC. Such site which is read same from both the ends of the DNA, it is called as palindromic sequence. And restriction endonuclease enzymes, they recognize palindromic sequence, and in the palindromic sequence they cut at specific site. Like for ECOR1, it always cuts the DNA between guanine and adenine. So palindromic nucleotide sequences, they form the recognition sites. Recognition site, or we can call them restriction sites of restriction endonuclease enzyme. This is again NCERT based question from biotechnology, principles and processes, and is written as it is in the NCERT textbook. Right? So a memory based question, easier question. Now, with regard to insulin, choose the correct options. The next question says, with respect to the hormone insulin, we have to choose the correct option. What they have given is, they have given us 4 statements, and out of the 4 statements, we have to find out which of these 4 are correct statements. Now, it says that in insulin, the first statement says that C-peptide is not present in mature insulin. Now, insulin, in the insulin gene, when it is transcribed and translated, right, it forms 3 polypeptides, A, B and C polypeptide. A and B polypeptides, or A and B chains, after the formation of the polypeptide or the inactive insulin, it undergoes post-translational modifications. After the formation of a polypeptide chain, it undergoes modifications after translation, wherein the C peptide is removed and A and B chains, they are joined together by disulfide linkages. So, functional insulin or mature insulin contains only two polypeptide chains, the A and B polypeptide chains, which are connected with, linked with disulfide linkages. So mature insulin or the functional insulin, it does not contain C polypeptide. C peptide, yes, that is correct. The insulin produced by rDNA technology has C peptide. This is again a correct statement, because when we go for recombinant DNA technology, we isolate the gene of insulin. We isolate the gene of insulin, or we synthesize a complementary DNA using insulin mRNA. From insulin mRNA, we synthesize a complementary DNA. And this complementary DNA, which now acts as the insulin gene, is transferred into the bacteria. Why do we do this? Why don't we directly isolate the insulin gene? And why do we go for reverse transcription? Now we have to understand, we are transferring a eukaryotic gene into a prokaryote. Eukaryotic genes, they are split genes. Split genes, that means they contain both euchromatin and heterochromatin parts in alternate fashion, right? So a eukaryotic gene, it contains alternate euchromatin and heterochromatin parts, which are called as exons. The euchromatin part are called as exons, which contain the information for functional protein, while the introns are the heterochromatin part, right? So now the mRNA which is synthesized contains exons and introns both. But this mRNA undergoes post-transcriptional modifications where the introns are removed and only exons are joined together. Only the functional sequences are joined together. So the functional mRNA, it contains only the sequence of exons. Prokaryotic genes are not split genes. They do not contain heterochromatin. So if we transfer the insulin gene directly from humans to prokaryote, E. coli, in that way we will be transferring the entire gene along with the heterochromatin, right? And prokaryotes, they do not have post-transcriptional modification machinery. They cannot remove the introns and join the exons. So even if we transfer the gene of insulin directly, we will not get a functional protein. So instead, we can isolate the functional mRNA, and from this functional mRNA, we can synthesize a complementary DNA. Now this complementary DNA contains sequence only for exons. But again, this contains the sequence for all the three polypeptides, A, B as well as C. So even with rDNA technology, the insulin that is produced will contain all the three peptides. So C peptide is present in the insulin which is produced by recombinant DNA technology. And this C peptide is essential because it helps in formation of disulfide linkages between the A and B peptides, right? The proinsulin has C peptide? No. The proinsulin does not have C peptide. A peptide and B peptide are interconnected by disulfide bridges? Yes, that is also correct. So 3 of the 4 statements are correct, and we have, now if we see the 4 options, A, C and D only, A and D only, B and D only, B and C only. Now, option A, correct. Option B is also correct. Option C, the proinsulin contains C peptide. Yes, the proinsulin, that means immature insulin which is formed. Yes, it contains polypeptide. So all the 4 options were correct, but again the best possible answer here is A, C and D only. Since all the 4 options here in this question were correct, all the 4 statements were correct regarding to insulin, but only one option. There is no option saying that all A, B, C and D are correct. So the best option here is A, C and D. And the question was towards the difficult side of it from applications of biotechnology and was a little bit understanding based question. Moving on to the next question. Which of the following characteristics is incorrect with respect to cockroach? The question is from structural organization in animals. Here in females, 7th and 9th sterna, 7th to 9th sterna form genital pouch. Yes, this is correct. 10th abdominal segment contains anal cerci in both the sexes, both males and females. Yes, that is also correct. The 10th abdominal segment, it contains anal cerci which are 15 segmented, long, narrow, antennae-like structures. Right? The third option, ring of gastric caecae is present at the junction of midgut and hindgut. And the fourth one is hypopharynx lies within the cavity enclosed by the mouth parts. Which one? They have asked us to identify the incorrect statement. Right? We have to identify the wrong statement. Here, the first statement is correct. The second statement is also correct. The third statement, ring of gastric caecae is present at the junction of midgut and hindgut. It is wrong because at the junction of midgut and hindgut in cockroach, Malpighian tubules are present. Hypopharynx or the linguae, which acts as a tongue-like a structure, it is present in the cavity, buccal cavity, which is enclosed by the mouth parts. So A, B and D, these are correct statements, but the question asked which one is wrong. So the option C is wrong statement. This is again from structural organization in animals, type study cockroach, memory based question directly from NCERT. Now let's move on to the next question, which wrongly represents the nature of smooth muscles. Right? The question is talking about the nature of smooth muscles which are involuntary muscles. Communication among cells is performed by intercalated disc. These muscles are present in wall of blood vessels. They have no striations and they are involuntary muscles. Question says wrongly represents, right? Smooth muscles are involuntary muscles. This is correct. Smooth muscles they are called smooth muscles because they do not have any striations. This is also correct. These muscles are present in the wall of blood vessels. Yes, they are involuntary muscles, right? And in the wall of blood vessels, we cannot control the contractions in wall of blood vessels. So this is also correct. Communication is performed by intercalated disc. This statement is wrong because intercalated discs, they are a characteristic feature of cardiac muscles only. They are present only in cardiac muscles. So option A is the correct answer here. Again, a slight understanding is required. Understanding of the structure was required here. But the question was easy. Right. Now again we have a match the list. Right. On list 1, we have been given 4 different methods of birth control. And on list 2 is the mechanism how they prevent fertilization. Right. Again in this question, this was again an easy question. 4 options which were given. Now we look at it closely. Vasectomy and tubectomy. Right. It means removal. right? Whenever we use the suffix tomy, it means removal. Vasectomy means removal of vas deferens. Tubectomy means removal of fallopian tubes, right? So the C1 is matched with option, the second one in list 2, and tubectomy D is matched with 4 in list 2. If we see here, C is matched with removal of vas deferens, second option in list 2 only in one of the options here, and D with fourth only in one of the options, right? So the answer is D here. Now vaults, now to understand vaults, they are also called as cervical caps. They are cap-like structures which are placed on the opening of cervix, and they prevent the entry of sperm. So vaults, they prevent the entry of sperm through cervix and hence prevent fertilization. IUDs, intrauterine devices like copper T, they make the cervical plug or the cervical mucus rich in copper ions, and as a result of that, sperms cannot survive, and the dead sperms are phagocytosed by the uterine wall. So IUDs, they prevent fertilization by causing phagocytosis of sperms within the uterus. So the question again was easy, smartly done. We can see the options which are given. If we go from options, vasectomy and tubectomy, they are obviously matched by the names, and there is only one option which this correct matching. Now, next question says that in a cross between a male and female, both of them are heterozygous for sickle cell anemia. What is the percentage of progeny who will be deceased, right? Now sickle cell anemia, right? The question is saying that both male and female are heterozygous. So male is heterozygous for sickle cell anemia, while female is also heterozygous for sickle cell anemia. Now a cross between these two, if we make a Punnett square, on one end, let's take the female gametes, and on one side, let's take the male gametes, right? So one of this offspring is normal. Here, the offspring is affected, right? But carrier, right? Offspring is carrier or heterozygous. The offspring, third offspring is also a carrier and heterozygous, while the fourth offspring is deceased, right? Now, sickle cell anemia is a recessive genetic disorder, and if it is a recessive autosomal genetic disorder, now we can simply go with the Mendelian cross. From F1 to F2, when we go for selfing, we cross the heterozygous plants and we obtain the ratio as 1:2:1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Or if we take the phenotypic ratio, comes as 3:1. 3 dominant and 1 recessive. Now, if both the parents are heterozygous, right, and we are crossed, both the parents are being crossed, so the chances of homozygous recessive offspring will be 1 out of 4. So we can directly answer the question that it is 25%, slightly application based question, right, required a little bit of understanding and towards the difficult side. Right. Which of the following RNAs is not required for synthesis of protein? We have been given 4 different RNAs: rRNA, siRNA, mRNA and tRNA. saying which of these RNAs is not involved in protein synthesis. In protein synthesis, we require 3 different RNAs. From DNA, mRNA is formed which carries the information of amino acids, right? Then rRNA, ribosomal RNA, which is a part of ribosomes, and tRNA, the transfer RNA, which brings the respective amino acids. So mRNA, tRNA and rRNA, these 3 RNAs are involved or required in protein synthesis. Only one option is left, siRNA, and that is the obvious answer. These are small interfering RNAs and used for gene silencing. So again, the question was on moderate difficulty level, but if you look at the options closely, solving the question would have been very easy, and this is again NCERT based question, right? Again we have a match the column. On one end we are being given characteristic features, in the second list phylums are being given. Now again, if we look at the options and then start matching, right? Cnidoblast cells, these are characteristic features of coelentrates. So the D in list 1 is matched with first of list 2, and if we see the options, there is only one option where cnidoblast cells are matched with coelentrates, making it the obvious answer, right? So complates, these are locomotory organs, helps in locomotion in ctenophores. Canal system is a characteristic feature in poriferans, while metamerism for the first time in animal kingdom is seen in annelids, the true segmentation. This question, select the favorable conditions required for the formation of oxyhemoglobin at the alveoli. Now, we have been given 4 options: high pressure of oxygen, high pressure of CO2, less hydrogen ion concentration and higher temperature, right? Similarly, option B says low PO2, low CO2, less H plus and higher temperature. For the process of formation of oxyhemoglobin, oxygen must bind with the hemoglobin, that means PO2, right? In the cells, if oxyhemoglobin is being formed, right, PO2 must be higher, PCO2 must be less, acidity, H plus concentration must be less with lower temperature, right? This here question is opposite of the oxyhemoglobin dissociation curves, required conceptual understanding as well as application of the concepts was on the difficult side, and an extrapolation of information given in NCERT, because the dissociation of oxyhemoglobin curve, when the oxyhemoglobin dissociates, it dissociates when the pressure of oxygen is less or CO2 concentration is higher. At that time, oxyhemoglobin dissociates. So opposite must be true for formation of oxyhemoglobin. So question was, you can say, an extrapolation of NCERT, can say it out of NCERT, and required application towards the difficult side of it. Now, moving on, which statement about histone proteins is wrong, right? Histones, as we know, are basic proteins which helps in folding of the DNA. Histones, they are rich in lysine and arginine amino acids, positively charged so that they can associate with negatively charged DNA, right? And they are organized in the form of a unit of 8 molecules to form nucleosome on which DNA is wrapped around. But the pH of histones being slightly acidic, no, histones are basic proteins. So option D is the wrong answer. This again is question is directly from NCERT. Question is directly asked from NCERT itself. Identify the type of cell junctions that help stop the leakage of substance across a tissue and facilitation of communication with neighboring cells via rapid transfer of ions and molecules. The language of the question sounds difficult, but the question is actually easy. 4 options are being given and they want us to identify what type of cell junctions help in leakage of to stop the leakage of substance across the tissue and to communication, right, to facilitate communication between the neighboring cells with rapid exchange, easy exchange of ions and material between the two cells. So, easy exchange of ion and material between two cells, it is facilitated by gap junctions which form tubules, right. So, for the second one, the correct answer should be gap junctions, right. So, this means that option 3 and option 1, these are wrong. Now, out of the 2 adhering and tight junctions which help to stop the leakage, it is the tight junctions. At tight junctions, the plasma membrane of 2 adjoining cells, it fuses completely and prevents leakage of material from inside the lumen of the cell. So, the correct answer is option D. But the question is still NCERT based question from structural organization of animals. Now, the next question says, which of the given is not a step in multiple ovulation embryo transfer? The question is from animal husbandry or animals in human welfare. Multiple ovulation embryo transfer, in this technique, what we do is we inject the female with follicle stimulating hormone so that instead of releasing 1 ovum per cycle, per ovulation, the female is induced to release more than 1 ovum, usually 6 to 8 ovums per cycle. These ovums are then artificially inseminated, the female is artificially inseminated for fertilization. And when the embryo reaches at 8 to 32 cell stage, at that time the embryos are removed artificially and implanted into surrogate mothers, and the actual superior breed female is now ready for next cycle. So here the options given, cow is fertilized by artificial insemination, yes, that is a process. Fertilized eggs are transferred to surrogate mothers at 8 to 32 cell stage, that is also done. Cow is administered hormone having LH like activities, no. The cattle is administered with hormones which have FSH like activity to induce ovulation, for formation of more than 1 Graafian follicle. So, the option 3 is not a step in MOET technique. And the option D says cow yields 6 to 8 eggs at a time. Yes, because of introduction of FSH like chemicals, more than 1 Graafian follicles mature and more than 1 ovums are released per cycle. Again, the question is directly from NCERT. Now, the next question, if we see, matching the list. Again, we have 4 diseases with their causative agents. Now, the most common disease, filariasis, right? It is caused by Wuchereria bancrofti, the filarial worm. And if we see the matching, filariasis is matched with third option in list 2, in only one of the options. Only one options matches A with third. Right? So of course, D is the correct answer. Even if we don't know about filariasis, amoebiasis, amoeba caused by ant amoeba, right? Matching B with fourth. This also is happening only in one of the options, right? Making the fourth option the correct answer. So smartly attempting this question, the student could have saved time here and invested it in other sections of the question paper. The next question from zoology is, which of the following hormone relaxin, which of the following secretes relaxin hormone in the later stages of pregnancy? Again, this is a memory based question from human reproduction. Relaxin hormone is secreted by corpus luteum at the time of parturition, and it relaxes the pubic symphysis. It relaxes the pubic symphysis so that the embryo can move easily down the birth canal. So here the correct answer is corpus luteum. Again, a memory based question directly from NCERT and easy question, no need for understanding of the concept or application part of the concept, towards the easier side, right? We have one more, match the list. In list 1, this question is from evolution. In list 1, we are given some terms like adaptive radiation, convergent evolution, divergent evolution, and evolution by anthropogenic actions. In list 2, we are given examples of these processes. Adaptive radiation, right? Again, question is directly from NCERT. Adaptive radiation, right? This example is clearly shown by Darwin's finches. The Darwin's finches, which migrated to Galapagos Island, they have a common ancestor in South America. But on the Galapagos Island, Darwin observed more than 22 different varieties of finches, which were different in the structure of beak and their feeding habits. So from one common ancestors, different species radiated, right, because of the adaptations to the environmental changes. And such type of speciation, formation of new species, is called as adaptive radiation. So adaptive radiation, the example is Darwin's finches, and A is matched with fourth in only one of the options, making it the correct answer. Convergent evolution. Convergent evolution means different lineages having similar structure. For example, we have convergent evolution, different organisms showing similarity in structures which perform same functions. For example, wings of birds and wings of bats. Divergent evolution, wherein the example is shown clearly by homologous organs, right? Wherein the structure of the organ is same, but the function is different due to different adaptations. Like, for example, bones of forelimb in man and whale. They all have the same basic structure: humerus, radius ulna, carpals, metacarpals, and phalanges. But in humans, the function is grasping and holding. But in whales, the function is swimming. Right? So option C becomes the correct answer. Slightly understanding part and moderate difficulty level in the question, we can say. Again, we have a match the following question, wherein 4 bones are matched with the different types of properties? Scapula, the shoulder bone, is a flat triangular bone present in the pectoral girdle. A is matched with 4 in 2 of the options, making C and D wrong. Right? So only 2 options are correct. A and B. Out of these 2, now we have to find out. Cranium, the skull, the bones of cranium, they have fibrous joints, the fibrous sutures, which are immovable joints. So only one option which matches, list A, cranium with fibrous joints and scapula as the triangular flat bones. Right? So again the question is from movement and locomotion. Remembering question, not much of understanding or application, but moderate level of difficulty. Now we have an assertion reason question towards difficulty level. A person goes to high altitude and experience altitude sickness with symptoms like breathing difficulty and heart palpitations. And the reason given is that due to low atmospheric pressure, the body does not get sufficient oxygen. Right? Say the 4 options given, assertion is true, reason is false, assertion is false, reason is true, assertion and reason are true and reason is correct explanation of assertion. Now assertion says when we go at higher altitude levels, the person experiences altitude sickness because the air becomes thin. As we move higher, the air, the percentage of oxygen at higher altitudes decreases. So there is less oxygen available to the tissues, and that results in altitude sickness and heart palpitations. So assertion and reason both are true, and reason is correct explanation of assertion. So option C is the correct answer. This question is slightly difficult based on understanding as well as application of the concept, right? But again, the question is NCERT based question. Now the last question that we have here is that codon AUG codes for methionine as well as phenylalanine, right? We know that codon AUG is the initiation codon and always codes only for methionine. That means statement 1 is a wrong statement, right? Now, statement 1 is false, so that means option A is wrong and option C is also wrong, because both the options they say statement 1 is true. Now come to the second statement, AAA and AAG, both the codons, they code for the amino acid lysine. Yes, amino acid lysine is coded by 2 codons, AAA and AAG, which is again memory based. So question was towards moderate difficulty level, and the answer here is that statement 1 is incorrect, but statement 2 is correct. So overall, the question paper here was towards the easy difficulty level, but the students who have thoroughly studied NCERT and attempted the questions with a little bit of smartness and common sense would have been able to score more questions, because as we have seen, most of the match the following questions and few questions, they have very obvious options wherein you can easily eliminate 3 options and the remaining 4th one was the answer. So with this, we will finish with the discussion of the zoology section, and I hope that you all have done good in the zoology paper. Now, for the continuation of this discussion session, I would invite Kishore sir to discuss with chemistry part.
So hello everyone, and now we are into the chemistry part. I hope you guys have had a clear discussion about zoology and botany. So there was an elaborate discussion about all the questions of zoology and botany, and now we are into the chemistry part. And as I have told you that the chemistry part was of an easier and moderate level, yes, it differs from students to student. The students who have prepared well. So when I say the preparation is well, it means you are following the guidelines of the teacher. That is, you are doing the homeworks, following the NCERT book, and doing the worksheets. If you have done that, so then definitely the paper was easier for you. Okay, so before getting into this, one more important point which I want to tell you is that this time the paper was, the number of questions which were there were more from the 12th syllabus and lesser from the 11th syllabus. I would say 60 and 40%. Okay, and it's a good sign. I would say it was very helpful for the students, because you are writing an exam who are studying in the 12th, so you are in touch with the 12th syllabus, and hence it's easier to retain the 12th knowledge by revising the 11th part. So getting more questions from 12th is a favorable point which is helpful for the students to score even more. Okay, and as I have told you, the questions were of moderate and easier level, or we can say a standard according to what the NEET usually gives. So it was an ideal paper which every year it is repeated. So the paper again evenly distributed in terms of organic, inorganic, and the physical part. So I am going to discuss few questions which I feel that it has to be discussed, because there are some questions where you have to take some extra care. Apart from the questions which are directly from the lines of NCERT, and some questions which are directly from formula based, we are going to discuss few questions where you have to be some extra care about the options and the questions and the comprehensiveness of the paper. So we are going to discuss few questions which are to be discussed in the case of NEET. Okay, so coming to the first question over here, so it is from the P block elements, that is from the noble gas, and they're asking us, see, there is the first thing that you have to observe is the statement is about incorrect statement. So they're asking us about the incorrect statement, so you have to be very careful about the incorrect statement. So they're asking us about the incorrect statement. So that is the first thing that you have to note down, because you in a hurry we always forget about that incorrect and correct one. So we sometimes do mistake with that, and we later realize that it was incorrect, and again we read it. So there is a wastage of time. So you have to be very careful reading this question. Incorrect statement, and these are the questions, these are the options that were given. And this question, why I am discussing this question seems to be a easier one because it's a theoretical one. No, this is a question which is trying to check your knowledge, the basic knowledge. Let's see option by option. The first option, if you see, noble gases have weak dispersion forces. Obviously, because being a covalent molecule and a non-polar molecule, they definitely have what they do not have interactions like bonding like dipole, and etc. They have weak van der Waals force of interaction, that is the weak intermolecular force of attraction between the molecules, that is the London forces or dispersion forces. The first statement is correct. Okay, so they are trying to check whether you know what is this van der Waals force of attraction. So these are the force of attraction that you will see between the molecules which are non-polar in nature. Okay, so that was the first thing. They were trying to check the van der Waals knowledge. Then the second point, noble gas have large positive values of electron gain enthalpy. So now they are trying to check whether you know about the electron gain enthalpies. Yes, what is that? It's the energy involved whenever you add an electron to a gaseous atom. So what about the noble gas? They are already stable. So why do they want something else to enter into them? So whenever you add an electron to this atom, what happens? It is by force. It means we have to, it is what? Endothermic. So we need to spend energy. So they are positive values. The electron gain enthalpy values are positive. Okay, right. The noble gas are and largely positive. We will say largely positive because it's because they are highly stable. So it's impossible, or we can say it requires a lot of energy to put those electron into the outermost shell. The third option is the noble gas are sparingly soluble in water. The noble gases are sparingly soluble in water. So here they are trying to check whether you know the concept of like dissolving into a like. So we know that the solubility line that we say, that I usually say in my class, like dissolving like. Polar solvent, polar solute into a polar solvent, and a non-polar solute into a non-polar solvent. So here water being a polar solvent, and what about the noble gases? Because they are non-polar, so is that combination? So the noble gases are sparingly soluble in water. So non-polar are usually sparingly soluble. What do you mean by sparingly soluble? The solubilities are very less. So yes, the statement is again true. So they were trying to check the concept of like dissolving into like. What about the last one? The last option is noble gases have very high melting points and boiling points. So what do we know about the boiling points and melting points? It's all about the interaction. More is the interaction between the molecules, more stronger are the IMFs, intermolecular forces. We say the boiling points are higher. And we know that these noble gases being covalent molecules, non-polar molecules, they have weak dispersion forces. So weak forces means definitely they will have low or high. Yes, that is low melting points and boiling points. So that is the wrong statement. So they're asking us the wrong statement. The incorrect statement is this one. So that's how this question is important. So it seems easier, but you have to be very cautious with each and every line, each and every sentence, each and every word of each option. So that is how we have done this first question, which is from P block elements. Okay. So next, let's move on to the next one. So the next question, if you see, right. So the next question is about metamerism. So they're asking us which compound shows metamerism, and they gave us a few options. Okay. So first we should know what is this metamerism. Okay. So what is this metamerism? So metamerism, if you see, it comes under the topic of isomerism, the basic concepts of organic chemistry, the GOC part, which is there in your 11th syllabus. So there you learn about the structural isomerism, where metamerism is a part of structural isomerism. And what's special about this metamerism? So metamerism is shown by few organic molecules where there is a change in the alkyl groups attached to a group of atoms. Okay, for example, this metamerism is usually seen in molecules like ethers. So you will see in ethers like this one. Okay, and you also come across you will also come across acetones. Okay, so okay, this is so in acetones and in ethers you will usually come across this kind of metamerism. So what is metamerism? The molecules which are structurally related, but what is that? There is a change in the alkyl groups on either side. Okay, so those are called as metamers. Now, how do we identify metamers? So from this definition itself, you can strike away one of the options. What is that? This is an alkane. So this option goes away from my list of options. I am left with 3 more options, and you can see in all the 3 options I have O, I have O, and I have O. So this might be an acetone or a ether. This might be an acetone or a ether. So how do we confirm whether it is an acetone or a ether? So there is a difference between acetone and ether. How can you find it? You can do it with the help of the unsaturation value, because the unsaturation value of acetone is 1 and unsaturation value of the ethers are 0. Okay, so how do you find out the unsaturation value? So unsaturation is equal to C plus 1, the number of carbons plus 1, minus or number of nitrogens, hydrogens minus X by 2. So this is a basic formula which we use to find out the unsaturation value of any organic molecule. Okay, so here I do not have nitrogens or hydrogens or halogens along with carbon and hydrogen. So this formula reduces to U is equal to C plus 1 minus hydrogens by 2. So this is the formula that you can use over here to find out the unsaturation values. So let's find out the unsaturation values of each one. So what will be the unsaturation value of this case now? So there are 3 carbons, so 3 plus 1, 4, 4 minus the number of hydrogens are 6, so 4 minus 6 by 2, it is 1. 4 minus 6 by 2, it is 4 minus 3, that is 1. So what about unsaturation value over here? So U is equal to, there are 4 carbons, so 4 plus 1, 5, 5 minus 10 by 2, that is 0. Okay, and what about this one? The unsaturation value is U is equal to C plus 1, that is 4, 4 minus 8 by 2 is 4 again, so 0. So I could clearly see that it could be acetone or, so this one is the acetone part, this is the acetone part, okay, this is the acetone part, and these two are my ethers. Okay, but one small common sense thing that you have to apply over here. See, so for a molecule to show metamerism, the number of carbons should be at least more than 3, because you can see if you have 3 carbons, suppose I am going with 3. Okay, so what is the other way you can write over here? So the other way you can write over here is, let's say this one. So both of them are same. So with 3 carbons, you cannot show metamerism. So for the basic condition for metamerism is nothing but you have to have at least carbons more than 3. So that is possible where, so it is possible only in which case? This case. So this is my answer. So because you can see all the 3 are capable, one is acetone, the other 2 are ethers, but they are not capable of showing metamerism. The only one which can show metamerism is this one, C4H10O. So you will be getting a diethyl ether. Okay, so this will be one molecule. So this is the one diethyl ether, and you will also be getting methyl propyl ether. So this is one. So there will be 2 isomers which are formed for ethers. Okay, so these are the metamers, and the answer is B. So this could be answered actually in 1 minute, or it could be answered in 10 seconds also. How could you answer in 10 seconds? I told you the basic concept is that what is that? It should be at least 4 carbons. So just look for the 4 carbons here. Obviously this is out of my options, so this is the answer. So you can do it just by looking at it within 5 to 10 seconds. Okay, right. So here I just wanted to tell you how to find out the unsaturation number. This is very important to identify the different functional groups, whether it is an ether or a ketone, like that. Okay, right. Right. So now let's move on to the next one. So the next question is also, so if you look at the questions which we are discussing here are all of, are all the basics, the roots are from NCERT, but they are of understanding level. So I would put all the questions, whatever the questions which I am going to discuss, where you have to understand the question properly, you have to understand the concept and then answer. And there are some questions which are applicable also. Okay, the remembering questions are the ones which are easily available in the NCERT, and you can answer them with just a look. So we are discussing the questions which are usually understanding, and it takes a bit of consciousness. So let's learn about the next one. So the next question is the major product formed in the dehydrohalogenation. So in the dehydrohalogenation reaction of 2-bromopentane is penta-2-ene. So they gave us the reaction. So 2-bromopentane is converting into penta-2-ene, and the product formed is based on, so they are giving us some conditions. Okay, so they gave us some conditions, whether it follows which of conditions. Okay, so by looking at the options, always look at the options in such a way that you are removing some of the unrelated options. So if you see over here, do you see Hunt's rule, anything related to this thing? No, it is related to the electronic configuration, right? So it has nothing to do with this question. Next, if I look at Huckel's rule also, this is also an option which is not into my consideration because it is related to aromaticity. So I will have a confusion between what is that, Hoffman rule or Sagejeff rule. So now what is Hoffman and Sagejeff? Both the terms are there in your NCERT book. Okay, both the terms are there in your NCERT book. I would say this is a question straight away from your NCERT lines. Okay, so here what is happening here is, and it is a beta elimination too because it is dehydrohalogenation. So what is happening over here is you have I think you have pentane, 2-bromopentane, 1, 2, 3, 4, 5. So this is your 2-bromopentane, which is converting into what? This is converting into, when we add it to alcoholic KOH, which is the dehydrohalogenating agent with beta elimination. Okay, so there will be a beta elimination happening. And what is the product you will be obtaining? The product that is obtained is what? It is penta-2-ene. So this is your answer, 1, 2, 3, 4, 5. Penta-2-ene is the product obtained. So when we will be getting a penta-2-ene as a major product? So this is the major product obtained. So whenever there is a beta elimination, and if the elimination is according to the Sagejeff rule, if the elimination is according to the Sagejeff rule. Now what is the Sagejeff rule here? Or it's also named as Sagejeff. In some books it is like Sagejeff. Okay, it is Sagejeff. Okay, in some books it is also written as Zaitsev. So this major product is according to the Sagejeff rule or a Zaitsev rule. Now what is the Sagejeff or a Zaitsev rule? According to him, he says that whenever you do a dehydrohalogenation and the alkane which is obtained, the major product is decided based on its stability. So more stable alkane, or we say more substituted alkene are formed as a major product according to the Zaitsev rule. So the more substituted alkene is a major product according to the Sagejeff rule. And what is Hoffman's rule? Hoffman's rule is nothing but you will come across Hoffman's product whenever there is an elimination using a bulkier base, or where there is some problem with the steric hindrance, where there is a step called as deprotonation. So if it is unable to, if the base is not able to remove the proton which is more stable, not able to remove the proton which is from a stable, which will give you a stable alkene, and removing the proton which is unhindered. Okay, so it will remove the hydrogen from the unhindered carbon. So there you will be getting a Hoffman product. So this is a exceptional case where you get Hoffman products because of the size compatibilities. Okay, so here you will be getting actually two products. What are the two products? Tell me. One of the product will be the major product, and the minor product would be this one, because the hydrogens, the beta elimination could be using this hydrogen or using this hydrogen. So this hydrogen will give you a Sedgwick product, and this hydrogen. So this is called as the non-Sedgwick product. It's called as the non-Sedgwick product. Or we can also say it's a Hoffman product. So usually we get a Hoffman product in elimination due to the steric factors, where the less stable alkene is a major product. So the next question is all about so the next question is again from organic chemistry. So the previous question was from organic chemistry, and it was from the first chapter of the haloalkanes and haloarenes. Okay, right. So what is the IUPAC name of the organic compound formed in the following chemical reaction? Okay, so they gave us a chemical reaction, and this reaction is from the chapter called as aldehydes and ketones, which I always say that the most important chapter of your organic. So if you see, whenever I say most important chapter, there are possibilities of getting more than one question. So there are some questions which are repeatedly asked. So there are multiple questions from one from a particular chapter. So those chapters are even more important for our preparation. So let's see the options now. So there is a reaction between acetone. So it is a RMGX reaction where acetone is reacting with the Grignard reagent in the presence of dry ether. Obviously, we have to use a dry ether whenever there is a Grignard reagent. And the second step is a hydraulic, it's a hydrolysis, acidic hydrolysis. So what is the product that you will obtain? So acetone, right? So what you will expect the product to be? So if you go with the mechanism acetone, okay, so we have the acetone part, and you know that this will they will undergo what type of reactions? Nucleophilic addition reactions. They undergo nucleophilic addition reactions. So what happens? So there will be, you know that you will be having a partially negative end and a partially positive end. And the RMGX, where is the RMGX? C2H5 MGBR. Okay. So this is the partially negative end, and this is the partially positive end. So that's very simple. Just add the negative to the positive, positive to the negative. So what will happen? This part will go to this one, and this part will go to here. So the product that you will be getting is CH3. You have C, O, and MGBR. Okay, and you have C2H5 connected to this, and you have the remaining alkyl chain. So this on hydrolysis, we are also using the second step. We are going to remove, we are going to hydrolyze it. Okay, so acidic hydrolysis, what will happen? This part will be replaced by H plus ion. So you will be getting what? Tell me, alcohol. So you will be getting an alcohol which is a tertiary alcohol. Okay, you will be getting a tertiary alcohol. They are asking us the name of this. So what is the name of this? You can name it very clearly. So what is the name? So this is the chain which I can see, the parent chain, and you can see there is at the second carbon there is ol and also a methyl group. So and it is a butane. So butane, 2-ol, 2-methyl. So this is my answer. Okay, so they might also give you a different kind of question from this part. I'll give you a small table to remember what is a product that you can obtain. So the products that you can obtain is like, for example, if I use formaldehyde. If I use formaldehyde, whenever I'm reacting with RMGBR, okay, what is the product that you will obtain? And whenever you use any aldehydes. So this is what I give in my class, and it's quite easier to remember. So ketones plus RMGX, what do we get? With formaldehydes, usually you will be getting primary alcohols, okay? You get primary alcohols, and from aldehydes, you will be getting secondary alcohols, and from ketones, just the question what we have done, you get tertiary alcohols. Okay, so this could also be asked in some kind of a way. So this is a very, very important topic where they are asking us about the reaction. I always say that in my class, the reactions involving, so the reactions which are named reactions, the reactions which are having Grignard reagent as a reactant, okay, and the reactions with which are identification test. Those are very, very important, and those reactions are being asked. Okay, so here there is a reaction with Grignard reagent. There is also a reaction where you have to identify primary, secondary, and tertiary. There is also reactions called as named reactions. So all of them were asked. Okay, right. So that was about the aldehyde and ketone, and I would say it is to a medium level. So most of them could have answered this because it involves two things. What is that? Writing down the product and also naming it properly. So you have to be extra cautious while ticking your options. So you have to be very careful with the naming part too. Not just the answer, the naming part too should be the correct one. Okay, so let's move on to the next one. So the next question is a question from electrochemistry, where I always say that there will be a question from Kohlrausch law or its applications, or you might get a question from a Nernst equation. Yes, this time we did not have a question from Nernst equation, but yes, they have targeted this particular concept, that is Kohlrausch law and its application. So there are two questions from this particular topic. There is a question where you have to find basically what are the applications that you come across for a Kohlrausch law. So it helps us to find out. The molar conductivity values of weaker electrolytes, because for the stronger electrolytes we can find it with the experimental data, but for the weaker electrolytes we need a law, that is Kohlrausch law. So with the help of Kohlrausch law, at infinite dilutions we can find out the molar conductivities of weaker electrolytes. And there is one more application that we can also find out the extent of dissociation of this weaker electrolytes. We can also find out the alpha value. So there are questions on both the topics, finding out the molar conductivity values of the weaker electrolyte and also finding out the extent of dissociation. So this is a question where they're asking us about finding out the molar conductance of the weaker acid, that is acetic acid, and they gave us the data that is necessary to find it. And we know that we have learnt, these are the lines from NCERT, there is a method to find the molar conductivities of weaker electrolytes. So you have to make a chemical reaction in such a way that they are in a combination. You have a combination of stronger along with the one which you want to find. For example, here in this case, suppose if I want to find the the contribution due to this one. So what is that I can do here? So I can take the example of, so what are the options given? So these are my options given. So let me do this question. So I have, what are the components I have? I have NaCl with me, I have HCl with me. Okay, I have NaCl and HCl with me. I also have acetate, sodium acetate with me. Okay, so let me draw a reaction where I have to find it. So CH3COOH, okay, reacting with, let's say, NaCl, okay, a stronger electrolyte, and giving us what, tell me, CH3COO minus Na plus, right, and HCl. And if you see over here, sodium acetate is a stronger one, a stronger one, a stronger one, and a weaker one. So this is the one which is weaker. So this is the weak electrolyte. So by using this combination, I can find it very easily. How can I find it? So the molar conductivity values of acetic acid will be equal to what? Very simple. You have to add them and minus NaCl. So the molar conductivity values of the acetate ion CH3COO minus Na plus, along with the molar conductivity values of HCl. So this, okay, this minus, okay, so this minus the molar conductivity values of NaCl. And we have all the values given in the question. So you have all the values given. So what is the value of NaCl given? So NaCl is about 126, okay, 126. And what about HCl? HCl is about 426. HCl is about 426 approximately, 16, okay. And what about the acetate? The acetate is about 91. The acetate is about 91. So you can make the calculation very quickly. So you will come across, see, this is almost like 426, like 516 minus 126, 516 minus 126, it is approximately equal to 390. So this is my answer. So 390 is my answer. So this is how I can find out with the units, okay. So this could also be found out, generally what you do is that to get this molar conductivity values of acetic acid, the same thing we can use. We try to add, see, we get the CH3COO minus part from here, we get the H plus part from here. So what we do, we add these quantities and we remove the contributions due to Na plus and Cl minus ions. So that is what we have done. So we have added and we have removed the contribution of Na plus and Cl minus ion. So this is how we got the answer, 390. So this was all based on the Kohlrausch law, which is stated in your NCERT book. So we need to apply it. So that is what we are going to apply, or we have to understand this and we have to apply the formula. So this comes under a moderate level, I would say, because there are chances that you might not know how to make this a thing. Okay, so this is a question which I would say to a moderate level. Right. So the next question, the correct option for the number of body-centered unit cells in all 14 types of Breve lattice. So this is the first chapter of your 12th, and most of us learn the first chapter with a lot of enthusiasm, and definitely this might have been answered by everyone. Okay, but still there might be some confusion. How do I remember what are the different type of body center, which crystal system has what type of unit cells? So how do we remember it? So this could be answered very easily. There is a sequence which you have learned, so which we tell in the class. So you just have to remember a sequence which is CT, or HMT, CT or HMT. Okay, it's like a brand. CT is a brand. Okay, for example, just to remember, CT or HMT. So you want to buy a smartwatch which are of two brands, CT and HMT. So you're confused between them, CT or HMT. Okay, so these are the different crystal systems which could be written in the sequence. All the 7 crystal systems: cubic, tetrahedral, orthorhombic, ending with the triclinic. Now, what I will do is that now I am going to add some more important things, like what type of unit cells are possible, the different Breve lattices. So let's see the primitive. This is for primitive, this is for body centered, this is for face centered, and this is your edge center. Okay, we know that you will find primitive in all the cases. Primitive is found in all the cases, right? In all the crystals you will find the primitive unit cell. After taking down all the unit cells, primitive is found in all the, that's very easier and everybody could answer that. Now what about the rest of them? The rest of them, you have to remember a number called as 213. You have to remember a number 213. Okay. So what is this 213? So just put 2 ticks here, 1 tick here, and 3 ticks here. So 213. So 2 ticks, okay, 1 tick over here and 3. So 213, and just go to the bottom, and the bottom one, okay, that is monoclinic. Monoclinic will have the end face centered one. So this is how you can write down the entire thing very easily. So you have to know the sequence, and you just have to remember this number 213. So 2 ticks, 1 tick and 3 ticks, and this one special for the monoclinic part. So now you can answer, what is that? The correct option for the following number, what is that? The number of body centered unit cells in all the 14 Breve lattice. How many will have that? Obviously the 3 unit cells, that is C, T and O, will have the 3, all the 3 crystal systems will have the body centered lattice. So this is how you can answer by knowing this table. Okay, so very simple to remember, and you can answer it very quickly if you are unable to memorize it. So that is that could be answered very quickly if you know the trick to do it. So this comes under, I would say, easy to moderate level question, because I would say easier one because it's the first chapter and everybody can answer this. I am expecting it to be answered by everybody, that is 90% of the people, right? Next one. The next question is again on the physical part, where this is a very familiar question which you have been doing from your 10th standard onwards, where you find out with the help of the mass percentages, we find out what is that called as qualitative analysis. We find out the empirical formula. So we have learned something called as qualitative and quantitative analysis, where we find out what are the elements and then we find out the percentage weights, the qualitative analysis. So where we find out the empirical formula. So this is a question where from the first chapter, mole concept, under the stoichiometric chapter, where we find out the empirical formulas. So let's find out the empirical formula and what is the data given. So there is an organic compound, and there is organic compound with 78% carbon. So there is 78% carbon, and the rest is obviously hydrogen, because the remaining is hydrogen. That's what they say. So immediately looking at this question, you should not waste your time, because empirical formula is a very easy question which is there in your NCERT syllabus. So quickly you should go for a table. What is a table? A table where you have the elements written, okay, the elements written, carbon and hydrogen, okay, and then you have to write down the percentages. You have to write down the weight in grams, okay, and then you have to write down the number of moles, okay, you have to write down the number of moles and the simple ratio, and that's it. You get your answer, the simple ratio. You have to get the simplest ratio over here. So this is how you can find it very quickly. So how do we do it? So 78%, right? So this is 78%, and obviously 100 minus 78 is 22%. So the percentages are treated as the weights because the sample has we take the sample as 100 grams. So this becomes the 78 grams of carbon and 22 grams of hydrogen. So how do you find out the number of moles? Number of moles is equal to weight by molecular weight, weight by molecular weight. So 78 divided by 12, okay, and this will be 22 divided by 1. So this will be approximately 6.5, okay, and this will be 22. And how do we get the simplest ratio? The simplest ratio is choose the smallest decimal and divide the entire thing. So the smallest one is 6.5. So when you divide 6.5 by 6.5, you get 1. And when you divide 22 by 6.5, you will be getting approximately 3.3. Okay, so this is the confusion they have created in the minds of students. So you will be confused between CH4 and CH3. CH3 is the correct answer. Okay, so this is nothing but your CH3. Maybe they are talking about C2H6. Okay, so they were talking about C2H6 whose empirical formula is CH3. So the correct answer is CH3, not CH4. So you don't have to approximate it to the next number. Okay, it is CH3 is the correct answer. So clear? So you might be confused here. So there are some students who might be confused over here. Some might have answered it as CH4, but the correct answer is CH3. Clear? Right. So that was an easier one, but could be made. No, you could have done mistake over there with that 3.3 kind of thing. Right. The next question is all about the chemical kinetics. So what is this question about? So there is a reaction where A is converting into B. Okay, A is converting into B. The enthalpy of the reaction, it means the enthalpy of the reaction is delta HR is given as minus 4.2. Okay, the delta HR is negative value. So what is the indication? Whenever you have this delta HR, which is minus 4.2, right? Yeah, minus 4.2 kilojoules per mole. So the minus value indicates it is an exothermic process. It indicates it is an exothermic process. So one indication we got. But here, why I have chosen this question is that by reading this question, students might think that there is some data given. They gave us delta HR. They also gave us about activation energy. So you get confused with what formula should I use? Do I have a formula in chemical kinetics where I can find out what kind of graph I'll get? How to find out the energy of reactants and products? Nothing like that. Okay, there is nothing to do with the data. The data is given to understand the question. So there is something called as presence of mind required from your side. So you should not be in a hurry. You know what to do, but what happens, in our sometimes in our life, the task is very simple. But because of this hurriness, what happens? We spoil the things. So there are students who will do that thing over here. So I guess there will be some students who have got confused with the values they have given us, and definitely they might have guessed that and got a negative mark. So no. So there should be no hurry. Read the question properly, understand the question, and understand the value of the data, why the data has been given. So here the data is given. Why? One conclusion is it is an exothermic reaction. And there is one more thing which is given as activation energy as 9.6 kilojoules. So this is enough for me. This information is enough for me to answer this. How? Because if I want to answer this delta HR, which is negative. So delta HR is negative means what? Tell me. Delta HR is negative means nothing but it is less than 0. So delta HR is less than 0. The enthalpy of the reaction is less than 0. It indicates that the energy, the enthalpy of the reactants is greater than the enthalpy of the products. So this is the conclusion that they are trying to give us. That is the indication they gave us. So with the value, the data which is given actually helps us to understand this important point. So this is the point that you have to understand. If this point is understood, the answer is there on the screen. So you have to understand, you have to just identify that potential energy graph of that reaction where the energies, the enthalpies of the reactant is more when compared to the product. Okay, you just have to look at the graph properly. So if you look at the graphs over here, see, this is the reactant part and this is the product part. So their energies are almost same. So here the delta H will be 0. Okay, and coming to this part, so this is your reactant, this is your product, and you can see it is lesser. The energy is lesser and this is more. Opposite, right? For us, for exothermic reaction, the energy of the reactants is more when compared to the energy of the product. That's why there is a release of energy. So this is completely opposite to what I'm thinking. What about these options now? So I have two options more. So energy of the reactant is again lesser when compared to the energy of the product. So this is also out of my options. Okay, so the only option which is left over is, you can see, the energy of the reactant. So energy of the reactant is greater than the energy of the product. So this is my correct answer. So my answer should be option number D. right? So this question is a question where you have to keep your presence of mind. So the presence of mind, the students who have read it properly, understood what actually they are asking us, because always what we see is that whenever there is a graphical question, we get we get agitated by this graph and we think in a different way. No. Whenever there is a graph, always think that there is some additional information. So whenever there are graphical questions, the question is even more easier than the question where you have to apply a formula. Okay, so this question can be answered within 10 seconds. Okay, so this could be could have been answered 10 seconds, but looking at the data and etc. graphs, you might have hurried up and you might have confused with the data and you might have ended up with a negative mark also. So be careful with such kind of questions where there are graphs. So be pleasant, read it with a cool mind and answer it. Okay, right. So that was the question from chemical kinetics, which was an easier one, or we can say a moderate level question. Okay, so the next question is from again the solid state. So students might be very happier looking at this kind of question from first. So starting chapters are the ones which you are very very comfortable and very confident about. So the first chapter, solid state, again there is a question, but this time the question is a bit what you can say, a moderate, or where you have to think a bit. Okay, you have to think a bit where you have to apply some things. So here the question is not about the regular unit cell, that is the body centered, the simple cubic, or it is not about the face centered. It is about the hexagonal unit cell. Yes, there is a word mentioned about the hexagonal unit cell in NCERT, but not elaborately. So it's very important to know about the hexagonal unit cell. So this could be only answered for those students who have some preparation for your NEET. So let's see how could this answer. Okay, so how can we answer this? So there is a basic formula which you might have seen in your NCERT book also. How do we find out the tetrahedral voids and octahedral voids? So tetrahedral voids and octahedral voids can be found out very easily. How you can find out? There is a line that number of tetrahedral voids will be double the number of close packed atoms. Okay, it means it will be double the number of the closely packed. It means the lattice points. Okay, the effective number of atoms. Z is the effective number of atoms per unit cell. It will be twice of that. And octahedral voids are equal to Z. So this is the formula which a student should know, whoever has studied about tetrahedral void and octahedral void. And we know about FCC crystal. So for an FCC, a unit cell, we know that what is the Z value for FCC? The Z value for this is how much? It is 4. So how many tetrahedral voids and octahedral voids will be there? 2 times 4 is 8 and equal to this. So it will be 4 here. So these are the values which we know already, which everybody knows. Okay, a school going student will answer this very easily. Now what about HCP now? What about HCCP? Okay, the hexagonal unit cell or the hexagonal packing. So what about this? How many tetrahedral voids and octahedral voids are there? So you can still apply the formula 2Z, okay? You can still apply, you can go with the formula 2Z, you will be getting an answer. What is that? You will be getting the answer as, so for that you should know the Z value now. So how will you find out the Z value for HCP? So for HCP, to find out the number of unit cell per unit, the number of atoms per unit cell, first you should understand the HCP crystal, okay? So if you look at the HCP crystal, okay, the HCP unit cell, if you see here, the points that you have to remember is, there are corners. How many corners are there? Tell me. So there are 12 corners, 6 up and 6 below. So there are 12 corners are there, and each contribution is 1 by 6. Unlike the cube, its contribution is 1 by 6. So there are 12 corners, 1 by 6, plus there will be face atoms on both the faces, up and below. So 2 face atoms, so 2 into 1 by 2, okay, plus there will be 3 atoms which are at the center. That there will be 3 atoms which are at the center of the unit cell. So 3 into 1. So this Z value will be how much? So Z value will be, this is how much? 2, 2 plus 1 plus 3, that is 6. So this is the number that you have to remember for HCP unit cell. So the Z value for HCP is 6. So we can answer now very quickly. So 2 times 6 is 12 and 6. Okay, so this is how you can answer this about the HCP unit cell. Okay, this is one way you can answer. There is one more way you can answer it. Very simple formula. The HCP values will be exactly 3 by 2 times the FCC unit cell. So this is one important formula which you can apply and you will get any answer. For example, if I want to know how many tetrahedral voids. So if I want to know the tetrahedral voids, what I have to do is equal to 3 by 2 into how many tetrahedral voids are there in the FCC unit cell? It is 8. Okay, so just see how much I am getting, 12. So you can apply, even you can find the Z value. How can you find the Z value? Suppose if I want to find the Z value, how will I find the Z value? Z is equal to 3 by 2 into, what is the Z value of FCC unit cell? It is 4, right? So 2 and 2 gets cancelled. So 2 3 is 6. So you can find the effective number of atoms, you can find the number of tetrahedral voids, you can find the number of octahedral voids in HCP with this simple formula. So this is a formula which will help you to find out the information related to the hexagonal unit cell or the HCP packing. So clear? So this question, if I would say, I would not say it is an easier one. I would say it is to a difficult level, moderate to difficult level. So the ones who knows how to find out the Z value would have applied this thing and got the answer, and who know this formula directly, for them it is a moderate question or a easier question. Who doesn't know this formula, it would be a moderate to difficult question. So this is a question which could be answered in 10 seconds and also in 30 seconds. So it varies on what you know about it. So this is a question where you can go wrong again. Okay, this is one more question which is from equilibrium, ionic equilibrium chapter. And by looking at the data which is there, they're asking us, they gave us two acids, they gave us an acid and a base, dimethylamine and acetic acid, and they gave us the information about pKb and pKa. So they gave us pKb and pKa. Now why I chose this question is because by looking at these values pK and pKb, I have seen the students getting confused again, because they have learned about Ka and Kb values. Now they will be confused with the pK and pKb values, and now they will start, first what you will do is that you will start writing down the reaction between the dimethylamine. What is dimethylamine? It is dimethyl, right? So CH3 taken twice, amine, it is a secondary amine. Okay, it is a secondary amine. So this is your dimethylamine reacting with, what is that? I have acetic acid, so CH3COOH. So this is a weak acid, a weak base, and this is a weak acid. So weak base and weak acid will give you a salt. What is that? A salt of weak acid and weak base, and it will be how? It will be this one. Okay, it will be CH3 taken twice, NH2 plus. So this is the salt which is formed. Okay, so we have nothing to do with the formula, the chemical reaction, nothing. So there is one small trick how to find out so I need to find out what will be the pH value of this solution. So this will be on hydrolysis. Okay, so what kind of solution is this? Because we have learned that, so we have learned 3 different kind of combinations. What are the 3 different kind of combinations? We have learned that there is salts. There are different type of salts. What are that? Weak acid plus strong base. We also come across a weak base plus a strong acid, and we also come across weak acid plus weak base. And for all the 3 cases, we have formulas to find out the pH value. Suppose if I have a combination of weak acid plus strong base, what might be the solution there? It will be it is weak acid and a strong base. It will be slightly alkaline in nature. So how do we find out the pH of that solution? So the pH of that solution is equal to, it will be 7 plus, okay, 7 plus half of, what is the weaker part over here? Acid. So it is weaker part is acid, so pKa, weaker part is acid, so pKa plus the concentration, plus log of the concentration C. C is the concentration of the salt. So this is how you can remember the formula of the pH for this combination of salt. And what about weak base plus a strong acid? What will be the solution formed here? A strong component is there, strong acid, so it will be slightly acidic in nature. So the salt solution will be acidic in nature. So here the pH of this acidic solution is 7 minus, slightly acidic, right? So 7 minus the same formula, half of pK. What is that? Here the weaker component is weaker as the base, so it will be pKb. The weaker component is base here. Here the weaker component is acid, so pKb plus log C. Okay, and now the last part, what is that? Weak acid and a weak base. That is what our question is based on. To find out this one, so how do we find out the pH of this? It is 7 plus half of pKa minus pKb, because I have both the components which are weaker, pKa as well as pKb. So these are the formulas which are going to give you the answer. So I would say this question would be a difficult one for those students who do not have an idea about these formulas, and for a student who knows this formula but with confusion, a moderate level, and the one who is very confident about the formulas like this, then definitely he will apply very quickly the options, because very quickly the data, what is the data given? pKa is 4.77, so this is 4.77. 4.77, and what is the pKb given? 3.27. So he just have to do the basic calculations. So 4.77 minus 3.7 divided by 2 and 7. So the answer will be approximately equal to 7.75. Slightly alkaline, slightly alkaline. So clear? So this is how you can find out. So you don't have to get worried about the data what is given. Okay, so this question would be an easier, as I have told you, for a student who knows the formula very well and very confident of writing down this formula. So this is a moderate to a difficult level question from the physical chemistry. So among the following alkaline earth metals, so now we are into the inorganic part. So I have a question where there is alkaline earth metal halides. So I have the alkaline earth metal halides, as you can see in the options, and what they are asking us, which one is covalent and soluble in organic solvent. It means they are asking us, see, these are all metal halides. So metal halides, what do we expect? They are ionic compounds, right? So metal halides, being ionic in nature, will not be solubilized into the organic solvents. So organic solvents are what? Tell me. They are non-polar solvents. So we generally expect their solubility to be very, very less. But you might have learned about some concept called as the misbehaving ionic compounds. So there are some ionic compounds which misbehave. What do you mean by misbehaving? They do not follow the general rules, the general characteristics of an ionic compound. It means that there is a concept called as Fazan's rule. So this question is on Fazan's rule. So the students who have an idea about the Fazan's rule, it will be easier for him to answer. So what is Fazan's rule? So according to Fazan's rule, the point is here. So according to Fazan's rule, okay. So according to Fazan's rule, we know that wherever, so there is a concept of polarization, right? So there is a concept of polarization. It means the distorting of the electron cloud of the anion by the cation. Okay, so the cation being highly charged and high ionic potential is able to pull the electron cloud of the anion towards itself. Nothing but there is a percentage of covalent character induced. It means higher the polarization, higher will be the covalent character. Okay, higher will be the covalent character. It means the ionic bond which is there, character. Okay, character, covalent character. So higher the polarization, higher will be the covalent character, and this polarization depends upon, see here if you can see, all of them are halides, chlorides. So polarization based on only the cations, like here you have magnesium ion, Mg plus 2, right? You have Be plus 2 here, here you have Ca plus 2, here you have strontium plus 2. Okay, and what is the order we can write down? Tell me from top to bottom. The first comes, the smallest one is Be plus 2, then comes Mg plus 2, then comes calcium, then comes strontium. Okay, and down the group what happens? Tell me. Size increases, size increases, size increases. And what do we know about polarization? Polarization, smaller the cation, more is the polarization that you will see. So it means it is inversely proportional to the cationic size. You will see more polarization in a smaller cation. So it means which is the smallest one over here? Size is increasing from top to bottom. It means what happens? This is the smallest one and this is bigger. It means you will see polarization. So polarization, what happens to the polarization? Polarization decreases when we move from top to bottom. Polarization decreases, covalent character decreases. Or we can say these are more ionic in nature. These are more ionic in nature and this is more covalent in nature. And the question is about dissolving these, which among them will easily dissolve into an organic solvent, which is a non-polar solvent. So covalent molecules being non-polar in nature can dissolve easily into a non-polar solvent. So my answer is beryllium chloride. So beryllium chloride is that misbehaving ionic compound which instead of getting solubilized into a polar solvent will get easily solubilized into a non-polar solvent. Okay, so that is the misbehaving characteristic of this beryllium chloride. So these are also from the lines of NCERT, but you have to apply the concept properly. That is the Fazan's rule. So if you have an idea about Fazan's rule, then definitely you can answer it. Otherwise, it would be a difficult one. So a moderate to a difficult level question from inorganic. Right. So let's move on to the next one. The correct structure of 2,6-dimethyl 1-dec-4-ene. So this question, you don't have to look at all the 4 options. How do we proceed now? Okay. So the students who have started observing the name, not the options, first would have answered this very quickly and easily. If you see the name here, the chain name is dec-4-ene. Okay, and all of them look the same. So I don't want to count the numbers. I just want to check where I will find 4-ene. That's it. Because I would see there is a lot of difference in the position of ene. So this dec-4-ene will give me the correct answer. So 4-ene. So if I check from here, 1, okay, 1, 2, 3. So this is wrong. Okay, 1, 2, 3. This is completely wrong. So there is no 4 there. What about this one? 1, 2, 3. 1, 2, 3. So this is also 3. Wrong. What about this one? 1, 2, 3, 4. This is on the 4th one. Yes, this is my correct answer. That's it. You don't have to be, that's what's called as the presence of mind. You have to read the question properly, observe the options properly such that you eliminate the things quickly. You can see till now whatever the questions I have done, I always try to eliminate the question, eliminate the options. So when you are eliminating the options, you are actually reaching closer to the correct answer. Okay, right. So let's move on to the next one. So that was an easier one, I would say. It's not difficult. It's an easier one where you have to be very careful with this. So this question, why I have put over here is that you might have wasted some time over counting the number of carbons, naming them. No, it's not needed. Just look at the name 4-ene and look for the 4-ene. You get your answer directly. Okay, so this could have been done smartly and it could have saved your time. You could have answered this, everybody could have answered this, but it is all about the time. So some students might have done it in 10 seconds, some students in 30 seconds, or some students in 1 minute also. Okay, right. So the next question is again from states of matter, which is based on the simple formula PV is equal to nRT. So whenever states of matter comes to our mind, the topics which are very important is the questions on ideal gas equation PV is equal to nRT, the diffusion principles, that is the partial law, partial pressures, the Graham's law, okay, and we come across the van der Waals equation. So these are the 4 important parts in states of matter from where the questions have been asked. And definitely, and you can see there is a question on Boyle's law. There is also a question gas laws. Okay, so there is also a question on ideal gas equation also. So gas laws, ideal gas equations are very, very important. But this question, if you see, is not that regularly being done. So we have come across single graphs, but this is something different. And what is this law here? Boyle's law. So from Boyle's law, few things we know. What is that? P is inversely proportional to V, right? Or we can say PV is equal to constant. So these are the ones which everybody knows. P inversely proportional to the volume, because with increase in pressure, the volume decreases at constant temperature, right? Isothermal conditions. And I want to eliminate some options. Which I can eliminate? You can see clearly. So there is this A and D, which is out of my context, because there is no relation like directly proportional over here. So it is all inversely proportional. So either my answer C or B would be the correct answer now. And if you see option number B and C, they are exactly opposite of each. So how do I do this? So there is a small trick how to do a question related to variables wherever there are multi-curves. So this experiment which they have given us is being done at different temperatures. What is that? At temperature T1, at temperature T2, and at temperature T3. Okay, P versus V, inversely proportional. Now it is a multi-curve. So you have this experiment was done at different temperatures, and they're asking us the order of T3, T2, and T1. So how can we do this? So there is a very easy technique to do this. We have to do a very simple thing. You can apply this to any kind of multi-curve graphs. So what you have to do is that just go for PV is equal to nRT. So what is PV is equal to nRT? So this is the equation that we use. In this, I am assuming this to be constant because we are doing it for a fixed amount of a gas. So PV directly proportional to T. PV directly proportional to T. But here if you see, they are asking us about what, tell me, T. They are asking us about the temperatures, because these curves, if you see, they are differing in terms of temperatures. So they are asking us about the temperature lines, which what are these lines, T3, T2 and T1. So this temperature variation with the change of pressure and volume, how can we do that? So we can relate two things, right? We cannot relate three things. So what I am going to do is that to relate two things, either pressure with temperature or volume with temperature, I have to make one of them a constant. Okay, so I am not making it constant. I am assuming that at a particular, so we need to draw a line which is perpendicular to any axis, y axis or x axis. So let me draw a line perpendicular to, let's say, y axis. Okay, so I am drawing a line which is perpendicular to y axis. So what is that? I am assuming, so at a constant pressure, so at some pressure P, so at some pressure P, how is this volume? What is the value of the volume this it is having at that particular temperatures? So at this particular pressure, so at a particular pressure, I want to know what are their volumes. So this perpendicular line is coinciding with these points, right? So these are my points. So these are my volumes. So this is my V1, this is my V2, and this is my V3, because it is intersecting at the third point. So V1, V2 and V3. And from here I can see that, see here what I have done, tell me, I am assuming this graph, I am analyzing this graph at a particular pressure P. It means V is directly proportional to T. So I can relate volume and temperature at a particular pressure P. So at a particular pressure P, I can see that V3 is greater than V2 greater than V1. And hence it follows the relation. So if V3 greater than V2 greater than V1, then T3 greater than T2 greater than T1. So this is how you can do it. You can try this with cutting these curves vertically along the perpendicular to the x-axis. You will get the same answer. So whenever you have this multi curves, you can go with this drawing a line perpendicular to any of the axis and relating the variables. Okay, because we have three variables, pressure is changing, volume is changing at different temperatures. So what I have done, at a particular pressure, I wanted to relate volume and temperature. So that is how I got this relation, T3 greater than T2 greater than T1. So it means the graph, what happens, the temperature increases. For a isothermal, for Boyle's curve, the curves indicate that the temperature is increasing. It means, which is my correct answer? 200, 400, 600. So this is my correct answer. Okay, so this is how it could be an answer. So I would say it is again a moderate to a difficult, because you might have come across, many of our students might have come across the single curves and they know to relate those graphs which are very simpler ones, single curve. But when it comes to multi curve, you might go wrong. So that is why I would say it is a moderate to difficult question for students who have not gone through this kind of concepts. Okay, right. So let's move on to the next one. So this is the question from a solution chapter, and from solution chapter I have seen that there are two topics. So in solution chapter usually we come across the important topics are colligative properties, obviously the hardcore topic, that is Van Hoff factor along with the colligative properties, and we also get questions from Rolle's law, and this time we got questions from both the points. So we have a question here which is relating the colligative properties including the Van Hoff factor, and there is a question, one more question where you will find out the Rolle's law. So this is how they have given. So that's what I told you. They have taken care. I would say this paper is an ideal paper, an ideal NEET chemistry paper, where they have given questions very properly, keeping in mind the scenario which is happening around us, the pandemic. So they have given us all the important topics which we usually do in our classes. Okay. So coming back to this question, so in this question, if you see, there are three different solutions are there, and if you can see, there is in all the three cases, that is glucose. So there is glucose, there is glucose, there is urea, and also there is one more, that is sucrose. So there are three different solutes, glucose, urea and sucrose, and all of them, the amount is same, 10 grams into 50 ml, 10 grams into 50 ml, 10 grams into 50 ml. Okay, so it means the concentration part is same. So how can we do this? So basically, what is the formula that we know about osmotic pressure? So pi is equal to ICRT. So do not forget this Van Hoff factor. So this Van Hoff factor will be equal to 1 for a non-electrolyte, and you can see all of them are covalent molecules. All of them are carbohydrates. Okay, or you can say a covalent molecules. So for covalent molecules, I value is equal to 1. Right, and if you see here at a particular temperature, so T is constant, R is obviously a constant, so I could say pi is directly proportional to C. Pi is directly proportional to C. And what is the C over here? C is nothing but number of moles per unit volume. Number of moles per unit volume, where again the volume is constant. Okay, concentration, volume is constant. And what is number of moles here? It is weight by molecular weight, where weight is again constant. So when you relate all the things, you will end up with one equation, that is pi is directly inversely proportional to the molecular weight of the non-volatile solutes. So this is where you get the answer. So it's very simple. See, this could have been answered very easily if you know the basics of colligative properties. We have learned that colligative properties, the colligative properties which are there, are inversely proportional to the molecular weight of the solute and directly proportional to the number of particles, number of solute particles. This line is there in your NCRT. These lines are there in your NCRT. This number of particles is nothing but I. So if you know this point, this could be done very easily. It means what, tell me? This pi is inversely proportional to the molecular weight of the solute. So using this, you can do this question very easily. It means what, tell me? Wherever the molecular weight is high, so wherever it is high, the osmotic pressure is less. And I just have to look at the weights. So they gave you the solute particles are given and you know the molecular weights. So one is glucose, okay, one is glucose, right? One is urea and the other one is sucrose. So glucose is around 180 grams. You can find this molecular weight. This is 60 grams and this is around 340 grams, 352 grams around, okay. So if you see, urea will have the highest osmotic pressure when compared to sucrose, okay. Urea will have the highest. So P2 I will say, P1 then P3. So C will be my correct answer. So the theme of this question is what, tell me? Based on this. So pi is equal to ICRT. So why I have put this question over here is that there will be some students who might have got confused with what they will do is that they will write down this expression for all the three cases, okay. They will substitute the volume value, they will substitute the weight value and then confuse and finally put a negative mark, okay. So that should not be happening. So the concept is very clear. What is that? They are just asking the relative. So whenever you get a question based on comparison, you don't have to get into the substitution process. You just have to see the relation. So here the relation is what, tell me? The osmotic pressure is inversely proportional to the molecular weight of the solute. So heavier is the molecule, lesser will be its osmotic pressure. And smaller or the lighter is the molecule, smaller is the molecule, more will be its osmotic pressure. So that is what they are asking us. Okay, so given the conditions. So this was, I would say, a moderate to a difficult question. So you might have made it a difficult question by substituting the values, but it was a easier to moderate question. So this was from the chapter solutions based on colligative properties. Okay, the next question is from coordination compounds, which is related to EDTA. So most of you have come across this name EDTA, right? So now the question is about the structure of EDTA. So it's all again the structure which is there in your books. So if you have, basically we learn about EDTA, we know about a few of the points, that it is a flexidentate, it can have it is a hexadentate, but it's very important to know the structure also. Okay, where are the denticity? So which sites are ready for the donation? So how do we remember this EDTA? So there is a technique how to remember this EDTA structure. You might have known ethylene diamine, EN, bidentate, right? So just write down that structure, ethylene diamine. So CH2, you will have NH2 and you will have NH2, right? So this is how you will write down the ethylene diamine. Ethylene diamine. We know about ethylene diamine, bidentate, EN, which you will be using, which is also used in geometrical isomerism. So now this ethylene diamine, now I am going to remove the hydrogens and I am going to put acetate ions. That's it. This is your EDTA. So this is your EDTA, okay. And you can answer many of the questions. What are the questions, tell me? The questions which you can answer is, it is a hexadentate? Yes, it is a hexadentate. Why? Because there are how many sites? There are 1, 2, 3, 4, 5 and 6. So there are 6 sites which are ready for connecting to the metal atom. So hexadentate, or we can say flexidentate, because it can show 6, it can show 4, it can also show 2, depending upon the type of metal and the conditions. So hexadentate, or we can say flexidentate. And what are the donors here? You have 2 nitrogens as well as 4 oxygens. So 2 nitrogens and 4 oxygens. 4 oxygens and 2 nitrogens. So this is my correct option. So it is purely based on the structure. Purely based on the structure. But if you know one single point that it is a hexadentate, then done. You don't have to look for any other options. This is your correct answer. Okay, if the options were like hexadentate and they would have done something to this 2 and 4, then you should be very cautious about these options. But this is a very easier question. If you know that it is a hexadentate, then done. Your answer is option number C. So it is easier to moderate level question from the chapter coordination compounds, and it is from the starting pages of your book where you learn about the type of ligands that is based on denticity. Okay, right. So the next one, which of the following molecules is non-polar? Okay, which of the following molecules is non-polar in nature? So this question is from chemical bonding chapter, and where I say that there will be always multiple questions coming from chemical bonding, and yes, it has happened. There are questions from chemical bonding. You get questions from the geometry, that is nothing but the hybridization and its application. You also get questions from MOT. There was no question from MOT, but there was a question from hybridization, the structures, the geometries. You get questions on the Lewis structures sometimes, and you also get questions on the dipole moments. So this is a question based on the dipole moment. So there will be always questions from either from dipole moment, hybridization and its application, or from MOT. So these are the main core topics from chemical bonding, and there are chances of multiple questions from this chapter. So you have to take care that this chapter has to be revised, because this comes in the 11th standard. So students who are in the 12th have to revise this chapter thoroughly, because it can give you 8 marks. Okay, it can give you 8, maybe sometimes 3 questions. So that much is the importance of this chapter, chemical bonding. So what is this question about? Which of the following is non-polar? So they're asking non-polar, not non-planar. So this question is based on dipole moment. Okay, so dipole moment, this question is based on dipole moment, and you know that dipole moment is a parameter which tells us about the polarity of a nature. So higher is the dipole moment, which tells us about the polarity of a molecule. So in covalent molecules, the polar molecule, the polarity of a covalent molecule can be understood with, we say no, there is percentage ionic character in covalent molecules, percentage covalent character in ionic molecules. So this dipole moment will tell us about the percentage ionic character in covalent molecules. So generally we say that covalent molecules are non-polar. No, not all molecules are non-polar. There are molecules which are polar covalent bonds, and that could be understood with the polarity indication, that is dipole moment. And this polarity, once we know about the polarity, this could be linked with various other factors like solubilities also. So there are questions on solubilities also. So dipole moment connected with polarity, polarity connected with the solubilities. So right now the question is very simple. They're asking us about the polarity. So, and you know about dipole moment, it's a vector quantity which depends upon the geometry. If it is more than diatomic, for diatomic molecules directly you can find out the dipole moment depending upon the electron negativities. But for more than 3 atoms, a molecule made up of more than 2 atoms, you have to go for the geometry because it's a vector quantity. So now if you want to go for the geometry, now what you have to apply? You need to know the shapes of these molecules. It means you need to know the hybridization. So this is a question which is going to check your hybridization, how are you able to apply the hybridization concept and know the geometry of the molecule, and from the geometry of the molecule, how are you able to find out the dipole moment. So POCl3, okay, let's start with the POCl3 part. Okay, let's start with SbCl5, okay. If you look at the SbCl5, if you can see, the steric number comes to be how much? It is 5 plus 0. 5 sigma bonds plus 0 lone pairs. So it is 5, okay. So it means steric number is 5, it means it is sp3d, it is sp3d2. So if it is sp3d, it is sp3d, right? So sp3d means sp at the center, okay. Then I have chlorine over here, chlorine over here, chlorine over here, 1 above and 1 below. This is your trigonal bipyramidal. And how do we find out the dipole moment now? If you can see the angle in this plane, so these 3 chlorines are in the plane, equatorial plane, which are separated by an angle of 120 degrees. And you know the resultant of 120 degrees, what is that? So the resultant will be this side, and the resultant of this one will be this side, and both of them are equal, so they both get cancelled. Because of 120 degrees difference, the resultant is also equivalent to one of the vector. And here obviously we know that this will be upward and this will be down, so both of them will get cancelled. So basically the mu of this is 0, mu of this is 0. They are asking us about non-polar, so my answer is SbCl5. But this might not be the first option for many of our students, so let's go for other options also. What about NO2? NO2, you might have learned about NO2 in your p block elements also, okay. Odd electron, so there will be an odd electron over nitrogen, and it is a bent shape, it is not linear, it is bent shape. Okay, this is where you will get confused, whether I have to go for this option, whether I will go for this option. If you're not aware of this bent shape, then you will go for linear, and you will then confuse between whether to put NO2 or SbCl5. So that's the point which has to be noted down. So this is a bent shape, and where the net dipole moment will be towards the nitrogen. So it is not 0. What about POCl3? POCl3, you know, it is a tetrahedral. It is a tetrahedral molecule. So tetrahedral molecule means what, tell me? So the because chlorine being more electronegative, the net will be downwards, and oxygen being more electronegative, it will be up. And if you can see, both of them are unequal. Since they are unequal, the mu will not be equal to 0. Okay, this will not be equal to 0. It will have some dipole moment, but little. But here also you will have dipole moment which is not equal to 0. What about the last option, CH2O? It is formaldehyde. This is your formaldehyde. Okay, and you can see very clearly it is here, okay, and this is up. Obviously, mu is not equal to 0. So I have done all the options for students whose options are not in this sequence. So the correct answer is what? They are asking us about the non-polar. So non-polar means nothing but mu equal to 0. So this is my correct answer. Where is SbCl5? This is my correct answer, A. So here the question is trying to check two different concepts, whether you know how to write down the geometries and whether you know about dipole moment. And what about dipole moment? In dipole moment, it is geometry, depends upon the geometry because it's a vector quantity. And there is also a point where you will do mistake, that is with NO2. NO2 has an odd electron with it, because of which there is a little bit lone pair bond pair repulsions, and there is a little bent in the structure. It's not linear. Clear? Right. Okay, the next one. From the following pairs of ions, which one is not isoelectronic pair? So what do you mean by isoelectronic pair? We have come across isobaric, isotonic. This comes usually in the chapter called as atomic structure. So in atomic structure, you learn about the different type of species: isobaric, isosters, isotonic. So what is this isoelectronic? The name itself is saying isoelectronic means nothing but the cations which have the same number of electrons. Okay, right. So let's check each one now. So how do you check? So manganese is how much? Tell me. Manganese is 25. So 25 minus 2 is how many electrons? You'll have 23. Iron is 26. 26 minus 3 is how much? Again 23. So they're asking us what? Tell me, from the following pair of ions, which is not. So this is the point, has to be noted. So before you start solving it, read the question properly. They're asking is which is not, because by reading this, see, if I would not have seen the not part, I will start doing the question and I will see that all of them are same. So that's what we have to do. You have to be very careful with that not or incorrect. You have to keep that mind, keep that thing in your mind, which is not. Okay, so we need to go for the not part. So what about this one now? See, both of them are having different atomic numbers, and you have to remove 2 electrons from them. Definitely they will be not isoelectronic. So this is my answer, because different atomic numbers, right? One is 25, the other is 26, and I am removing minus 2. So 26 minus 2 and 25 minus 2 is different. So this is my correct answer, isoelectronic. But what about others now? Oxygen is 10, right? It is minus, right? Sorry, 8 plus 2, 10, and this is 9 plus 1, 10. So both of them are again isoelectronic. Sodium, 11 minus 1 is 10. Magnesium, 12 minus 2 is again 10. So again isoelectronic. So be careful with the word not, incorrect. So these are the ones which will waste our time. Finally, we will identify that yes, it was in what is that? It is not correct, or they are asking us about the incorrect. They are not asking us about the wrong statement. We will get it, but there is what? Tell me, there is a wastage of time. So because of this wastage of time, what happens? Tell me, it is going to reflect on the next part. Okay, right. So that's the point to be noted. So be careful about these not, incorrect things. The next question, if you see here, choose the correct option for the total pressure. We have to find out the total pressure, and I have a mixture of gas. These are my options. So I have a mixture of gas. What is that? I have oxygen, and I also have hydrogen with me. Okay. So now this question, you know, I would put this question as a moderate or difficult. Why? Actually, this is an easier question where you have to apply very simple formula. PV is equal to nRT. This is PV is equal to nRT. But what happens when a student is reading this question? He will see 2 different gases, 4 grams of oxygen, 2 grams of hydrogen. Then the first thing that comes to his mind is partial pressure. So once you enter into the partial, if you try to think the question, if you try to understand the question in terms of partial pressure, then you are gone. Because this is a question which is not related to partial pressure. This could be done with very easily based on PV is equal to nRT. what the only thing you have to do is that, see, you have the R available, you have the temperature available, you have the number of moles, I think you have to find the pressure. So volume is available as 1 liter, I guess. Volume is 1 liter. So all the data is given. They're asking us about the total pressure. The only thing is that you have to find number of moles. What is number of moles here? It is the number of moles of oxygen plus the number of moles of hydrogen. So what is the number of moles of oxygen and hydrogen? So number of moles of oxygen will be, it is 4 grams, right? So 4 by 32. And this will be, hydrogen is how much? 2. So 2 by 2. So this is 1 by 8 and this is 1. So it is 9 by 8. So this part is 9 by 8. So rest everything is easier. So I would say it is an easier question where you just have to find out the total number of moles. But there are students, there will be some students who will get confused with this mixture of gas. By looking at the word mixture of gas, they will go into what? Partial pressure and then gone. So you have to be careful. Read the question. So if you see, always reading the question is the first important thing. If you read the question carefully, 70% of the question is done. So you are done with the question because all the ideas, everything is ready with you. You just have to apply them and get an answer. So read the question with a cool mind. Okay, right. Let's go for the next one. So as I was telling you that there is one more question from solution from electrochemistry part from Cold-Dash law, and it is on application part. So I would say this question, I would say it's a difficult level or moderate to difficult level. Okay, because it's on application part. So usually whenever it comes to application part, we are not that great at application part. So the students who are sincerely with proper discipline, if they are properly following the guidelines of their teacher, then definitely this question you might have done this question a lot of times in your homeworks or illustrations or somewhere in the worksheet. Right. So how do we do this? So in this question, there are different parts are there. It's not with a single formula. You have to use multiple formulas. So basically it starts with, there is acetic acid. Okay. So for acetic acid, which is ionizing, so you have to apply two things over here. What is that? The ionic equilibrium. So you need to find out the Ka value. So what is the Ka value here? Tell me. Basically it is C alpha square, which you have learned for mono acids, C alpha square by 1 minus alpha whole square. Okay, 1 minus alpha, right? So here in this case, if you see, for dilute solutions, I can write Ka is equal to C alpha square. Ka is equal to C alpha square. So they're asking me what? Tell me. They're asking me what is the dissociation constant. Dissociation constant is nothing but Ka. So to find Ka, concentration is given. How much it is? 7 into 10 raised to minus 3. I just need to find alpha. Now how can I find alpha now? Okay, how can I find alpha? We have learned that in Cold-Dash law, there is an application of Cold-Dash law. We can find alpha with this simple formula. What is that? Molar conductivity of some concentration C divided by molar conductivity at infinite dilution. So this is a formula which will give you alpha value. Okay. So is the molar conductivity given at this concentration? Yes, it is given. How much? 20. So this part is given in the question. So they made us easier for us. So this is 20. I just have to find the infinite dilution molar conductivity for a weaker electrolyte. It means you have to apply the basic concept. What is that? The molar conductivity of a weaker electrolyte at infinite dilution is equal to the sum of the contributions of all the ions. It means I need to add the contribution of both the ions. It means this one and this one. It means CH3COH means I have to add what and what? 350 plus, that is due to the H plus ion, and due to the acetate ion it is 50. So basically it is 400. So 20 by 400. So 20 by 400 will give you what? So alpha value is equal to 0. It is like 1 by 20, right? So 0.05. So this is your alpha value. The question is not yet over. It is alpha. You have to go back and substitute in this formula. So Ka is equal to C alpha square, 7 into 10 raised to minus 3. Alpha is 0.05 whole square. So here also we will do mistake. We will forget the square part here. So I am telling you all the possibilities, you can do a mistake. So we forget about the square and we just multiply and look at an option and tick it. No. So be careful with the calculation part also. So on calculation you will get it around 10 power minus 5, that is 1.75 into 10 raised to minus 5. So this is a question where you have to, get the Ka expression, C alpha square, and you should know how to get this alpha value with the help of molar conductivity values at a concentration C and infinite dilution. So I would say it is an application oriented question, yes, which not everybody would have answered this, but definitely the students who are preparing, who are having some preparation towards NEET from their teachers, from their NEET teachers, definitely they would have answered this. My students would have definitely answered this because I have done this a lot of times in my class. Okay. Next one is a question based on Arrhenius equation. Okay, so it means it is from kinetics. So let's see what is the question about. So Arrhenius equation. So what is the expression that we know? This is the expression that we know, minus EA by RT, right? K is equal to A into e power minus EA by RT, and we have come across this Arrhenius equation when we were learning the the characteristics of K. How is this K getting affected with different quantities? The factors affecting K. So temperature is one such case where it can change the value of the K. So the effect of K over the rate constant can be studied with the help of Arrhenius constant, right? Now the question is not on this expression, the question is on the graphical one. So whenever the graph comes, again we not being a mathematics students, not related to mathematics, we get scared of these graphs, but it is actually easier. I'll show you how. So apply logarithm on both the sides. Okay, so you should have some basics of logarithms. So I'm going to apply natural logarithms. So ln k is equal to ln a. It will be minus EA by RT. So this is one basic thing that you should know. Once you get this equation in terms of natural logarithm, your question has been done. Why? Because I need to just draw a graph. I need to draw a graph between what and what? Between temperature and ln k. Okay. So how do I get it? Y is equal to mx plus c. So this is my y, this is my mx, and this is my c. And you can see there is a negative term given. So negative slope. So it is a negative slope. And what is the slope here? Minus EA by r. Minus EA by r is the slope, and this is my intercept, which is what? The intercept is ln a. So they are asking us what? Tell me. They gave us the slope value. You can see here. They gave us the slope value. So with the help of this slope, I am going to find it. Is this equal to 5 into 10 raised to 3. So this and this gets cancelled, and EA is equal to 5 into 10 raised to 3, that is kilojoules, into the R value. You have your answer ready. So I would say this question is again a moderate to a difficult level question, because you have to draw a graph and understand the graph. You should know what is the slope also, right? So I would say this question is from moderate to a difficult level question, where you just have to apply the formula that you know. Everybody knows this formula. Everybody knows this formula. Everybody knows the purpose of this formula also. But how can we use this formula in terms of graphical? So there are some graphs in terms of logarithm also. So this was a question based on a graphical pattern. Okay. So I would say not everybody might have answered this, but definitely the students who are having some type of coaching, definitely they would have answered this. Okay. So this was a question from chemical kinetics based on a graph, right? So the answer would be approximately, see, the multiplication is about R value is 8.314, right? So it is, it will be around 40 something. So this is my answer. I don't have to do calculation also. So that is one more thing. So whenever you have calculations, keep on observing the options also. You don't have to do it completely. Just when you are starting the calculation itself, you have to assume the value what you are going to get and what are the options given. If they are not so close, then definitely you can go for approximations. If they are very close, like suppose the options were 41.9, 41.42, 40.9, then you should go for the end calculation. But whenever you have a lot of difference in the calculations, then definitely you can go for your answers. Okay, right. Okay. So this is the question from thermodynamics chapter. Usually we get a lot of questions from thermodynamics chapter, but I could see there is only one question from there were two questions. One was a question which was straight away, that's called as the Mayer's relations, Cp minus Cv is equal to R. So there was a question from there, and there is a question from the conditions. So here, why I have put this question here is that this is easier for those who read the question properly. See, so read the question. There are conditions given: irreversible expansion, and there is one more, isothermal condition. Irreversible, isothermal. So based on that, I am going to answer this. How? So when I look at isothermal, temperature is constant, right? So temperature is constant means delta T will be 0. And when delta T is 0, I should know one relation between the temperature, because internal energy is dependent on temperature. It is a function of temperature. So delta U. So delta U is equal to, what is that? NCV delta T. So delta T is 0, right? If this is 0, then this is also 0. So delta U is equal to 0. So either option C or option A will be my correct answer. Now look at the other condition. What is the other condition? Irreversible. It is an irreversible expansion. If it is an irreversible expansion, what will be the delta S total or delta S universe? Though delta S total irreversible, it is greater than 0. So delta S will be greater than 0 because of expansion. If it was equal to 0, if this condition was not given, if it was a reversible condition, then delta S is equal to 0. So my correct answer is this one because it is irreversible. If it was reversible, this is my correct answer. So there is a chance that you might have got confused with these conditions. So read the question properly, understand the data what is given, and then go for the concept, right? So that's how this answer should have been done. I would say it is moderate question, easy to moderate. So easy for them who have read the question properly, and moderately for them who have to look at who doesn't know about the relations. Okay, right. So let's go for the next one. So there is one more question from solution chapter, as I have told you, Rolle's law and colligative property. So we have done a question from colligative property. There is the question from Rolle's law, and if you see this question also, again I will say it is between easier and moderate. Why I would say it is between easier and moderate? Because usually whenever they give you a question on Rolle's law, they will give you the amount of both the components, component A and component B. From the weights of component A and B, you will find the number of moles of component A and B. From there you will find the mole fraction of A and B, and you will finally apply this to the total vapor pressure. What is that formula? P naught A XA plus P naught B XB, right? So this time, instead of giving you straight away the weight of component A and B, with which you can find out the number of moles of A and B, from where you can find out the mole fraction of A and B, now they have made it a little bit tricky. What is that? They gave us the ratio. What did they give us? They gave us the ratio of component A and component B. I am calling component A is my benzene and component B is my component A. Benzene and octane. So it is component A is benzene and component B is octane, benzene and octane. So Na and Nb. So using this ratio, how will I get my Xa and Xb? Because P naught A and P naught B is ready. What is my P naught A and P naught B? This is my P naught A, which is ready in my data, and P naught B is also ready. So I am only waiting for my Xa and Xb. So can I use this 3 by 2? How I am going to use this 3 by 2 now? So this ratio, if you see, can be converted into Xa and Xb. What is that? What is Xa and Xb? Tell me. It is a ratio of the moles. So you just have to do what? Tell me. Na by total number of moles. Nb by total number of moles. What does that tell me? The ratio given is 3:2. So this is 3 and this is 2. So what you will do? This will become 3 divided by 3 plus 2. 3 divided by 3 plus 2 is 5, 3 by 5, and this is what? Tell me, this is 2 by 5. So this is my Xa and this is my Xb. So the students who have identified this Xa and Xb values, then you are done. You are only left with substitution. So P naught A is 280 mmHg into Xa value is 3 by 5 and P naught B value is 420 into Xb value is 2 by 5. So there will be students who you might who have stopped at this particular point. So this is what you should have learnt. What is that? They gave us the ratio of Na and M. You can also find the C. We know that mole fraction ratio. Mole fraction ratio is equal to number of moles ratio. So from here also some people can do that because the ratio is given. Na by Nb ratio is given 3:2. So you can use that expression and find Xa and Xb. Clear? So it is 3 by 5 and 2 by 5. So whoever has identified that ratio, then just write on the formula. Don't write the formula, just substitute it. What is that? 280 into 3 by 5 into 420 into 2 by 5. You will get your answer. And it will be approximately equal to 336. When you substitute the values and get an answer, it will be approximately equal to 336 mmHg. So that was the point which I wanted to show, where you might have felt some problem. Okay, so this was a question again from Rolle's law, which was easier to moderate level. Okay, the next question is based on from the ionic compounds, or we can say it is a question based on the spin magnetic moment. So this is a question again from coordination compounds, where you have to take into the consideration something called as the CFT, crystal field theory. So in crystal field theory, you might have learned about there is something called as see, there are two types of ligands given. What are the two types of ligands given? That is the cyanide and H2O. So you might have come across something called as strong field ligand and weak field ligand. So this is a strong field ligand and this is a weak field ligand. So what is the difference in that, tell me? So whenever we are looking at the configuration, the electronic configuration of the central metal atom, what will happen, tell me? Pairing happens. Because of the less energy gap between the T2G and EG, what happens? There is pairing happening in the case of strong field ligand, and there is no kind of pairing happening in the case of forced pairing happening in the case of weak field ligand. So based on that particular point, you can answer this. So I have identified the two ligands. Now I am going with the different type of ionic ions I have. So in this case, if you see what is the oxidation number of Fe, it is in +3. This is also in +3. Okay, this is because H2O is a neutral ligand, so +3. This is again +2 and this is again +2. So it means what, tell me? I have two types of ions, Fe +3 and I have Fe +2. Okay, now what is the configuration of Fe? So Fe is how much? 26, which is 4s2, right? Argon 4s2 3d6, 4s2 3d6. So what will be the Fe +3 configuration? It will be +3, right? So 2 electrons gone from 4s, so it will be 3d5. So this is 3d5 and Fe +2 is 3d6. Okay, so I have done what, tell me? I have identified the ligands and I am ready with my configurations of my ions. So now I have to look at how are they arranged. So whenever there is a strong field ligand, what happens, tell me? There is a forced pairing happening. So what will happen, tell me? These 5 electrons. Okay, so these 5 electrons, what will happen here? So these 5 electrons will start pairing. So this is how you will have, okay. So there will be one unpaired electron left over. So these 5 electrons after pairing will be left over as one unpaired electron. And in the case of Fe +2, what happens, tell me? There are 6 electrons in the d orbital. So it means you will have 1, 2, 3, 4, 5, 6 and there will be 2 empty. Okay, so this is about the 3d6 configuration in the case of strong field ligand. Now in the case of weak field ligand, what happens? There is no, the gap is more, so there will be no transfer of electrons from T2G to EG level. So it means 5 electrons will stay as the unpaired electrons, 1, 2, 3, 4, 5. Okay, and what about the 6 electrons? So they will be as 1, 2, 3, 4, 5 and 6. So you can see how many unpaired electrons are there. So here the number of unpaired electrons in this case is what, tell me? Here the unpaired electrons are 1, okay, and here it is 5, here it is 0 and here it is 4. So once we know the number of unpaired electrons, you can easily answer the magnetic moment. What is the formula of magnetic moment? Mu is equal to root over n into n plus 2 Bm. So these are the unpaired electrons. So you will get, but just substitute these values, 1 into this, you get your answer. 4 into this, you get your answer. 5 into this, you will get your answer. Okay, so there is a small shortcut I will give you how to get these answers. So there is a small thing you can remember that. So the number of electrons. Okay, so what you can remember is the number of electrons and the mu value. So they are somewhat related. How you know? Whenever there is one unpaired electron, the mu, when you substitute 1 in this, what is that? Where is that? When you substitute 1 into this, so 1 into 1 plus 2, okay, that is root 3. It is approximately 1.73, right? So 1.73, 1.73. Whenever you have 2, what happens? You will get 2 point something. Whenever you have 3, 3 point something. Whenever 4, 4 point something. Whenever 5, 5 point something. So basically you don't have to find these root values. Okay, so this is a very important observation that you can do. If you have done a lot of questions on magnetic moment, then you have seen this, you might have seen this, or your teachers might have taught this. So very simple, right? So if you know the number of unpaired electrons, you just have to look at the options. See, wherever I have 0, so wherever I have 0, obviously it is 0. Wherever I have 1, so 1 unpaired electron, right? So 1 unpaired electron means in the case of Fe plus 3 Cn. So Fe plus 3 and Cn. So this case is nothing but this one, 1.73. I have 4 in the case of Fe plus 2 and H2O. Fe plus 2 and H2O. Fe plus 2 and H2O, I have 4. So it means this will be 4 means 4 point something. And what is the next one? I have 5 here. What is this case here? Fe plus 3 and H2O. Fe plus 3 and H2O. Fe plus 3 and H2O. So this is this one. Done. So see how easily it could have been done if you are very clear about the electronic configuration, and if you are aware of this part. If you are not aware of this part, then this is a difficult one. You will lose your time over here looking at the correct one. So this is what, if you are having a clarity over the CFT theory, the crystal field theory, and the electronic configuration, and a smart way of understanding the mu value. So this is how you can understand. Sometimes you get a questions where they will give you the mu value and they'll ask you how many unpaired electrons. Suppose if they gave you 5.92, it's very easier, right? 5.92 means nothing but 5 unpaired electrons. If it is 4 point something means 4 unpaired electrons. 3 point something means 3 unpaired electrons. So you don't have to apply this formula. So this formula is not so used. So find out. Okay. So this table you can use it directly. So this is how you could have answered it very quickly. Might be you might have taken 10 to 20 seconds if you are very clear about the concepts. Otherwise, it would take 2 to 3 minutes to be done. So this was a concept based on the spin magnetic moment and coordination chapter related to CFT. So very important topic also. Right. So probably we are at the end of the questions. So we have two more questions, two more important questions to be discussed. So this is a question on again inorganic properties, where they have given us the sequence of the properties, and they're asking us which one of the following arrangements and the given sequence is not. So again they're asking us about not. So they are trying to waste our time. So we should be smart enough not to waste our time. Read the question properly. You don't have to waste your time. So not, okay. So they're asking about the not. So what about this? Tell me, if you see over here, all of them, see option number 1, 3 and 4, all of them are hydra acids. Okay, it means they are the ones who can release hydrogen. So whenever we talk about hydra acids, in this case, top to bottom, what happens? Tell me, the bond dissociation energy decreases because the bond length increases. Because the bond length, for example, if I take HI, so HI, if you see, iodine being bigger in size, the bond length is more. So H can be released very easily from HI when compared to HF. So when we go from top to bottom, as the size is increasing, their capability of giving away the hydrogen is much easier. So what happens? The acidic nature increases from top to bottom. So acidic nature increases from top to bottom, correct? And one more thing is that they will confuse you with these terms, Ka and pKa. Okay. Ka tells us about the strength by which the hydrogen can be released. It tells us about the acidic strength. More is the Ka value, more acidic. And pK and Ka are inversely related. Why? Because it is minus log of Ka. So they are inversely related. They are inversely related. So higher the Ka, lesser will be the pKa. So be careful with that. So increasing acidic strength, yes, correct. What about this one now? They are asking us about the oxidizing ability. The ability of these molecules to provide oxygen. This is also based on bond dissociation energy. From top to bottom, what happens? The size increases and easier to break, easier to provide, because the bond length increases, easier to break it. So higher is the bond length, easier to break it, and bond dissociation energy is less. So we can, these can provide. So the molecules which are at the bottom are good oxidizing agent, I would say. So this is also a correct one. Oxidizing power increases down the group because of decrease in bond dissociation energy, and obviously increase in acidic strength, yes, correct, because down the group the ability increases because of lowering bond dissociation energy. And what about pK? Just now I told you, as the K from top to bottom, Ka value is increasing. If Ka value is increasing, the pK value will decrease. So it means this should be decreasing pK value, and my incorrect statement is this one. Okay, so this is again where you have to be very cautious about the statements. So any mistake done in terms of relation, if you have gone with the, if you are not if you are not clear about the Ka value and pK values, then definitely you will come across this confusion and definitely something wrong. So you will be confused. It feels like all the statements are correct. If you are not clear about this one, what you will feel? All the statements are correct, but I have to put one of them, right? So you will put any one of them with a minus sign, you will get a negative mark. So you have to be very clear with the concepts K and pK and the statements which are given, right? Okay, the next question which we are going to do is again from organic chemistry, and as I have told you that this is a question where I was telling you that there are organic questions which are straight away from the named reactions. There are questions from a test of identification. There are reactions based on mechanism, right? So this is a question based on the identification. So this is a question where we have to identify, and this is there in your NCERT, but not elaborately, but there. Okay, what is that? It's Hinsberg reagent. You can go back and check your NCERT books. There will be definite a word mentioned as Hinsberg reagent. So what is this Hinsberg reagent? You will see this concept in the chapter called as amines, where it helps in the identification of primary, secondary and tertiary amines. Okay, so it means I can just go wrong with option number C. I eliminated option number C. I am left with 3 different options, where I have the first option primary, the second option is tertiary, and the last option is secondary. Okay, so I have the primary, secondary and tertiary with me. So how do I remember this kind of test? Okay, so if I go into the easier method of understanding, you can remember this with a small table. So I have the primary amine, I have the secondary amine, and I have the tertiary amine. Okay, and you just have to make a table where, okay, so where what I am doing, I am going to add my Hinsberg reagent, that is pH. This is my Hinsberg reagent. Okay, this is my Hinsberg reagent, which reacts with the hydrogens available with the amine. So what happens over here? So here yeah, so this is my Hinsberg reagent, which I am going to use for this particular test. So it has been seen that with the Hinsberg reagent PHSOOCl, so with this reagent what happens is that and I am going to do one more confirmation test. I am going to add the Hinsberg reagent followed by adding NaOH, excess NaOH. It is a solubility test. So there are 2 types of tests. First is identification test, and the second part is confirmation test. So first we have to identify, right? So how do I identify? It has been seen that with Hinsberg reagent only primary and secondary will react, and there is no reaction with the tertiary amines. So this is how I can separate out the primary and secondary amines based on the first part. And on the second part, now what I'm going to do after this step, what is happening is there is a formation of a solid which is again solubilized in a polar solvent. So that is why I am using NaOH in alkaline solution, because the solid which is formed here will be slightly acidic in nature. It will have that acidic hydrogen with it. So this acidic hydrogen which is there with this primary and secondary, which is there after the formation of this, will react. If there is presence of that acidic hydrogen, then it will react with it will be solubilized into NaOH solution. If it is not there, it will not. So what will happen is that it has been seen that primary having that acidic hydrogen is able to dissolve into it, and in the secondary, say in secondary the hydrogen is lost because of this reaction. The hydrogen is lost, so there is no that acidic hydrogen available, so there is no solubility. So this is how you can remember the Hinsberg's test. It means primary will go on giving the confirmation test, secondary will stop at the first step, and tertiary obviously will not respond to any kind of test. So this is how you can use this table to conclude at this answer. So my answer will be what? Tell me. Identify the compound that will react with Hinsberg reagent to give a solid which dissolves in alkali. It means it is confirming itself also. It means it is primary amine. So where is the primary amine? This is your primary amine. So this is my answer, option number A. So this I would rate this question as moderate to a difficult level question, because yes, it is mentioned in your NCERT books, but not accordingly. Not about the confirmation part. So they have just informed about the Hinsberg reagent, that it reacts with it and it helps in the separation, but not so elaborate like this. So this is all about the chemistry part. So we have discussed, so here we have the questions, whatever we have discussed here over here are the questions which I feel you might have gone wrong with, or which I would say are moderate to difficult level of questions. There are some easy questions also where you might have gone wrong while you're reading the question, or with the options, or with the formula that you can apply. So I have shown you all the different questions where you might have gone wrong. So this is how I showed you the correct way of answering it. So that's it from the chemistry part. I hope you guys have got some idea about how do we attempt the questions from NEET. The NEET questions are not difficult, they are easier if you prepare properly. Preparing properly is nothing but going to your live classes. We have the live classes where we give interactive live classes. We discuss lots of illustrations on each concept, where we have then we followed by regular homework sessions. There are worksheets which we give them. So because of this rigorous practice over the NEET syllabus, the NEET questions, the previous year questions, the question bank which we have, all the questions which we make them the students are practicing throughout the year with us, and they are all getting ready for NEET. So this is how they are preparing for the NEET examination. And that's it from chemistry, and I'm sure you are eagerly waiting for your physics teacher to discuss your physics question, and I would request Goutham sir to continue the discussion of physics. Thank you.
Hope you can, yeah, all right, fine. All right, so let us let us go to the physics part of the discussion, question paper analysis here. Now coming to the physics part, right? So we have seen that the syllabus wise breakage, right? If you look at the syllabus wise breakage, like I said, so you had 2/3 of the question. So you can say 66% of the questions, 65 to, yeah, you can say 65% of the questions were from 12th standard, all right? So it was favorable for 12th standard students, right? And coming to the unit wise distribution, right? So electrodynamics, you had lots and lots of questions asked from electrodynamics. It shared the, it actually took the major chunk of the complete questions, all right, fine. And so like I said in the beginning of this video, right, in beginning of this session, so the question paper coming to the physics part, right, the questions were tricky, you can say. It was not easy, I agree to that, right? And here I have chosen few questions that are moderate to difficult, right, so that I can discuss with you people, fine. And let us go to the questions, right? And one important thing what many of you don't want to do is that you don't want to put the problem in the form of a diagram. That is really important. At least in this paper, you'll, when I'm solving the questions, you'll get to know, right? So read each and every sentence. I'm talking especially about physics, okay? Read each and every sentence. Please put it in the form of a diagram. You will get an idea. You will see an approach to solve the question, all right? So that is how one must solve questions in physics, all right? Fine. So I have few questions here. I would like to discuss with you people. All right, so let us solve here. Now this is tricky, you can say, right? So the equivalent capacitance of the combination shown in the figure is, right? So this is the figure. Now the options are given 3C by 2, C by 2, 3C by 2, 3C and 2C, right? Now this is straight out of NCERT. I cannot say that, right? So because you have this, just a second, let me choose the pen. All right, fine. So here, the question, it was not from NCERT, at least not in any of the worked example problems or your exercises, right? Okay, so it was obviously but the concept you have studied it in your NCERT, right? Okay, nice. Now let us rate the difficulty level later. All right. Now you need to find the capacitance about these 2 points. Call it as AB. Fine. Now, as you can observe, there are 3 capacitors, right? Now, all of them are identical, right? Now, the terminals that are here, right? Now, if you want to identify if you want to find the equivalent capacitance, say you are connecting it to a battery, right? Now, when you connect it to the battery, you will notice that the capacitor number 3 here, say 1, 2, 3, let me call it as capacitor number 3 here, right? Now, both the terminals, they are connected to the same plate, right? So, they are at the same potential. If they are at the same potential, you don't get to see any charge on the capacitor. It's as if this capacitor that you have placed here, it's dummy. It is not taking part in the circuit at all. It is not getting charged only, all right? So, what you can do is you can remove this capacitor from the circuit here. It is present physically, but electrically it is not giving any contribution to the circuit, all right? So, obviously you have these 2 capacitors in parallel. So, therefore, the correct answer is 2C. Capacitors are in parallel, so they'll get added up C and C, you're getting it as 2C. So, this was not remembering, right? So it was understanding, and if you look at understanding or applicative based, so it was actually understanding here. So I can say 70% of the students would have solved this, all right? Okay, so this was about this question, all right? It was a moderate question, you can say. Like you can see that it was tricky, all right? Fine. So let us go to the next one. All right. So a radioactive nucleus, they have given, undergoes spontaneous decay in the sequence as shown in the question. So where Z is the atomic number of the element X, the possible decay particles in the sequence, right? Now what is the sequence? So first this element, it excuse me, right? So it decays to B, then B to C, then C to D. Now if you notice from, that is the first decay, right? So you have the atomic number getting decreased by 1. So where do you think this will happen? This will happen in beta plus decay, right? So first one is beta plus decay. Then if you look at the decay from B to C, right? So you have the atomic number getting reduced by 2, right? Getting reduced by 2. So that is definitely alpha decay, right? Now from C to D, if you observe, you will see that the atomic number increases by 1, that is minus 3 to minus 2. So obviously you have beta plus, alpha and beta minus. Okay, so this was not that difficult, you can say. So the correct options are this. Now the correct option is beta plus, alpha and beta minus. Okay. Now speaking of the question, right? So you have problems like this in your NCRT, I'm sure of it, right? So this was remembering, I can say, right? So somewhere in between remembering and understanding. Now you can say around 52 or you can say 60 students would have answered, 60% of the students would have answered this question, all right? Okay, so let us go to the next question. Right, so it says the electron concentration in an N-type semiconductor is same as the whole concentration in a P-type semiconductor. Okay, an external field is applied across each of them. Compare the currents in them. Okay, so now you have alright, so the options are like this. So current in N-type is greater than current in P-type, then no current will flow in P-type, current will only flow in N-type, then current in N-type is equal to current in P-type. Now finally you have current in P-type greater greater than the current in N-type. Alright, now what happens here is that you have this N-type semiconductor and P-type semiconductor, right? Now in N-type semiconductor you know that you have additional electrons. It's not that it is negatively charged or anything, right? So when you add pentavalent impurities, you'll get one extra electron, right? So you know that these electrons, right? So even if you apply a little bit of energy, so they'll start conducting, right? Now when you learn about holes, you will study that they are actually not positive charges. They are vacancy for electrons, right? So when you apply an electric field, what happens is that the minority carriers, right? So say you are taking a P-type semiconductor and you are applying a potential difference, right? Now same thing you are doing here also, N-type, and you are applying a potential difference, right? Electric field. So you are applying electric field, you know, E is equal to V by D. That relation if you use, you can understand that there is also a potential difference across the ends, right? Now this electric field, right? So you should, yes, the electric field, it will easily pull out the electrons from the N-type semiconductor, and once the electron is free, it will start conducting. But in P-type semiconductor, do you think you have electrons? Electrons are not the majority carriers, they are the minority carriers, right? And these electrons, they'll start going in the direction opposite to the top of the electric field, but they will start jumping from hole to hole. It feels that as if the hole is, if electrons are going in this direction, we feel that the holes are going in the opposite direction. Hole is nothing but a vacancy for electron. That is all you need to remember. So obviously the current in the N-type will be greater than the current in the P-type. So this is the correct option. All right. Fine. So let us take the next question. Right. So the effective resistance of a parallel connection that consists of 4 wires of equal length, equal area of cross section and same material is 0.25 ohms. What will be the effective resistance if they are connected in series? All right. Now go through the question again. Please try to sketch a rough diagram. All right. So the effective resistance of a parallel connection that consists of 4 wires, right? They have equal length, equal cross section area and same material, right? So these are the 4 wires, let me say. All right. Then what you are finding is that the resistance about these 2 points, right? So it is actually 0.25 ohms. Okay, that is what they have given, right? Next, what will be the effective resistance if they are connected in series, right? Now the options are 1 ohm, 4 ohms, 0.25 ohms and 0.5 ohms, right? Okay, now let us see how to solve this. Now you have these 4 wires, right? Now treat them as some resistor, take a variable, right? So take a variable, call it as R and R, right? Now let me simplify this, right? Now I have 4 resistors, that is identical resistors in parallel, and I know the parallel combination value, that is the effective resistance value, right? So therefore I can say equivalent resistance is nothing but R divided by 4. Now in the question they have given it as 0.25 ohms, right? So therefore R will be equal to 1 ohms. Correct? Now come back to the question. They are saying what will be the effective resistance if they are connected in series, right? Now each of this wire has a resistance of 1 ohm, right? Now all of them are in series. So 1, 1, 1 and 1. What are we getting? 4 ohms, right? So therefore, the correct option is 4 ohms, right? So this was, don't you think this is the remembering question? No, definitely you need to understand, okay? This is understanding. So according to Bloom's taxonomy, so you need to understand the question, fine? Right, so understanding level, then you can clearly see that around 70% of students would have done this, right? So this was not that difficult, okay? But like I said, you need to read the question and put it in the form of a rough diagram. Otherwise, it is not that easy to get, right? Okay, fine. So let us go to the next one. All right. So a body is executing simple harmonic motion with frequency n, small n. The frequency of its potential energy is, okay. So this is quite easy, right? Now you have simple harmonic motion. You have studied pendulum. You would have studied about spring also, right? Spring mass system, right? Now what happens here? If I take, this is the rigid support. Now say this is the pendulum, okay. Of course, the oscillations must be small here, fine, okay. So say this is the reference level where I define potential energy to be 0, okay. How do you define time period? So what is frequency here? Number of oscillations per second, correct? So therefore, as per this question, I can say n is equal to 1 by t. Okay, n is equal to 1 by t, right? So from left extreme, mean right extreme, okay, then 1 and 2, come back. So if you go from left extreme, mean right extreme, mean left extreme, you will say that you have completed one full oscillation. Now the time taken for that oscillation is what we call it as time period, right? Now when you go from, see, this cycle has to be completed. So like this, then you have to come back like this, right? So this is what we call it as time period, right? This is what we call it as time period, right? So what happens to the potential energy, that is how potential energy varies with time, let us try to understand here. So I told you that potential energy is 0. Now when the body or the pendulum mass, when it comes to the left extreme or right extreme, so obviously the potential energy will be higher than 0. So let me call it as some u dash, okay. So u dash is not 0. So if you observe left extreme and right extreme, they are at the same level, right? So obviously u dash is same for left extreme and right extreme. Now I want you to look at this, okay. So see, when you go from 0 to max, then again max to 0, then 0 to max, max to 0, right? So if this is the time period of oscillation, okay, now this represents mean position right extreme, okay, then mean position left extreme, then mean position again, okay. Let us take it like that, fine. So just a rough sketch, all right. Now if you so this was the time, what do you say, frequency, right? And so frequency of oscillations actually, okay, oscillations. Now if I talk about frequency of potential energy, now what is happening? 0 to right extreme, right? So obviously you have the potential energy going, it will increase, right? So it will increase from 0 to right extreme, then again from right extreme to 0, it will again decrease, okay. So in this half cycle, right? So in this half cycle, you are getting the potential energy. See, you can never get, here I have defined 0 for potential energy here. So you will start getting the pulses like this. Variation of potential energy with respect to time. So obviously you can notice that it is 2 times the frequency of oscillation, correct? So therefore the correct option, right? Okay, so let me, all right. So the correct option, it would be 2n, right? So it would be 2n. So here it is actually understanding, right? So you can say around 60 to 70% of your friends or the takers of this exam would have answered this, all right? Okay, fine. So let us go to the next question. It says a small block slides down a smooth incline. Slides down a smooth inclined plane, fine. So A is not here, all right. Starting from rest at time t is equal to 0. Let Sn be the distance travelled by the block in the interval t is equal to n minus 1 to t is equal to n, okay, right. So then the ratio Sn divided by Sn plus 1 is, okay. So this is not there in your NCERT book, I mean at least the straight relation what I am going to use now, right. So you can derive this. So I think you would have studied this in your classes, right? So competitive examination classes, we'll call it as the distance traveled in the nth second. Okay, so what does it mean? Say a body is traveling, say this block, right? It is going down the incline for 10 seconds, let us say. Okay, so this t is equal to 9 to t is equal to, this is the total time taken for total journey, 10 seconds, let us assume. So the time interval between this 9 to 10 second, that is 9 second to 10 second, we'll call it as the nth second, the last second, that is, you can say, that is from 9 second to the 10 second. This interval we'll call it as the 10th second, all right? Okay, so in general, the formula that we use, right, for the displacement covered in nth second, so that is going to be something like this. I'm sure most of you know about this. So we'll start it like this. So u plus a times n minus half. Again, as soon as you see this a, you should understand that this is for a journey with constant acceleration. Okay, so this is easy now. Now this block slides down a smooth incline, right? So when you see an incline, so obviously the acceleration, right? So if you resolve the components, so you'll be getting it as g sine theta down the plane. So g is constant. If theta is constant, g sine theta will also be constant, right? So obviously the ratio that you need to get is Sn divided by Sn plus 1. Okay. Now I can use this relation straight away. Now they have said it starts from rest. So you will go to 0, right? So therefore I can write it as, all right, so I'll be using this relation here. Now I want you to look at the options. So the options are 2n plus 1 divided by 2n minus 1. Then you have 2n by 2n minus 1, then 2n minus 1 by 2n, and this is the last option. So after solving, let us come back to the difficulty level here. All right, so Sn, right? So that I'll write it as u plus a times n minus half. Correct? Now they're asking you the ratio of Sn with Sn plus 1. So all you have to do is replace n with n plus 1 here. Okay, so u plus a times n plus 1 minus half. I have replaced n with n plus 1, that's all. Okay, so u was 0, right? Now if you take ratio, a will get cancelled. Okay, so obviously Sn by Sn plus 1 is going to be, now you have 2n minus 1 divided by 2. Now here you have, yes, so plus 1 minus half is plus half, correct? So you'll be having 2n plus 1 divided by 2. So obviously the answer is 2n minus 1 divided by 2n plus 1. So this is the correct option. Okay, so this was also, I don't say it was difficult and all. So this was actually easy, right? Moderate, you can say, right? So you can say that around 70% of your students would have answered this, right? Now again, this is not straight out of NCERT, but the concept is given there, all right? The concept is given, fine? Okay, right. At least in the exercise problems, additional exercises you'll see, right? So there, I'm sure that a problem which describes this is present, all right? Okay, fine. So next, let us look at this question. A spring is stretched by 5 centimeter by a force of 10 Newton, okay? The time period of the oscillations when a mass of 2 kg is suspended by it is, okay? So we need to find the time period, right? So these are the options, all right? Now let us solve it. Now I have this spring, you are stretching it by 5 centimeters they have given, okay? So the displacement from its natural length is 5 centimeter, okay? Now what is the force that you are using? Force that you are using is 10 newtons, okay? Now I can calculate the spring constant here, right? Only then I can find the time period, right? So they are asking you the, time period only, fine? Okay, so F, right? Spring force, the magnitude, it is given by K into X, right? So K into X, right? Or applied force, you can say, right? When the spring is in equilibrium, the applied force and the spring force are equal in magnitude and opposite in direction, okay? So let me take it as 10. So 10 is equal to K into 0.05. So this will come around, K, I can take it as 200 Newton per meter, correct? So 200 Newton per meter is the spring constant of this, force constant of the spring, I can say, right? Now next, let me try to get the time period. Now for that, I need to get omega, right? So omega, I can write it as square root of K divided by M. Now I got it as 200, right? So, and the mass they have given it as 2 kg. So obviously this will be square root of 100. So omega will be 10 radians per second, right? So 10 radians per second. So naturally, so I can say 2 pi by t is equal to 10. So t is equal to 2 pi by 10, so which is 6.28 divided by 10, which is 0.628 seconds, right? 0.628 seconds. So this is the correct answer. Option B is the correct answer. All right. Okay. So this was, you can say, not remembering definitely. Even this was a moderate question, right? So the second level, I'll take it, right? Then around 50 to 60% of your friends would have answered this, right? So all you have to do is substitute in this form, get force constant, put it there and get the time period. Simple, right? Okay. So let us go to the next question. So it says, if force F, acceleration A and time t are chosen as the fundamental physical quantities, find the dimensions of energy. I'm sure you would have learned some questions which are like this, right? Now, what do we mean by fundamental units? So these are the units which are not dependent on any other unit, right? So for example, if you take force, acceleration and time, that is, they are not fundamental. If you consider them as fundamental units, fundamental physical quantities, sorry, right? So you can see that, you can say that force is not dependent on acceleration, force is not dependent on time or any other thing which is present in that fundamental set, okay? So let us try to find the dimensions of energy now. So the options are given like this. So I'll say this is not remembering or anything. When this is a moderate question, okay, this is a moderate question and you have to solve to get the answer, right? Now what I'll do is energy, right? So I'll write like this. So dimensions of energy, correct? So that must be equal to, you can say F, right? So F power, let me take it as A, then A power B, then time they have given, right? So T power C, like this, okay? I'll be using dimensional analysis here, that is all, right? So this is from your first chapter, units and dimensions, okay? Now what I'll do is, let me substitute for the dimensions of energy, right? Now I have, I'll take it to the next slide. So now I have M, L power 2, T power minus 2, that will be equal to, now F, let me substitute the dimensions of F, right? Now I have M, L, T power minus 2 to the power A, right? Now next I have acceleration, right? So meter per second square, L T power minus 2 to the power B. Then finally I have time itself, right? So time itself, I'll take it as C. Okay, I'll take it as C. This is small c. Fine. Okay. Now all you have to do is compare the LHS and RHS, right? Now I have 1, correct? So 1 is equal to A, right? So m power 1 is equal to m power A only, right? I don't have anything else. Now next 2, right? So m is done. Now let us go for length, right? So 2 is equal to, I can say A, then B, that's all. So I can say A plus B, right? Then I have minus 2 is equal to, you can say minus 2A, then minus 2B plus C. Minus 2A, minus 2B plus C, okay? So A is 1, I got it, right? So B is also 1, I got it here, right? So if A is 1 and B is 1, I can say minus 2 is equal to minus 2 plus C, right? So C will obviously be 2, okay? C will obviously be 2, right? Now obviously the from the options, right? So you can clearly see that A is 1, B is 1 and C is 2. So A is 1, right? So B is 1, so and C is 2. So this is the correct answer. So let me write that, right? So this is the correct answer. Option D is the correct answer, right? I think around say close to 60% of the students would have got this, fine? Okay, all right. So let us go to the next question. Water falls from a height of 60 meter at the rate of 15 kg per second, all right? In each second, 15 kg of water it is falling down. To operate a turbine, the losses due to frictional force are 10% of the input energy, okay? So how much power is generated by the turbine, all right? Now what is happening? So this is a typical example of a hydroelectric power station, right? So this is how they operate, right? Okay, so water is falling from this height. So yeah, so height they haven't given. So let me take it as h, which is 60 meters, right? So water is falling like this, fine. You know, water which is at a height, it will have gravitational potential energy, right? So that gravitational potential energy, when it is, you can say it will convert to kinetic energy when it reaches at the bottom, right? So this kinetic energy is the one which will rotate the turbine. However, you have to consider 10% of the input energy is lost due to the frictional losses, viscous force. When we speak about friction, you can take viscous force here, right? Okay, nice. Now, they have given the amount of mass that is falling per second, right? So, in 1 second, 15 kg is falling, right? Now, whatever the potential energy the water has, right, it will be converted to kinetic energy. And finally, here they did not ask you to consider any losses apart from this 10%, okay, fine. So, all this potential energy will convert to kinetic energy, no doubt in that. So, in that kinetic energy is again converted to electrical energy using your turbines, you know that, fine. So, they are asking us the power, right? Power is generated by the turbine. So, the options are like this, all right, fine. So, let us try to get the answer here. Now, power, right? So, that is work done per unit time. Otherwise, you can say energy transferred per unit time, right? Now, every second 15 kg is 15 kg is falling down, right? So, in 1 second, we have it already, okay? So, I can say mgh, right? So, mgh divided by T, right? Now, you have this m by T already, right? Or you can say 1 second, right? 1 second. So, this T will become 1. So, P you have it as m, which is 15 per second. So, that has T is already 1 here, okay. So, you should take 10, okay. Then I have H, which is 60, okay. So, 15 into 10 into 60. I have not yet considered the frictional losses, right. Now, here I am calculating as if there are no losses, right, fine. So, let us solve it here. Now, P I am getting it as 15 into 60, right. So, 9, yes, 9,000 watts I am getting, otherwise 9 kilowatts I am getting, right. Now, you need to consider this. If you consider 10% of the input energy is lost, now how much is remaining? 90% is remaining, right. So, obviously 90%, right, 90% of the input energy, which is 9 kilowatts, it will be 0.9 into 9 kilowatt, so which will be 8.1 kilowatts, okay. So, this is the answer, right. Now, do you think this was easy? No, this is not completely remembering, right. So, you can take it as a moderate level question, right. I don't see the difficulty here. It's not that difficult, okay. So, it was moderately difficult, moderate you can say, right. So, you can say about 50 to 60% of the students would have got the answer to this, okay. Nice, very nice. So, let us take the next question. So, it says the velocity of a small ball of mass m and density d, okay. So, then you should read this question carefully, right. When I read it for the first time, even I got confused, okay. So, they have given mass of the ball and the density of ball is also d only, right. Okay. When dropped in a container filled with glycerin becomes constant after some time, okay. So, if the density of the glycerin is d by 2, right, then the viscous force acting on the ball will be this concept, right, you will learn in your NCERT. However, you just need to use it, you just need to do two or more steps, two more steps, that is all, okay. So, if you do 2 more steps, you will be getting the correct answer. It's very simple, okay. So, I can say it is straight out of, this question was not there in your NCERT, but the concept is definitely there in your NCERT. This is from your fluid dynamics, where you will study how to calculate this terminal velocity and everything, fine, okay. Stokes law basically, all right, fine. So, here let me solve for this, all right. Now, say this is the liquid column, right. So, they have said glycerin, right. So, here is the, they have said ball, okay. So, its mass is m, they have given density is d. Now, density of the liquid, right, which is rho, we usually take, let me take it as d by 2, okay. Now, density and mass, they have given it as capital M and T, right. Now, once it reaches, that is if the velocity, right, okay, velocity becomes constant after some time. What does that tell you? Velocity is constant means the net force acting on it must be 0. So, obviously, if you draw the free body diagram, right, so you will get something like this. So, gravitational force is acting, no doubt in that. Now, you have buoyant force, you must not forget that, okay, and you have this viscous force, right. So, the question is viscous force only, right. The viscous force you need to find out, okay. So, obviously, since it is moving with constant velocity, acceleration is going to be 0. So, I can say Fv, viscous force, plus the buoyant force, that will be equal to the gravitational force, which is mg, right? So Fv, I'll write it as mg minus buoyant force. Now how do we give buoyant force? Buoyant force is given as rho Vg. So what is V here? V is the volume of the body which is completely submerged in the liquid. What is happening here? The complete body, the complete ball is in the glycerin, right? Okay. So therefore, I can say the complete volume of the ball is in the glycerin now, the liquid now. So they have given rho as d by 2. See, this rho is the density of the liquid I have taken, right? So obviously this will be d by 2 Vg. Now what is dV? Density of the, so d is also the density of the ball, right? So d into V, I am getting it as m, correct? So because density is equal to m by V as per this question, okay? So finally I am getting the answer as mg minus mg by 2, so which is mg by 2, correct? So half of the gravitational force. So the correct answer is option C, okay? So this was not remembering, definitely not applying, not completely difficult. It was a moderate question, okay? So you can say about 60 to 70% of your friends have answered this question, okay? Nice. All right. Fine. So I think I wrote it in this slide only. All right. So let me erase this. Fine. A lens of large focal length and large aperture is best suited as an objective of an astronomical telescope since, okay? Now I think you know this, right? So you have different types of telescopes, right? And if you are quite curious, you would have learnt about this Hubble telescope, so which is there in the space for the last you can say roughly about 20 to 30 years. So it was launched by NASA. It has sent beautiful pictures of our universe, galaxies and much more, right, to our Earth, right? So it's very big, right? And there they will use the reflector type telescope there, okay? But here they are talking about an astronomical telescope. So they're asking you to consider a lens, right? So here basically we'll consider large objective. Why is that? Because why is that? These are the options, right? Now if you look at the options, I think you should remember something. So a large aperture contributes to the quality and visibility of the images. Yes, definitely true. So if you buy, even you can notice this, if you buy some cheap binoculars, right? So whenever you go for some festival or some nearby town festival or something, right? So there, if you are focusing farther objects, you will see some color aberrations. Will see, right? So at the edges, right? At the edges of the images, you will be seeing some green, yellow, red and yellow, some colors. The image is, it's not as if you are seeing it with your own eyes, right? So there will be some color aberrations, right? So if you choose lenses, small lenses, right? So a large aperture contributes to the quality and visibility of the images. This is definitely true. Then a large area of objective ensures better light gathering power, okay? So that is why if you consider any even you can buy it in Amazon these days, right? Telescopes, normal telescopes, right? So it will cost around 20 to 30,000, right? So you will be able to see Saturn rings and everything. But the telescopes which are developed to observe space, there you will get much better pictures. So the pictures of Saturns and everything, whatever you can find in your internet, right? So they are captured using one of the telescopes which are having large objectives, okay? So fine. Then a large aperture provides a better resolution. This is also true, right? So the correct option is all of the above. A, B and C all are right, okay? So this is basically remembering, okay? So this was easy actually. You can say around 50% of the students who have taken the exam would have answered this question, okay? Right. So let us take the next one. For a plane electromagnetic wave propagating in x direction, which one of the following combination gives the correct possible directions for electric field and magnetic field respectively? So options are given, all right? And how to solve this, let us try to understand. Now you know that if you cross, right? So if your electric that is electromagnetic wave is propagating in x direction, so obviously your electric field and magnetic field, they must be oscillating in y and z directions, correct? So if electric field is oscillating in y direction, right, then yeah, so when you learn it for the first time, at least we'll learn it like this, right? So say this is x-axis, this is y-axis, and this is z-axis. So let me take it like this. No, this is not right. So this is x. This is x and this is y. Okay. So x cross y, you should get it as z, right? So electric field vibrations are like this. Okay. Then magnetic field vibrations are like this. Okay. So then you can see that the electric field is varying about you can say, okay, this diagram did not come proper. All right. Anyways, but here you don't have the x and y, that is, you don't have the electric field and magnetic field vectors in any one of the axes. You can see that they have y and z components, right? So they have y and z components. All right. So now what you have to do is you need to solve, right? So you need to cross them such that the answer that you will be getting, right? So the resultant that you will be getting, its direction will be in the x-axis. Okay. So how to do that? You know that E cross B, right? So if I cross it, right? So yeah. So first is E, right? So if you cross E, right, electric field vector and magnetic field vector, it will give you the direction of the electromagnetic wave. Simple. Okay. Now what you have to do is, see, you have the electric field vector value and magnetic field vector value given in all the 4 options, right? Okay. So you have to cross E and B. That's all. Okay. Let us get E cross B, right? We'll see what is the unit vector that we are getting at the end. So we have to check whether it is in x direction or not. That is all. Okay. You have to work option by option, right? Now how do we do this? So I, then I cap, j cap and k cap. Now substitute. So first vector, this is E, right? So I have 0, then 1 and 1. Now I have 0, then -1 and -1, right? Let me check. Okay, let me check. Now I have I cap. Now I have 1 into -1. So -1, then minus of -1 again. So this will become +1, right? So -1 plus 1. I'm not getting I cap. I need to get I cap, right? Because they are saying the wave is propagating in x direction, right? So we have to work like this. Now all you have to do is carry a pencil and an eraser, right? Okay, then you can erase these values, right? If you erase these values, you can substitute for the remaining options here, correct? Now let me erase these values here. Now finally, if you work option by option, you will see that after that is if you consider the last option, right? Okay, minus j plus k cap, then minus j minus k cap, right? So you have -1 plus 1, then you have -1 and -1, okay? -1 and -1, right? Now here I have I cap, then 1, then minus of -1 I'm getting. So this is obviously I'm getting it as 2 I cap. See, I cap, I got it right. E cross B, right? So in whatever that is whatever the result you get it for E cross B, right? So that will be the direction of the electromagnetic wave. Fine. So obviously the 4th option satisfies. Okay, it will give us the unit vector in x direction. I cap I'm getting. Okay, so this was moderate. Okay, so say around 60 to 70% of your friends have answered this question. All right. So let us go to the next one. It says the half-life of a radioactive nuclide, right, is 100 hours. The fraction of the original activity that will remain after 150 hours would be, okay. Half-life they have given. What is that? 100 hours. So this was an easy question actually, right? Yeah. Let us look at the options now. It's 2 by 3, then 2 by 3 root 2, then 1 by 2, and 1 by 2 root 2. Okay. Right. So let me. Yes, so T half-life they have given it as 100 hours. So T it is 100 hours. Okay. Now they're asking us the activity after 150 hours, right? So you know we'll use this relation, right? So A is equal to A naught divided by 2 to the power T divided by T, capital T is which is half-life in this case, right? So what is 150? So if you can simply write that T is 100, you substitute 150, you are going to get it as 3 by 2, 2 power 3 by 2. So obviously, yeah, so A is equal to A naught times, so you have 2 power 3 by 2. So 2 cube whole square, square root of 2 cube you can say, correct? So 2 power 3 whole power 1 by 2, right? So this is obviously it is going to be 1 divided by 2 root 2. So option D is your correct option. So this was also a moderate question. Okay. So Yes, so you have questions like this, at least not, activity is also there, and they'll ask you to calculate the number of, that is the concentrate, that is number of nucleons present, right, the number of nuclei that is present after a certain time, right. So you know that activity is directly proportional to the number of nuclei, number of nucleons present, correct? No, the number of nuclei present, right. So therefore, you can say this is, you can say it is from your NCERT, but not straight away, right? Okay, so about 50 to 60% of your students would have answered this question, right? So this was moderate, right? Not easy, not definitely easy. It was moderate. It was a moderate question, you can say, correct? Let us go to the next one. All right, a thick current carrying cable of radius r carries current uniformly distributed across its cross section. The variation of magnetic field B of r, see B as a function of r they have given, okay, due to the cable with the distance r from the axis, right, axis of the cable is represented by this options. So you have 4 options, you need to look at them carefully, right. Now usually if you consider magnet, that is a thick current carrying conductor, right. So here this is straight out of your magnetism chapter, right. So moving charges and magnetism, there you will study something called as Ampere's circuital law, correct, Ampere's circuital law. Now we'll assume the current which is flowing in this is uniform, that is current density, you can say, call it as uniform, no problem, right. Now if you consider Ampere's loop right there, okay, you will see that this current, right, so we will start getting ratio something, we'll start getting once you derive this, right. So finally inside, right, so inside you will see that B will be proportional to R, okay, inside B will be proportional to R, and once you are outside, that is this is valid up to the surface, right. Now once you are outside, right, so this is for R less than the radius, right. Now B will be inversely proportional to R for R greater than this R, you can say less than or equal to in this case, right. So the appropriate graph, as you can notice, it will be option A, that is when you go from center up to the surface, you will see that the magnetic field increases. Now once you are outside and once you are going away, the magnetic field decreases as per this relation. So this tells you that B is proportional to R, right, and this graph tells you that B is inversely proportional to R, that is once you cross a distance R, capital R, let me say, okay. So option A is the correct option, fine. Okay, let us go to the next one. Yes, so in terms of this was present, this was actually an example problem, right. So this, I think this was an example problem or additional exercises, probably you had this, right, because we use Ampere's circuital law to understand this, right. Okay, so and I suggest you guys to solve this exemplar problems also, right. So they are also equally important, right. So at least not the straight question, but you had it in your NCERT, at least the applications of Ampere's circuital law, all right. Then it was not easy not to remember, okay. Then you can say that about 60% of your friends would have solved this, right. So this was an easy question, moderate question, you can say easy to moderate, right. Let us go to the next one. Yes, a particle is released from height s from the surface of the earth. At a certain height, its kinetic energy is 3 times its potential energy. The height from the surface of the earth and the speed of the particle at that instant, right, are respectively. So these are the options, right, and we need to sketch a rough diagram here, right. So as soon as you look at the question, I know many of you would have left this question, not many, okay. So say at least 40% of your friends would have left this question, okay, nice. So all you have to do is sketch a rough diagram, and we are going to use a law of conservation of, that is conservation of energy, that is all, okay. Even if you don't know conservation of energy, you can say this can be solved if you have necessary knowledge about this motion of a body with uniform acceleration, because it is falling, right, uniform acceleration, okay. So let us sketch a rough diagram here. So say this is the reference level, earth level, let us say, okay. Now this is the building whose height is s, let me say, okay. Now the body, particle, it is released from here. Now you are free to assume mass as m or anything, that is up to you, okay, because all they are asking is the ratio, fine, okay. So the speed, right, and the height they are asking, all right. Now what are they saying? At a certain height, its kinetic energy is 3 times its potential energy, right. Now when it is falling, what do you know, at the top of the building, right? So all the energy, it is in the form of potential energy, right? It is in the potential energy. So kinetic energy is 0. Yeah, the moment you drop it, right? So you know, the moment you drop it, we'll take the initial velocity as 0, right? So potential energy is there, kinetic energy is 0, right? Now once it is moving downwards, its potential energy decreases, right? And the kinetic energy increases by the same amount, right? So this is important, all right? Now say it is at this level. Now what they have given? They have given that the the kinetic energy is 3 times the potential energy. Now treat this Ke, Pe as variables, no problem, okay? Now let me call this height as h. At this height h, you can say kinetic energy is 3 times the potential energy, okay? Nice. Now let me calculate potential energy here, right? So potential energy, I know that it is actually mgh, correct? That is at this level, I know that the potential energy is mgh, right? So say at point 1, okay, at this location 1, right? So I'll say potential energy is mgh. I'm talking about at this point, okay? Now what is the kinetic energy? You can do this, it's not that difficult, right? Now what is the distance that this mass has traveled? Obviously s minus h, right? Okay, now all you have to do is get half mv square, correct? So half m then v square, correct? Yes, so what is v? So v was, yeah, I can calculate, right, using equations of motion. V square is equal to u square plus 2as, right? So u is 0, initial velocity, right? Now I can say 2g into s minus h, right? So 2g into s minus h. So this is what I can write. Now 2 and 2 will get cancelled, fine. Now I have, yes, 2 and 2 will get cancelled. Now I have potential energy as mgh. Okay, potential energy I have it as mgh. Then kinetic energy, I got it as mg into s minus h. See, this is easy. Now you can take the ratio, right? So let me take the ratio here, just a second. Yes, so let me take, if I take the ratio, so I can erase this, so it will be easy for me to calculate. All right, so let me take the ratio. So kinetic energy divided by potential energy by kinetic energy, I'm going to get it as h divided by s minus h, right? So obviously this ratio they gave it as 1:3, right? Kinetic energy is, sorry, what was that? Kinetic energy is 3 times its potential energy, right? Kinetic energy is 3 times its potential energy. So Ke I can write it as 3 Pe, or I can say Pe divided by, yeah, so Pe divided by Ke is equal to 1/3, right? Yeah, so Ke, yeah, so Pe divided by Ke is 1/3. This is right. Okay, now I can say 3h is equal to s, right? So 3h is equal to s minus h. So obviously I'm getting h as s by 4, correct? H I got it as s by 4. Now the answer that I have for, yeah, s by 4, it is there in 3 options, okay? Now I need to get the velocity now, right? Yeah, now this is easy. How do you get velocity? V is equal to square root of 2gh, right? Now all I have to do is V, let me calculate it here. So I'm getting it as square root of 2gh. Now what was h here? So it was actually s by 4, or let me look at the options first. Yes, so V is equal to 2g, no, not h, this was actually, okay, so this was the speed I need to calculate, correct? Fine, so 2g into, so this was actually s minus h, right? Okay, so s minus h. Now I have, yes, I'll get s minus s by 4. So that will be 3s by 4, correct? So let me write it directly here. Now I have to write s minus h, which is 3s divided by 4. Yes, then we will get the answer. 3s divided by 4. Yes, 3s divided by 4. So that is your answer. So 2, yeah, this is whole root, sorry. So 2 and 2. So obviously you're going to get it as square root of 3gs divided by 2. Yeah, this is whole root. So option D is the correct option. Okay, fine. So let us go to the next problem. right? So this is from your moving charges and magnetism, right? First, yes, let us go through the question. An infinitely long straight conductor carries a current of 5 ampere as shown. So this is an infinitely long conductor, okay? An electron is moving with a speed of 10 power 5 meter per second parallel to the conductor. The perpendicular distance between the electron and the conductor is 20 centimeter at an instant, okay? So calculate the magnitude of the force experienced by the electron at that instance, okay? Right, so this is the electron, right? And it is going at a speed of 10 power 5 meter per second, all right? So 10 power 5 meter per second, sorry. All right, now they want you to find what is the force it experiences, right? Now the force, you know, the magnitude they're asking, right? Okay, fine. So options, all right. So there you just have the options there. Okay, so let me display the options first. All right, so the options are like this. Okay, now let me try to get the force. Now you know that force is given as Q times V cross B, okay? But here you need to understand that this current, right, this current, it gives rise to a magnetic field which is given, sorry, it is like this, correct? So you have the magnetic field which is this way. So B is like this, V is like this. So V and B are perpendicular. So Q times V cross B, right? So I'm going to write it as QVB sine 90 I'm going to get, correct? So sine 90 you know it as 1, okay? So here we are talking about electron, right? So 1.6 into 10 power minus 19. Then velocity they gave it as 10 power 5. Now I need to get magnetic field at this distance of 20 centimeter, right? Okay, now this would be given as mu naught I divided by 2 pi r, correct? So we know that. So mu naught I divided by 2 pi r, right? Okay, now let me simplify this. Now you are getting 1.6 into 10 power minus 14, okay, minus 14. Now you have 4 pi into 10 power minus 7, okay. Now I is 5, so this is 2 pi. Now the distance, right, that is actually r here. So you have 20 centimeters, which is 0.2 meters, okay. So pi and pi, so you have 2 here, okay. Then 0.2 again you will get it as 10 times, okay. So 16, this is 8, okay. So finally you are going to get it as 8, then 10 power minus, all right. So all right, the final answer you are supposed to get it as 8 into 10 power minus 20 Newton. After you simplify, this is the answer you are going to get, all right. So the correct option is option D, right. So here it was, yeah, you can call it as NCERT concept, right. So this was not that difficult, okay. All here they have clubbed two concepts, right, that you are studying in your NCERT book. What is that? You need to find out magnetic field first due to this infinite wire, right. Then you need to use the magnetic force on a charged particle when it is moving in a magnetic field, that is all, okay. So this was not completely easy, you can say, not remembering, right. So you can say about at least 70% would have answered this question, right. So this was moderate question, easy to moderate, I can say, correct. All right, so you have studied this. I mean, for answering this question, the NCERT knowledge is sufficient, okay. If you have studied your NCERT textbook, you would have answered this question, right. Okay, next, let us see this question. So two charged spherical conductors of radius r1 and r2 are connected by a wire. Then the ratio of the surface charge densities of the spheres. So this is the options, right. Now this is a standard result, okay. So you have two spheres, right. They are charged, obviously. Then you are connecting them with a wire, okay. Now say you have r1 here and this is r2, correct, right. So you will clearly see that since you are connecting the wire, right, so the potential difference between them will become 0. Charge sharing will take place, okay. Charge sharing will take place such that the potential difference between them will become 0. Then finally you will get to know that the surface charge density, it will be inversely proportional to the radius of the charged conductor, okay. So obviously sigma 1 divided by sigma 2, it will be r2 by r1, okay, r2 divided by r1, right. So you might have even studied about a isolated conductor of irregular shape, right, isolated conductor of irregular shape. Usually we will call it as radius of curvature, okay. Now say charges are distributed here. What you will see is that the surface charge density will be more where you have less radius of curvature as per this relation, okay. So at this tip, right, you will be having more charges compared to here, okay. So here the charges are distributed farther enough, okay, they are distributed farther. So, charge per unit area, it is going to be less in these regions compared to the region where you have less radius of curvature. Simple, okay. Same concept applies here also, right? Wire is there, same potential. Conductor, the potential is same throughout, correct? Or you can say, yeah, so potential is same throughout the volume, right? Same, that is inside a conductor, the potential is same as that of the surface. You would have studied that, okay? So, this was an understanding question, okay? I am sure that at least 70% of the students would have solved this, okay? Right, and this is not straight out of your NCERT, correct? You have to work to get this, you have to work to get this answer, right? If you don't know the standard result. All right, okay, let us go to the next question here. Find the value of the angle of emergence from the prism. Okay, here the refractive index of the glass is given as √3. All right, so these are the options. Fine, you need to find the angle of emergence. That is the final ray that is coming out of the prism, all right? And the reference what we take is the normal drawn at that particular interface, correct? So, here it was, say this was an easy question, okay. So, if you have covered your concepts from NCERT, you would have solved this question. So, after getting the answer, you will see that nearly 70 to 80% of your friends who have taken the exam would have answered this question as per my guess, okay. Nice. So, let us try to solve this question here. Now, as you can see, the angle of incidence here, it is 90 degrees. So, obviously, the angle of refraction will also be the same 90 degrees, correct? Angle of refraction will also be 90 degrees, all right? Now, at this interface, right, you have to draw a normal, okay? So, the angle of incidence in this case, right, now 60, this angle will be 60, so obviously, this angle will be 30 degrees, right? This angle is 30 degrees. Then, at this interface, if you observe, light is leaving, that is, it is leaving the denser medium and it is entering the rarer medium. It is going from glass to air, correct? Glass to air, right? So, you need to focus on this concept. Everybody knows that if angle of incidence is 90, angle of refraction will also be 90, right? That is done, okay? So, next let us focus on this part. Now, say the light after, and this is the emergent ray. So, final ray which is coming out of the prism, okay? Nice. Okay. So, let us try to solve here. Now, I want you to consider this ray which is going from glass to air, okay. So, at the second interface, this interface, I can use Snell's law. So, mu1 sin I, okay, is equal to mu2 sin R, right. So, I is the angle of incidence in the denser medium. What is that? 30 degrees, okay. So, I will say mu, that is √3, right, they have given. Mu1 is √3. Now, this will be my mu2. So, glass to air, right. So, glass to air. So, √3, then sin 30, correct, that will be equal to mu2 air, which is 1, then I have sin R. So, this angle is what I am calling it as R here, okay, nice. So, next let me solve this. Now, you have √3 divided by into 1 by 2, okay, that will be equal to sin R. So, obviously, √3 by 2 is equal to sin R. So, obviously, R will be 60 degrees. So, the emergent ray, it makes an angle of 60 degrees with the normal. So, obviously, the answer is 60 degrees, okay. And again, this was an easy question, I can say easy to moderate, fine. You have to solve to get the answer and you need to apply Snell's law properly, okay, fine. So, let us go to the next question, all right. So, this is again from your optics, interesting question, okay. And a point object is placed at a distance of 60 centimeter from a convex lens of focal length 30 centimeter. If the plane mirror were put perpendicular to the principal axis of the lens and at a distance of 40 centimeter from it, okay, the final image would be formed at a distance of, okay. Let us try to solve this. Now, the options are like this: 30 centimeter from the plane mirror, it would be a virtual image, okay. So, you need to guess the image distance, that is the final image distance and its nature also, right. Then 20 centimeter from the plane mirror, it would be a virtual image again. Then 20 centimeter real image, sorry, this is from the lens, right, okay. Now, option C and D, if you observe, it is the references from the lens, okay. Now, option D, it says 30 centimeter from the lens, it would be a real image, okay. So, let us try to solve this, okay. So, this is not easy, okay. So, you can put it as a moderate to difficult question, say about only 70, or you can say about 50%, right? So 50% of them would attempt this. Okay, so we can 50% would have attempted this question as per my guess. Okay, right. So let us try to solve this. Right, it needs time. Okay, and you need to put a rough sketch, otherwise it is not that easy to solve. All right, so this is that lens, convex lens, which they have given. Now they have placed the object at 60 centimeter, right? So object is at 60 centimeter. All right. Now the focal length, they have given it as 30 centimeter, right? So focal length, let me write it as 30 centimeter. Now where is the mirror placed? The mirror is placed at a distance of 40 centimeter, correct? So here is your mirror. Okay, here is the mirror which is at a distance of 40 centimeter. All right. Now let me get the image formed by the convex lens first. Okay, what we need here? We need the lens formula, 1 by V minus 1 by U is equal to 1 by F. And keep in mind, you have to keep the sign conventions in mind, right? Okay, now I need 1 by V. Now I am going in this direction, right? So I have 1 divided by minus 60. So finally this term will become 1 by 60. Okay, so that will be equal to, now focal length, right? I am going towards right because focal length of convex lens is this way, right? So I am going to write it as 30. Focal length is 30. Don't write 40 here. Okay, so finally I am going to get 1 by V. Okay, so 1 by V, it is going to be 1 divided by 30 minus 1 divided by 60. 1 by 30 minus 1 by 60, which is again 1 by 60, right? So V, I am getting it as 60 centimeter, right? Now understand like this, if you didn't have that mirror there, right? So the image, right? So the image, it would have formed here at a distance of 60 centimeter, correct? At a distance of 60 centimeter from the lens, right? So because image distance is determined, that is measured from the pole, right? Correct? Fine. Now here 60 centimeter is the image distance, right? Now you keep the mirror there. Okay, if you keep the mirror there, so what type of rays are incident on the mirror? As you can observe, the rays that are incident on the mirror, they are actually converging rays, right? So wherever this, obviously you know that it will get reflected and the final image will be formed here, correct? The final image will be, actually this has to come little bit symmetrical. Okay, fine. All right. Now wherever the real, that is rays, right, actual rays, they are incident rays, they are appearing to be intersecting, right? You know they'll get reflected, but if the mirror was not there, they would have met here, right? Here is where you would have got the final image, that is image formed by the lens, correct? So this object, this is actually the virtual object. Now if you consider for the plane mirror, that is actually the virtual object, correct? Now this is 60, okay? Now they have given this distance as 40. So obviously from the mirror to the virtual object, this has to be 20 centimeters, right? Okay, so for a plane mirror, we know that image distance is equal to the object distance. So obviously this distance, right? Okay, so let me draw the diagram again. So this is the lens. Okay, now this is the mirror. Now this is your virtual object, right? Now this is your final image I. Okay, so this distance is 40 and this distance is 60, right? Obviously 60 and 40. So this distance I must get it as 20. So from the mirror it is 20 and from the lens also it is 20, if you observe, right? So from the lens to here it is 20 and from that final image to the mirror it is again 20 centimeters only, right? Now let us look at the options, fine? Okay, 30 centimeter from the plane mirror. No, 20 centimeter from the plane mirror, it would be a virtual image, right? Did we get a virtual image? No, because if the if the reflected rays are meeting, reflected rays, they are intersecting, right? Okay, they are actually intersecting. So there you will get a real image, fine? We will be getting a real image. So obviously the answer is going to be 20 centimeter from the lens, you would get a real image. Okay, option C is the correct option, fine? So you need to put the diagram on the paper, right? Whatever we did here, right? So you have to do this much, then this distances you need to measure and analyze, only then you will be able to solve this. Okay, so obviously the question, right? So you can take it as applicative based, right? So you can say about 50%, right? So close to 50%, like I said, they would have attempted this question. Okay, nice. So let us look at the next question, right? So this is simple. An inductor of inductance, capacitor of capacitance, resistor is given as R, right? Connected to an AC source of potential difference V, okay, as shown in the figure, right? Now the potential difference across L, C and R, that is inductance, capacitance and resistance. They have given it as 40, 10 and 40 again, right, respectively. The amplitude of the current, amplitude means what? It is the maximum current, right? So peak current will say, that is 10 root 2 amperes. The impedance of the circuit, you know this is easy. Now the options are like this. So this is, you can say, if you have covered your NCERT only, you are able to answer this question, because you know that impedance is given as Vrms divided by Irms, correct? Now they have given the voltage across the circuit elements, right? Now you know that Vrms, right? So it is RMS voltage, this V only, right? So if you sorry, if you express it in terms of these values, you know that these values you should not add them like you do it in your DC circuit, right? You need to consider their phases and everything, right? We'll be using phases if you remember, right? So Vrms, we'll write it as VR square plus VL minus VC, the difference only, right? Squared, okay, that is all. Now whole thing divided by IRMS, right? Now remember, in the question they have given you the peak current 10 root 2, right? Now you know IRMS is equal to I naught, that is the peak current divided by root 2. So obviously in the denominator we are going to get 10. Now what do I have here? So I have VR, which is 40, so 40 square plus, now this difference, right, it is going to be 30 whole square, right? So 40 square plus 30 square whole root, I am going to get it as 50 divided by 10. So I am going to get it as 5 ohms, right? 5 ohms is the correct answer. This was an easy to moderate question, you can say, right? If you have studied, if you have understood the concept, okay? So about 70% of your friends who have taken the exam would have answered this, right? 70% of the participants you can take, so you can say, right? They would have answered this question. Okay, nice. Let us take the next question. So you had match the following type questions also, right? It was interesting, right? So column 1 gives certain physical terms associated with flow of current through a metallic conductor. Column 2 gives some mathematical relations involving electrical quantities. You have to match, right? So drift velocity is given, electrical resistivity, relaxation period and current density, right? Okay, so you need to match, right? So drift velocity, what is the expression we have? E tau divided by m. Do we have it here? Yes. So A matches with R. Okay, A matches with R. Then electrical resistivity, you need to focus properly. Okay, so electrical resistivity, we have a standard relation, right? Nevertheless, let us go to the next one, relaxation period and current density, right? Now here we have an expression which all of us are familiar about. What is that? I is equal to NEAVD. Now if I take I by A, right? If I take I by A, I am going to get current density, right? So again, I by A is NEVD, right? So current density, it matches with this. So these are the options that you have, correct? So A and R, then D and Q. I'll explain all this one, okay? I'll try to simplify all of them, right? So A, R and D, Q. See, you have it in two options, A, R and D, Q, right? So B and D you can rule out, all right? Now let us focus on electrical resistivity, okay? Now this option is right, it has to look familiar to you, right? Now what do we have there? So we will write J is equal to sigma E. What was that? Equivalent form of Ohm's law, correct? Now if I get E divided by J, right? So E by J, I am going to get it as 1 divided by sigma, which is, I can say, rho, right? So what is rho now? So rho is actually, okay, let me write it in the next slide, it will be easy for me and for you also, it will be easy to read, right? Fine, so I have J is equal to sigma E, correct? So E divided by J, I have 1 by sigma, which is rho. See, there you got your third one, correct? So A goes with R, then D, it goes with Q, right? Very good. Then electrical resistivity, right? It goes with S. Now I have AR, then DQ, then I have B with S. Okay, so B with S. So option C is the correct option. Now let us verify this CP also. Okay, so C and P, right? Now if you look at this option, right, it doesn't look that familiar. I mean, there is something missing there, right? So you have M divided by NE square rho. Okay, M divided by NE square rho, right? What is rho there? Rho is actually the density, resistivity, we'll say. Correct? Rho is the resistivity, right? Now all you have to do is M divided by NE square rho. Now we know that rho is M divided by NE square tau actually. Okay, so M divided by NE square tau. They have given this option just to confuse you. Okay, so see, M and M will get cancelled, NE square and NE square will get cancelled. Obviously, you will be left with only tau. What is tau? Tau is the relaxation time, relaxation period, we'll say. So therefore, this will also match. Okay, this will also match, right? So the correct option is option C. Now, okay, so let me erase this. Let us look at the last question. Fine. A dipole is placed in an electric field as shown. In which direction will it move? Fine. So the options are like this. Towards the left, as its potential energy will decrease. Towards the right, as its potential energy will increase. Right. Now towards the left, as its potential energy will increase. And again the last one, towards the right, as its potential energy will decrease. Okay. Now what happens? Let us try to see here. Alright, so this was actually there, this was present in your NCERT, I am sure of it, right. So you have studied about dipole kept in a non-uniform electric field, it is there in your NCERT book, please take a look at that. Alright, nevertheless you can analyze this easily, right. Now if you observe the electric field lines, right, so wherever you find the field lines more dense, right, so there the magnitude of the electric field will be more compared to the region where the electric field lines are not that dense. So if you go from left to right, if you observe this electric field diagram here, electric field lines here, right. So the electric field lines, right, its density of the lines I am saying, right. So it is decreasing when you go from left to right. Okay, that means that your electric field magnitude, right, it is decreasing when you go from left to right. Okay, the magnitude of the electric field decreases when you are going from left to right. Okay, so notice this. So you have this positive charge of the dipole which is in a region where you have more magnitude compared to the place where you have kept the negative charge, right. So this positive charge experiences more repelling force compared to the less attractive force experienced by this minus q, or you can say this minus q is, let me redraw the dipole here. So this is positive charge and this is negative charge, right. So force is acting on this, right. So force is acting on this, say plus. Now there is a force acting on this minus, you know, F is given as F is equal to QE. So force experienced by a charge when it is kept in the electric field, same thing. Okay. So clearly you can see that at this point, right, the magnitude of electric field is more compared to this point. If you have understood this concept, you would have been able to answer this question. So the net force, right, it is acting in the right direction, towards the right. Okay. And speaking of potential energy, right, whenever you set anything free, right, so when a system is interacting between themselves, obviously there has to be some source for this electric field, right. So take it as a point charge, right. So take it as a point charge. So you say these force, the electric field lines which are this way, they are not shown. Okay. So if you keep it like that, right, any system, it will always try, that is if you are not interfering with it, that is only if they are interacting between themselves. Okay. So the potential energy, it will always try to take a minimum value. Okay. So minimum value in the sense, if you release this dipole in this situation, it will always try to go away. So if it is going away, obviously, right, because they are interacting between themselves, right. So as I explained you here, the net force is acting towards the right. So the dipole will start making its journey towards the right, and obviously its potential energy will decrease. Okay, potential energy will decrease and it is going to the right. Okay, towards the right as its potential energy will decrease. Okay, so this is easy to moderate, you can say. I am sure about 70% of the candidates would have answered this question. Okay, so this is it for coming to the physics part. There are few questions that I have not taken. They were pretty simple. I think you had one more question from geometrical that is ray optics, right? So where you had to you were given with a convex lens and a concave lens, right? And they asked about this dipole moment, correct? So something related to this dipole moment of a polar molecule, right? So what are polar dipoles? They have permanent dipole moment. They do not have permanent dipole moment. They'll get dipole moment when you place it in an electric field like that, okay? So these were quite simple. Now, like I said, you have to solve to get the answers in this 2021 session at least for your NEET exam, right? It was not easy like your last year paper, fine. So the difficulty is going to get increased, all right? So because COVID situation is coming down, all of you know that, right? So please prepare well, all right? And I wish you all the best if you are trying to write it for the next year, right? I hope all of you get good colleges, and thank you for watching this video. Subscribe to Extramarks, right? And keep learning, never stop learning. Thank you.